appreciate the work that we're doing here on Standing for Truth, please hit that subscribe button because we are just getting started.
All right, it looks like we are live. Hey, Ramat and I are extremely excited for this interview. I got to say, it is an absolute privilege to interview Dr. Rob Stadler today on this very important topic. That being said, Rob is no stranger to this channel. You would have seen him in an interview with John Maddox, as well as one of my personal favorite streams on this channel, Educating Professor Dave. I do want to pull that up on the screen for everybody. Anybody who has not yet seen it, please do check it out. It's definitely a must-watch. Share around. We definitely want to get it out there for as many people as possible to see. And also check out Dr. Stadler's interview with John Maddox. Definitely worth your time. So anyways, let's get right into this interview though. First, I want to thank Dr. Stadler for being here and giving us his time for this. Whether you are new to this topic or you have been studying these issues for years, you are sure to learn something today. I'm going to hand it over to Ramat to introduce our guest. Ramat, the floor is yours.
Awesome. Yeah, thank you again, Dr. Stadler, for coming. And this is for everybody who doesn't know. Rob has been the author of two must-read books. One is titled The Scientific Approach to Evolution, What They Don't Teach You in Biology, and The Stairway to Life. Those are his two major books. If you can flash them on screen, that'd be good. If not, it's all right. But Dr. Stadler received his PhD in medical engineering from the Harvard MIT Division of Health Science and Technology. He's been a scientist in the medical device industry for over 20 years, and literally millions of his cardiac devices are implanted in millions of people worldwide. He has also been elected Fellow of the American Institute of Medical and Biomedical Engineers. He has obtained more than 140 US patents and has over 20 peer-reviewed publications. But anyway, that's enough from Standing and I. Rob, thanks again for being here. If there's anything else that you would like to add for your introduction, or if I said anything wrong, let us know.
Well, thank you, gentlemen. It's great to be here today. I appreciate this opportunity in hopes of reaching people out there. Thank you.
Great.
No problem. We are excited for this, guys. Check out the screen. And we've also got the links in the description box for The Scientific Approach to Evolution and The Stairway to Life, both must-read books, guys. So why don't we get right into it? Actually, I should say before I forget, anybody in the audience, if you do have any questions pertaining to the topic at hand, we're going to be discussing Rob's book, The Scientific Approach to Evolution. Tag me. I'll field the questions that are relevant to the topic, and I'll, whenever there's a moment during the interview, I'll be sure to ask Dr. Stadler. So let's get right into it. I'm excited, Rob. The title of your book suggests that evolution has not been approached scientifically, even though many scientists support it, and that schools are not teaching evolution scientifically. What do you hope to accomplish with this book, Rob?
Yeah, I think that question is very pertinent to what our society is going through right now. And this comes up with the election and with COVID. I think society is going through what I would call a crisis of confidence in science. It feels like a lot of society is kind of turning their back on science or giving up on it. And that comes from the way we're handling COVID, the way we deal with climate change, the way we explain evolution, we explain our origins, and how it's taught in schools. And I'm not going to get political. I think there are many reasons why this is happening. And I'm talking about kind of an increasing divide between what science is saying and what the general public seems to agree with or adopt or believe in. And this growing rift, I think, is very concerning to me. And there are many reasons for it, I'm sure. But one of the reasons that I think is a big contributor to this, and it goes back to this book, is that we don't have a great appreciation of how to apply a level of confidence to some scientific evidence that we hear. You hear it through any kind of media, you hear about some evidence, and you should be able to frame that as to how much confidence does that piece of evidence provide? Is it a low confidence kind of result, or does this really tell me something that I can trust in, and it's a high confidence result? And I think often we're getting that upside down, and we'll talk more about that as we go on here. But the book goes through how to sort of judge the level of confidence that some scientific evidence provides. And then the book applies that to evolution. And so one of my goals in writing this book is to improve the scientific rigor in the study and the teaching of evolution. And I really want to try to reduce the bias that leads us, it risks leading us away from the truth so that we can actually get to the truth. So we want to open up science to be able to follow the evidence wherever it leads, and not put the blinders on and keep us from being able to see the truth. That's the main goals for the book.
Oh, that's true. That's so true. Those blinders, man, if people could look non-biasedly at the evidence, I agree. I have a question though. I'm curious, what led you to write the book in the first place?
Yeah, it's not a book that I had intended to write. It kind of, it's my experiences kind of prompted me to do it. So it started with my career in medical research. So as you said, I've been doing medical research for more than 20 years. And in doing that, this is not something I was taught in school. You get it through the experience. But in our society, medicine is life and death. We take medicine very seriously because your life literally depends upon it. And in my career in medical research, you have to have a very high confidence level of evidence in order to get a medical product or a drug or something like that approved for human use. And that's because society demands very high confidence before I take this life and death treatment. So I got used to, in my career, what is required to produce this sort of high confidence evidence. And this is required by FDA in the United States or whatever regulatory body in your country. But it surprises me as a scientist then when I look outside of medicine, it's not very often talked about, different levels of confidence or different degrees of evidence. If you Google the term levels of evidence, something like that, you'll end up with all these articles about medicine and about how different drugs have a certain level of evidence. Maybe it's a 1A or a 2B or something, and they're graded as to how much supporting evidence there is for the benefit of a certain drug or a certain treatment. And so in medicine, it's very well established, but not so much in other fields. So I'm used to that. And then my son went to high school biology class, right? And I knew there would be, some challenges to that. But I read his textbook in public school and high school biology. And because my mind is already focused on these different degrees of evidence and the high levels of confidence that I have to get a medical product approved, when I read my son's biology textbook from that frame of mind, it really hit me. It was quite a surprise because everything in that biology textbook, which is trying to convince the students of the validity of evolution, all of the evidence presented in there actually met none of the criteria that I myself am held to as far as quality of evidence or level of confidence in evidence. And it was exactly the opposite, that they presented very low quality or level of confidence kind of evidence. And that really kind of flipped a switch in my brain. I said, I got to write a book about this.
That's awesome.
Well, you mentioned in the book that medicine has long appreciated different levels of confidence in evidence. This concept of levels of confidence or levels of evidence is not commonly mentioned in other fields. Why is that?
Yeah, I pondered that, and I think what it comes down to is that medicine is life and death. And if you go back a few hundred years in history, medicine was a mess. People were putting leeches on to draw blood out and try to balance your humors, and they were giving, having mercury as a treatment to give you mercury, and all kinds of crazy bad things were going on, lobotomies. Because it wasn't regulated, there wasn't this scientific method, and society kind of rose up and said, we've got to stop having medicine hurt us. We want medicine to help us, right? And that placed these criteria and this appreciation for good science and high quality, high confidence kind of evidence. And I feel like other fields are coming along that route, but because other fields are not really life and death and not as litigious as medicine, right? So in the practice of medicine, if you do something wrong, you're going to be in big trouble. You'll lose your license, you'll get sued. So there's a lot of pressure to get it right there, and that's driven it. And I think other fields are going to come along eventually in this way.
I can see that. Yeah, I know exactly what you mean, because as an herbalist, I don't have that rigorous thing that you're talking about. For example, if I take something like a senolytic activator, most people have never even heard what it is, but it's something that doesn't have to go through any peer review. They can just say, oh, an onion is really good for killing parasites. And you're like, oh, okay, well, how? You can't find out how because there's no actual rigorous thing going on. So do the 6 criteria you mentioned apply only to medicine?
Yeah, so we're about to go through that. Real question is, how can I see some evidence that someone's presented to me and give a quick judgment of it? Is it a high confidence, or is it medium or low? And it's a spectrum. It's not just high or low, but how do you judge where it is on that spectrum? And we'll go through these 6 criteria that are in the book of how you make that decision. Should we go through that now then? Those 6 criteria?
Yeah, I think that would be a good idea. As to Rob, how exactly can we determine the level of confidence that science provides? And I'm going to put a visual up for the audience, actually, that'll help them understand. I think that would be a good way to approach this, Rob.
Yeah, thanks. That's great. And so in my career and in thinking this through, I've kind of boiled it down to these 6 criteria, which I hope are relatively simple, relatively easy to apply. And I think, this is good for everyone to practice. Like the next time you hear a news story, some kind of scientific finding, something about COVID, whatever it is, to apply these criteria. And it gives you a feel for the level of confidence that result provides. And again, it's not black and white. Like here we have on the left, the 6 criteria to give you very high confidence. And on the right are 6 criteria, the opposite, that only give you very low confidence. It's not intended to be black and white. It's a spectrum. But working through these 6 things will help you figure that out. So I'd like to explain these by way of examples, right? So in science, you begin by asking a question. You ask a question that hasn't been answered, and then you use the tool of science to try to answer that question. So you can even tell just by the question you ask whether or not it can be answered with high confidence if the science is done right or not. So to give an example of something that can be answered with high confidence, so this is the left side of the screen. Let's ask the question. Let's say I come up with a new drug, and I claim that this drug can cure cancer, right? That's pretty relevant. It's a life and death question. It turns out if we ask a question like, does this drug cure cancer? That question can be answered if it's done right, if the science is done right, with very high confidence. And we'll go through that. And then to contrast that with a question that cannot be answered with high confidence. So an example of stuff on the right side of the screen, let's ask a question like, can the fossils found in the fossil record, are those life forms explained by a process of evolution? In other words, did evolution produce the life forms that we see in the fossil record, right? That's a question, and that question cannot be answered with high confidence. It's quite the opposite. So let's go through these in turn. So number 1 is repeatable, right? If I take my cancer drug and I need to try to prove that it cures cancer. So I could give that drug to thousands of people in a clinical study, and I can get a result, right, to show that it did help or didn't help. And maybe you don't trust me, so you can do the study. You can repeat my results. And maybe somebody in another country tries it, right? So the more the result is repeated, if you get the same answer, you build confidence, you have higher confidence, you trust what comes out of it, right? That's so obvious. Everybody agrees with that. So on the opposite side, though, here we're saying, are the life forms that we find in the fossil record produced by a process of evolution? Well, it's not repeatable. You simply cannot repeat the process that you think produced those life forms. You can't repeat the process to make dinosaurs and make all the other types of things we find in the fossil record. It's simply not repeatable. Now, some people counter that when I make that statement. They say, hey, I can find a T-Rex fossil, and then I can find another one, and I can find another one. So that's repeatable. And I say to that, yes, you can repeatably find fossils, but that's not answering the question I'm trying to answer. I mean, what you've done there is repeatably answer a question of, can I find a T-Rex fossil, or did T-Rex exist at one time? Yes, you can repeatably answer that. But that does not address the question of how those life forms, how that T-Rex came to become a living thing, right? You can't repeat that. So that's number one on the list. Number two is to have direct measurements and accurate results. So with my cancer drug, right, I do this clinical study, I give 1,000 people the drug, I can directly measure who survives, right, and who dies from cancer. It's really quite simple. It's kind of obvious who survives and who doesn't. So that's very direct. On the contrast side, back to our fossil question, what we're trying to do with that is to determine if evolution is the cause of what started these life forms, right? What created these life forms. But what we have is fossils. We've got pieces of bones. We have evidence of death, the remnants of living things. And you're trying to use those remnants of living things to try to extrapolate or try to understand what caused the living thing to become a living thing. And that's extremely indirect, right? To try to study a process of creating life by looking at death and sparse remnants of death is very indirect. And there's a lot of extrapolation. So that's quite the opposite. It's the polar opposite. Number three is a prospective interventional study. Now, those are kind of fancy words. What that means is that good science is done by designing an experiment in advance, plan it out, write it down on paper what you're going to do, and then you conduct the experiment. That gives you higher confidence if you do it that way. And why that does is because it allows you, by planning the experiment in advance, to block out anything that would get in the way, anything that would confound the results. So for my cancer drug, I can plan to have 1,000 patients receive this drug and 1,000 other patients that receive a placebo, which is a sugar pill, right? It has no effect. And I can plan the study so that the doctors don't even know which pill they've handed out. And the patient doesn't know what pill they received. It's called double blinding. And that's a way of blocking out biases and blocking out any kind of anything that could confound the answer. And I could make who gets the sugar pill versus who gets the cancer pill. I can make that a random assignment, like use a random number generator to assign that. And that helps to block out anything confounding, like, oh, what if there's more smokers in the group that gets the sugar pill, and therefore more people died in the group that got the sugar pill? That would confound and confuse the results. It could lead us to adopt a fallacy, to accept something that isn't true. So you can block all that out if you design it prospectively. And on the opposite of that, back to our evolution example, we have an extremely retrospective process. We're looking back in time, far back in time. We certainly can't prospectively turn a dinosaur into a bird or something like that. So we look retrospectively, and you cannot control anything that's going on there because it's already done, it already happened. And so you're really not sure what is influencing what you've got here. So a nice mantra I pull out of this third criteria is that if the study is done right, a prospective study can actually establish cause and effect, right? So I give the cancer drug, the effect is that cancer is wiped out. And if I do it right, I can establish that as a cause and effect kind of relationship. But if all you have is a retrospective study where you're just observing things that happened in the past, you can't possibly establish cause and effect. All you can do is sort of hint at an association. You know, there seems to be an association between this and this, but I'm not quite sure what's going on because I couldn't control it. I may not even know the conditions of what happened so far back in the past. So we're halfway through. That's number 3. Number 4 is to be careful to avoid bias. And we all have bias. I have bias. Everyone has bias. And it's good to admit we all have bias because it's a fact of being human. And that bias can confound you. It can keep you from getting at the truth. So in the example of the cancer drug, where I'm trying to determine if this pill can cure cancer, I got to avoid bias. In fact, every clinical study that I design, I have to have a paragraph or two in the protocol that talks about how I'm going to minimize bias in my study. So I already mentioned with the cancer drug, I can randomize patients to who gets the drug versus who gets the sugar pill. And I can blind the doctor so they don't know who's getting the drug or the sugar pill. I can blind the patient so they don't know. And this takes away biases, even biases that are subliminal that you don't even know are happening. And so this is why, you know, a cancer study can be done with high confidence. You get that stuff out of there. On the opposite side, we have this evolution experiment, or we're trying to explain how life forms came to be. There's inherently a lot of bias there, and everybody comes to that with their preconceived belief. And in that particular question, I think the influence of bias can actually be greater, more influential than the actual data that you can find. The evidence that you dig up can have sort of less of an influence than the bias. The bias predominates and can blind us from the truth. And you can't block it out. You know, you just can't do that with that kind of a study. Number 5, we're almost there. Number 5 is to be careful to avoid assumptions. So assumptions are ways, they're kind of like shortcuts, you know, or ways of filling in for something that you don't know in a study. And sometimes you're trying to save money, you're trying to make the study go faster. So you say, hey, you know, why don't we just assume that smoking cigarettes doesn't matter in my cancer study? You know, and that would be a bad assumption. But assumptions are always made to try to make it faster or cheaper. So you really want to avoid those assumptions. And a well-done clinical study on a cancer drug, you know, if they have any kind of assumptions like that, they're going to list them out and be candid about them. They're not going to hide them. But they design the study so that you avoid assumptions. On the other side, with our question of evolution and the fossil record, there's many assumptions baked into this. And a couple of them that are my favorites are that, you know, if you're digging through layers of sediment, and in a given layer from a certain age, if you don't find a particular kind of life form, then that means that life form didn't exist at the time that that layer of sediment was produced, right? That's a typical assumption that's made. You can't really prove it, but you assume that's a fact. But then some evolutionists will actually make the counter assumption, and they'll say, well, we know that there are very few transitional fossils, right? And if I can't find anything transitioning to become a bat, for example, and that's kind of a fact right now, nobody has found anything that seems to have been a transitional form to get to being a bat or a fish or whatever. They assume that just because you can't find that transitional form in the fossil record, that doesn't mean it never existed in the fossil record. So those two assumptions are actually contradicting each other. But there are many assumptions going on when you try to interpret what a bone is doing buried in the ground like this. And that's dangerous. That leads to low confidence. And then finally, the last one. The last one is a little bit of a different criteria, but this is really important because in science, eventually you have to communicate what you come up with. You got to share your results and publish it or communicate it somehow. And how you frame it, how you describe what you came up with is so important. And you can tell just by the way people summarize their results, if they're overstating the confidence that they found, if they're stretching the truth even. So when I finished my cancer drug study and I published the results, maybe I would say something, if I had a conclusion like, this drug cures cancer in all humans, every kind of cancer, and I'm kind of slamming my fist on the table saying how awesome it is. You could tell that's probably not exactly the case. And then you read my study and you find, oh, I only studied thyroid cancer, and I only studied females, and I only studied people in the United States. So if I'm going to present my results with sober judgment, the concluding line should be something like, in females in the United States who have thyroid cancer, this study suggests that this drug is effective in curing cancer, right? That would be a sober way of concluding what I've actually done and what you can take home from it. The other result where I say, hey, this cures all cancer in all people all the time is an extrapolation. It's overstating confidence, and it's kind of going beyond the scope of my study, and that's really bad science. And you see that a lot when media picks up on results, and they kind of amplify and hyperbole and stretch the truth a little bit. So number 6 criteria when it comes to this fossil record, our biology textbooks, they'll say with great confidence in the statement that all living things have common ancestry, and the fossil record proves that, life forms descended from each other through common ancestry through the process of evolution. And they don't put any kind of hedging in there, like we think so, or evidence suggests that, or, they state it with very high confidence, which is an artificial kind of confidence, and it's bad science. And that deeply concerns me. So the next time, just summarize this, the next time you hear some evidence on some media, some paper you read, whatever it is, you can apply these 6 criteria to what you see and get a feel for the level of confidence that result provides. And that's really one of the take-home messages from this book, is to be able to do that.
Well, I think those were some great points, and I can't recommend your book enough. And it's true, they like to treat, for example, the fossil record as high confidence science, right? And for all the points that you brought up, Dr. Stadler, this is precisely why I prefer high confidence empirical science. I like the way you put it. We like to avoid bad assumptions. We like that which is measurable, as you said. When it comes to drugs, we can find a solid result through repeated studies and, say, giving the drug to thousands of people, where we can build confidence in the science, as compared to, and I like the way you put it, as compared to fossils. They're not repeatable. We can't repeat this process that produced these life forms in the fossil record, right? I think this is why, Rob, I find evolutionists, when I interact with them, they often like to hang out in the fossil record in terms of arguments, because it's so low confidence. There's so many stories and interpretations that can be put forth based on such indirect evidence, right, as compared to that which is direct, as you stated. So great points.
That's right. And you can make whatever story you want. And because the evidence is such low confidence, you can argue forever and ever and ever and never get to a resolution. And also, because it's low confidence like that, it can be very difficult to falsify. It can be difficult to prove that you're wrong.
Right.
If I pull out a bone and I say this bone is transitional between dinosaurs and birds, it's pretty hard to prove that wrong. And I can make up a story. And if you can go back to the screen for, I want to give a nice sort of object lesson to it.
Of course.
And it's important to know that some fields of science cannot really be practiced with this high confidence, these criteria for high confidence, right? If I want to study, let's say I want to study if living next to a nuclear power plant decreases your intelligence or your health or something like that, right? To do that right, to do that with high confidence, you would design the study in advance and you would assign some people to live next to a nuclear power plant, some other people you assign to live far away, and you study them throughout their entire lives, and you control everything else so that they live the same life, except, the same food and the same hobbies or whatever it is, except some people live near the nuclear power plant and some don't. That way you could get a very confident answer, right? But of course, that's just not ethical. It's not practical. It's not going to happen. So you can't study that question with real high confidence. And that's okay. That's the best you can do. But just don't try to tell me then when you get your results, don't try to tell me these are high confidence results, because to be honest, to be humble about it, you've only gotten low confidence results in what you've done. Now, if you're stuck there with low confidence science, the analogy I like is a drinking straw, right? This is a simple drinking straw.
Yeah.
So imagine that you have to referee a football game. It's now football season, so you can picture yourself being the referee. You're under a lot of pressure. You got to catch the play. You got to catch people who are cheating, right? Whatever's going on down there. But you're the referee. And now in a low confidence world, instead of being able to watch the game with your eyes, you have to close one eye and you can only look through the drinking straw with the other eye at the football game, right? And so you can see a little bit of what's going on, you get kind of a foggy picture of a little bit of what's going on there. And that's what low confidence science provides. And you have such a hard time to see the big picture and arrive at the truth. That's what it's like.
Yeah, I really love that analogy. The second I read it in your book, I thought, this makes such sense. It makes a lot of good sense that's especially with the fossil record. They're not looking at the bigger picture. It's like they've got a straw and they can't see anything in their peripherals. I like the way you put it. I guess if they were to, because the way you said it was, it's oftentimes the best they can do. It's the best they got. So if they were to just humble themselves and admit, okay, this is the best we can do with, say, the fossil record, for example. Yeah, it's low confidence, low quality science, but I think it just comes down to humility as compared to the way you said it. They treat it like in the textbooks in school, they treat it like it is high confidence science. It's irrefutable,
And I also want to clarify that, low confidence science doesn't mean you're a bad scientist, you're a bad person or anything like that.
Yeah.
In a lot of cases, if you're an anthropologist or whatever, you're trying to do the best you can. And you are doing the best you can. The problem is all you've got is a drinking straw and you're trying to look through it. And that's okay. It doesn't mean you're a bad person, but just as you said, with humility, just say, hey, this is the best I could come up with. And, we can't be certain, but here's what we got. That's fine.
Yeah, and you make that point in your book, so I really like that. Okay, we're getting a lot of good feedback from the audience on that. And this next question has actually been a question in the audience. So, Rob, do the 6 criteria apply only to medicine?
Yeah, thanks. I get that question a lot. And I try so hard to explain that they apply to all fields of science. These 6 criteria you can apply to any field of science of any kind. And I think they're equally applicable. It just so happens that medicine, is able to do high confidence science. And other fields of science may not, like anthropology, may struggle, but it still applies. It applies to all science. In my book, I go through, the example of just simply studying how fast an object falls when you drop it, and that can be studied with high confidence science. It has nothing to do with medicine, and it applies. So it's on all fields of science.
Yeah, well, I like what you said earlier about how it makes it nearly impossible to falsify, right? Because remember, the slow Darwinian process of life evolving slowly. Well, when they find something that didn't evolve, boom, it's stasis. It's no big deal. Or when they find something out of place that shouldn't be there, oh, it's punctuated equilibrium. It jumps forward massive amounts of time. So it makes it unfalsifiable. That's not very good.
Exactly.
You mentioned in your book, specifically on page 52, you talk about a TED Talk with paleontologist Jack Horner about finding and naming new species of dinosaurs that show a common error when we rely on this low confidence evidence. Can you explain that for everybody, please?
Yeah, I would recommend people watch that video. You can look it up on YouTube, Jack Horner TED Talk. But what's fascinating there is that you have paleontologists, right? And a paleontologist is trying to build their career like anybody, as a scientist, trying to make a name for themselves, trying to make a discovery. And so they discovered dinosaurs. They dig up something and they say, well, I dug up something and it doesn't look like the other dinosaurs that have already been named. So it's something new. It's a new species, and I'm going to name this thing and put their stamp on it, write a paper, and become kind of famous. So Jack Horner looked at this, and he realized that all the dinosaurs that are out there, the fossils, were claimed to be, they were adults, basically. And he actually looked at the dinosaur bones and was able to get permission to drill into the skull. And apparently when you drill into the skull and you look at the bone, you can determine the age, the maturity level of the animal when it died. And so he was able to piece this together that an infant, a toddler, an adolescent, and an adult dinosaur of the same species were named for different species because the adolescent didn't look like the adult and the infant didn't look like the adolescent. So people were naming them as different species when in fact it was the same species, just different ages. And he said this happens because people like to name things and they like to be successful. And basically what it comes down to is that bias, that you really want to discover something new. You want to be famous. And so here it is, I found it, I discovered it. And that bias can lead you to adopt a fallacy, right? To believe something's true that isn't true, all in the name of science. And so knowing that that's happened, and it's cool that Jack Horner was able to clarify and get to the truth in this case. But knowing that can happen, knowing that dinosaurs can be thought to be separate species when they really aren't, then if someone makes a statement like, through the fossil record, this transition to this, transition to this, transition to this, right? That's the story that we're told. How do we know that that's not just bias, right? In the same kind of sense. How do we know it's not the same thing going on there? And it's all low confidence, and it's kind of cloudy. You're looking through the drinking straw. It's pretty hard to get to the truth.
Well, I find that amazing, Rob. I've mentioned that specific case a number of times now after reading that in your book, that specific case. And as you put it, there's a lot of pressure in this field to find that next big find, right? Because not only that, I've read a ton of papers and watched some lectures on the hominid fossil record itself, and there being numerous cases of mixed bone beds, and then as a result, chimeric or artificial species where you've got a mix of like Australopithecus bones and human bones, and the paleoanthropologists are admitting this. So it's just the nature of what you're saying, right? It's just the nature of it being low confidence.
And it's okay that that's what it is, as long as you openly discuss that, hey, these are the assumptions we've made, here's some limitations to what we've done.
Yeah, I think those are great points. Exactly. I completely agree. So, Rob, you mentioned earlier your son's high school biology textbook. What evidence was presented for evolution, and how did it stack up against the criteria for high confidence?
Yeah, so, we have to start by recognizing that in the public school biology textbook, sadly, it's not so much about science as it is about salesmanship. It's like when you go to the car dealer and you pick up a pamphlet about some car, right? It's going to talk about the great qualities of this car, how awesome it is and all that. It's not going to say something like, well, it's true that it doesn't get the best gas mileage. And yes, we've had transmission problems in the past, but it is a pretty good car, right? It's a sales argument. And so the biology textbook expectedly is, unfortunately, a one-sided argument that's trying to convince kids about evolution. But when I look at the evidence that was presented in there, and I've seen many biology textbooks very similar, but they largely rely on four pieces of evidence. And first is the fossil record. So we've talked about that. It's very much low confidence. Next one is homology, which is to show that across different types of living organisms, we have similar genes or similar bone structures, like we have 2 long bones here and 5 phalanges and so forth. And you'll see that in humans and in bats and in fish or lizards. So that's homology. The third would be vestigial organs or bad design in living creatures. That's an argument. And the fourth one is biogeography, which is how different living organisms are dispersed around the planet, or some only live in certain regions. So in the book, I go into all the details. We're not going to do it here, but each of those 4 types of evidence, each of them fails all 6 of the criteria. So it's not just lowish confidence. It is kind of like the lowest confidence of evidence in each of those that the biology textbook praises as being convincing. Unfortunately, that's the situation we're in, and that's what they emphasize.
Right.
Great answer, Rob. A couple of relevant questions that, before I miss them, because once there's too much going on in the chat, I'll miss the question. So this one came in the form of a super chat. Doki Doki Bible Club, thank you so much. Asks, is RNA world high or low confidence? I think we know the answer to that one.
Well, good question. So first, RNA world this is relating to origin of life, right? To get the first living thing started. And of course, cells are extremely complicated. And so there's a hypothesis that life began first with just RNA, because RNA is able to be a catalyst like an enzyme, and it's also an information carrying molecule. And that's why there's some excitement about RNA world. But if you look at those 6 criteria, right, we can go through it real quick. RNA world is not repeatable, meaning we can't go back before there was life and the conditions that existed and try to repeat an RNA type world. You can try to do it in a lab today. You can do it in your little flask with your chemicals and your hoods and all that stuff. And you can try, but you're not exactly repeating what happened back then. You can't be sure of what happened back then. Number 2, it is very indirectly measured. I mean, we can't even go back in time there. We don't even have any real fossil evidence from supposedly 4 billion years ago. Nothing is left from back then. So it's extremely indirect. It's extremely retrospective to go back, unless you do it in the lab. And if you do it in the lab, you've got tons of assumptions, which is number 5, many assumptions required. There's clear opportunities for bias, because the people who tend to promote RNA world are trying to promote a naturalistic explanation for life. So quick answer, it is very low confidence, yes.
Great answer. And one last question here that is relevant to the point you just made on the textbook examples. Question here, org 343. Should those 4 textbook examples be more thorough or removed?
Well, great question. I think they should be explained with a sort of even-handed, unbiased statement that, hey, there may be some hints here. Because this is low confidence evidence, there's some hints, and it's possible to interpret those as supporting evolution. It's also possible to interpret it a different way. But what should be done, though, is to take that low confidence evidence and deprioritize it and then take the high confidence evidence and prioritize that in its place. And we haven't gotten to it yet, but there actually is some quite high confidence evidence and experiments that speak to evolution, and those should be put up high in their place. If I go to FDA with my cancer drug and I say, hey, I've got a randomized controlled study of 2,000 people, blah, 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 I did everything right and it's high confidence, and here's what that result found. And, oh, by the way, I actually also did a retrospective observational study where some people bought this stuff on their own choice and ate it as a dietary supplement, and it looked like it kind of helped. You know, if I do that, the FDA would just blow off the low confidence results, wouldn't even look at it, wouldn't even think about it, and they focus on the high confidence evidence. And that's what we should be doing when we talk about evolution.
Absolutely. Well, given that your book is rather discouraging, to say the least, to those who believe in evolution and molecules to man, it may be surprising to some of our viewers that your book has a chapter called High Confidence Evidence for Evolution. So what is exactly in that chapter?
Great. Yeah, this is where there's a twist in the plot. So there actually is a fair amount of data, and it's building every day, of evidence that meets all 6 of those criteria that teaches us about evolution. So let me give one simple example, right? Is often cited is evolution of dogs, right? So we know over the last few hundred years that wolves, through a process of artificial selection, have given us this great diversity of dogs that we have today. And it's pretty cool, you know, incredible diversity of the types of dogs. They're all kind of the same species because they can interbreed, but it's pretty awesome. And that's what, you know, Richard Dawkins makes a big deal of that. And if you look at that study of dogs coming from wolves, and you look at the 6 criteria, you can argue that it meets all 6 of those criteria. You know, you could do it today, take a wolf and start breeding and artificially selecting, and you could end up with a Great Dane or a Golden Retriever, you know, over hundreds of years. You could do it, you could repeat it, and you can meet all 6 of those criteria. So that's high confidence evidence, but it's high confidence evidence of, you know, what you call microevolution. So in the book, I love this quote from Richard Dawkins. You all know Richard Dawkins. And this is from The Greatest Show on Earth, which is his book about evidence for evolution. And he talks about the dogs, right? And he says, this is his quote. He says, it's on page 82. He says, in 82 in my book, he says, if so much evolutionary change can be achieved in just a few centuries or even decades, just think of what might be achieved in 10 or 100 million years. So what he's doing there is asking you to extrapolate. He's saying we have a result here, which we don't argue about, you know, that lots of changes have happened in 200 years. He's asking you to extrapolate that for 10 or 100 million years. That is something that nowhere else in science is that allowed, where you can observe something here and extrapolate that 10,000, 100,000, a million fold down the road and have any belief or any confidence in that result. It's unthinkable in science that you could extrapolate like that. Now, if you look at those 6 criteria of high confidence, the dog coming from a wolf, you can meet all 6 of the criteria for high confidence. In that quotation from Richard Dawkins, where he's asking you to imagine going forth 10 million or 100 million years into the future, that statement immediately contradicts all 6 of the criteria. He's asking you to go from high confidence to the absolute lowest of confidence and to trust him that that's what's going on here. And the ultimate irony for me is that in creating that, in that statement he made, which is just unthinkable bad science, what's ironic to me is that before Richard Dawkins retired, his job title was the University of Oxford's Professor for the Public Understanding of Science. So he was recognized as being the guy who's going to help the public to understand science. And yet he did something like that, which is extremely unscientific, very disturbing. So that's just one example of high confidence science that speaks to evolution. Let's go through a couple more. We have Richard Lenski's experiment. Some of you, probably most of you, are familiar with that, right? He's up to like 72,000 generations of E. coli in a flask. This is done repeatably because he's got 12 flasks, and you can also freeze and thaw, parts of the experiment. You can repeat what happened. It's very repeatable. It's directly measurable. He can look at the DNA at the beginning, look at the DNA 70,000 generations later, and directly determine what's different about the DNA. It was prospectively designed. I don't think there's much bias or assumptions, so forth. This is very high confidence science. It's very well done. And it tells us, what is evolution capable of. And one more case that I really, really like this case. It was done by Ann Gauger, published about 10 years ago. This is an example of high confidence science applied to evolution. And in this case, they took E. coli, and E. coli is able to manufacture an amino acid called tryptophan. It has an enzyme that produces the tryptophan, which is great. So Ann Gauger said, hey, let's do an experiment. I'm going to intentionally damage that gene. I'm going to intentionally damage the gene so it produces an enzyme that's half, broken. It's not very good at producing the tryptophan. Put that back in the bacteria. Then I'm going to put the bacteria in a bath, in a flask that has only a limited amount of tryptophan. And so it better be able to fix itself in order to survive and grow, right? Now, the way they damaged the gene was just changing one nucleotide in the DNA. They changed one nucleotide, which changed one amino acid in the enzyme. So a very minor damage. And they found that after having about 1 million bacteria produced, no, it was like 100 million bacteria produced, that actually a random mutation actually flipped the one nucleotide back to the way it should be. And the enzyme was fixed. And the bacteria could produce tryptophan, and it was able to grow rapidly and survive and dominate. So there you have in action, all right, high confidence, because it can be repeated, it can be directly measured, blah, blah, all 6 criteria. And you have evolution to change one nucleotide to make a better bacteria. Okay, that's cool. Then they did the next step, and they said, okay, let's challenge it with 2 nucleotides changed, right? They had the first mistake, they added that, they added a second change to that. So now there are 2 nucleotides changed. That produces a tryptophan synthase, the enzyme, that is completely broken. It doesn't work. But all the bacteria has to do is change 2 nucleotides to make it fully functional. And in fact, if it changed the first one, if it fixed the first one, it's somewhat functional. It's a step in the right direction. And then it needs to change the second one to be fully functional. So this was a test. Hey, evolution, I'm giving you a test. Can you solve the test? So they put that in the bacteria, put the bacteria in a solution that contained a small amount of tryptophan, and they went through 9,300 generations of the bacteria. 9,300 generations, and something like 1 trillion bacteria organisms formed through reproduction. So there's plenty of opportunity there in 1 trillion organisms to have a fortuitous mistake to make it better again. And they found not only did the E. coli not fix the problem, so it was not able to repair the tryptophan synthase enzyme. Not only was that the case, but in the 15 flasks that they tried, the repeatability of this, some of the flasks just jettisoned the gene entirely, or they inhibited production of the enzyme because they're better off. The bacteria were better off not even trying to produce that enzyme because it's a waste of energy. It actually takes tryptophan to make that enzyme. So they're burning their tryptophan they do have. So it was easier to just give up rather than try to fix those two nucleotide errors. So this gives you a feeling of the bounds of how far evolution can go. You know, Mike Behe describes it as the edge of evolution. How far can evolution go? And this was a prospective test to see how far can evolution go, and it failed the test. And this is high confidence. It meets all 6 of those criteria. So there is high confidence evidence that addresses evolution, and it's way above the kind of evidence that you'll find in the biology textbooks.
Yeah, that's awesome. So many great points there, Rob. It would take us all day just to kind of touch on all of them. That was an answer everybody needs to hear. Very important. Just like Richard Dawkins. Most evolutionists, they commit an extrapolation fallacy. I like the way you said that what they're doing is they're extrapolating the data. Just imagine, all these small micro evolutionary variations over millions of years. Imagine what could result, fish to fisherman, right? But what I find funny, Rob, is the fact that the changes we see are typically downhill and reductions in allelic variability. Like you were talking about with Lenski's experiment. What Lenski has actually observed at this point, from my understanding, is that his bacteria overall have shrank in functional genome sizes. There's been no real uphill changes. So what comes to mind is, okay, you want to take that extrapolation fallacy back millions and millions of years. Well, if all we see is reductions in information, loss of information, well, that's not really going to take your fish to fisherman. So their high confidence science doesn't help them at all, would be my thoughts. So a lot of great points there, Rob.
And I believe that's why they end up kind of de-emphasizing this high confidence evidence, and they emphasize the low confidence evidence and put that first and foremost. It's all backward.
Great point. Really good point. Okay, in the next chapter, Rob, you apply the findings from high confidence evolution experiments to the hypothesized evolution of humans from primates.
Yeah, so here we kind of get to the punchline, right? So if you do it the right way, which is to de-emphasize the low confidence stuff. So let's get rid of talk about the fossil record and homology and vestigial organs and just push that aside. Let's talk about taking these high confidence studies like Lenski, like the one I mentioned about tryptophan with Ann Gauger, like dogs. There's other studies on fruit flies. And we know how much evolution can accomplish in 70,000 generations. We know how much it can accomplish and what it can't do. So let's put this to practice now. And let's talk about humans. Did humans evolve from primates? Right? And it's time to apply that. This is kind of a reality check. So humans and chimps, we're told, have a common ancestor that's about 6 million years ago. The problem is that humans and chimps are very slow to reproduce. We have 2, 4, 5 offspring, and we do that in 20 years or 15 years, maybe 12 if you're really fast. So in 6 million years, you could tell how many generations are out there. And you can also guesstimate how many humans have ever lived ever, right? You can guesstimate that. And it's nowhere near how many bacteria that I can study in an experiment in my lab. I can easily surpass the number of humans that ever lived, just doing Lenski's experiment. He's got trillions and trillions of bacteria in his study, and he's got 72,000 generations, which if you do the math, that's almost 2 million years of human generations, right? So you just take what Lenski has learned and apply that to humans coming out of chimps. And I think that's a very interesting result because, you've had this many times on your show already, but we're all told that humans and chimps are 98% the same DNA. And I know you guys have gone through this many, many times, but with with the latest genome sequencing of chimps, I think it's called Pantro 6, the true number of the agreement between human DNA and chimp DNA, I think, is now in the mid 80s, something like 80%, certainly not 98%. But it almost doesn't even matter, because even if it is 98% similarity, and we have about 3.2 billion nucleotides of DNA, you're still talking about tens of millions of nucleotide differences between us and chimps. And we also know what's called orphan genes. I think you guys have been through this many times on your show, but every species has orphan genes, which are unique genes to them that no other living thing has, that make them special and unique, right? I think human orphan genes are going to give us big brains, consciousness, ability to speak and communicate. So there's a lot of interest in these orphan genes that we have and chimps don't have, nobody else has. If you just look at those orphan genes, you read the papers about this, there's something like 1,400 orphan genes that we have that chimps don't have. So when you add this up, you're looking at, you need to come up with 1,400 new genes, you need to come up with tens of millions of nucleotides of DNA between us and our common ancestor with chimps in 6 million years. And then we look to Lenski's experiment, this one by Gauger, and all this other high confidence science of evolution. And you end up with a huge, what's called Haldane's dilemma, or the waiting time problem, that there's just not enough time. There's not enough generations. There's not enough human beings ever that existed to accomplish the kind of changes that would be required.
Well, so many good points there, Rob, and some of our favorite topics. You're right. We could probably spend a couple hours on each point. And I constantly hear on that last one, the waiting time problem, you'll hear the critics, constantly repeat that, oh, it's been solved. And I typically respond with not even close, because from my understanding, in addition to the single letter differences, you're looking at, I've read about tens of thousands of chromosomal rearrangements would have had to occur, spread, and also become fixated as well. And tens of millions of base pairs of insertions and deletions. And what we know about the rates of fixation, how slow they are, and most new variants are lost. This is a huge, huge waiting time problem that I think a lot of the critics, or those that just kind of accept that type of evolution, they don't really think about these problems. So I'm really glad you brought those up.
Yeah, and it's easy to get single nucleotide changes. Those kind of like salt and pepper, they just pop up all over the place generation by generation. But if you need specific changes to occur in specific places in order to make a new function, or to come up with an entirely new gene that's never been produced before, we just know that the probability of that is just astronomical. And humans are so slow to reproduce, and there's so few of us in relative terms compared to a bacteria, that it just makes no sense that that's possible.
Yeah, great answer. One quick question before we move on is in regards to the topic of orphan genes that you were talking about. I find that to be a really fascinating line of evidence for independent origins, because I find that this is exactly what we'd expect to find, is these taxonomically restricted genes. How does the evolutionary community, Rob, how do they deal with that observation? What are their explanations? And I guess the best way to ask the question, are there explanations, whatever those are, for the existence of orphan genes? Would that be considered high confidence?
Good question. Yeah. So I think you guys have covered this a lot on your show, but there's two proposed explanations for orphan genes. And one is just random sequences that got lucky. And the second is that a gene gets duplicated, and then that gene changes its function over time until it's not recognizable anymore from its old twin, and has a different function in a different sequence. And the way they study those then is to do phylogenetic trees and to look at the DNA similarities and try to pull this together. And it certainly isn't high confidence because it's not repeatable and prospectively studied. You don't see these things. You can show me examples of gene duplications and then some divergence where the duplicated gene slowly changes a little bit, but you're not going to see it diverging so much that it's not recognizable anymore, which is what the theory would say, that they're no longer recognizable and now you have an orphan gene. You can't repeat that. You can't do that prospectively. So there really isn't much confidence to it, but it's the only thing they have, so they have to kind of grasp at what they've got.
Makes sense. Makes sense. One of the main points in your book actually is that low confidence evidence is being prioritized over high confidence evidence in the teaching of evolution. Why is this not recognized?
Yeah. Well, that's my main frustration, and that's the main point I want to convey is, like, can't we just do what rational scientists would do, which is prioritize the high confidence over the low confidence? It just plain makes sense. I think the low confidence is prioritized because, as we've said, you can make up a story and make that story agree with what you want to be true, to make it agree with your hypothesis. And you can promote that all day long, and it's very hard to falsify it. It's disturbing to me. And when I went back to our intro, when I said that our society is having a crisis in confidence when it comes to science, I think this speaks to it too, is that people have good intuition and good common sense. And if you try to tell them a story, and they don't see real confident evidence backing it up, they're just not going to believe you. And then you erode confidence in science, you erode confidence in whatever a scientist says, because they're calling your bluff. And the other part of this, there's another dimension to it we haven't talked about, and it's kind of the elephant in the room. And it goes by the fancy name of methodological naturalism. And maybe you guys have talked about this on your show too. But a lot of people argue that science can only be practiced under the assumption of methodological naturalism, which means that science can only address natural causes, that science is not equipped to speak of anything that is not a natural cause. And so we start off thinking, okay, how did life begin? There could be a natural reason, or there could be a supernatural, a non-natural reason. But if we're going to study this with science to try to get at the truth, which one of these is right? Well, science can't deal with something that's not natural. So that one's off the table, and we're left with the natural explanation, and that must be the truth. And so here you have what's called the truth-seeking argument, that if you apply science and you must have methodological naturalism, you're no longer seeking truth. All you're seeking is the best possible natural explanation. Doesn't matter what the truth is, you're going to come up with a natural explanation, and that's all you got because you've already blocked out the alternative. And that is, the way a lot of people want to practice science. And when it comes to origins, the question of where living things came from, there's only one naturalistic hypothesis, right? And that's evolution. That's neo-Darwinian. That's all there is. So it's the only game in town. Everything else is excluded. And so we have to find ways to support this. Science has to support this. And maybe that's what makes people do something that's very foolish, is to flip low confidence and high confidence evidence up on its head. So let's emphasize the low confidence because that can support our one hypothesis. And let's suppress this high confidence evidence that we don't like the looks of it. I think that's what's going on.
Awesome answer, Rob. We're getting a lot of good feedback in the chat. Really lively chat. Good audience today. Really enjoying this. Time's flying by. I've got a couple questions that I managed to save. in the brink of time before I lost it. Here's one here that's pretty relevant, Rob. Would low confidence work for 9th grade bio and higher confidence for 12th grade bio?
Well, maybe I'm not understanding that, but I'd prefer to have high confidence for both. I mean, no matter what age you are, what level of understanding, you got to prioritize the high confidence always. Put that on top, and you're very justified in suppressing. You can mention the low confidence with a little asterisk next to it to say, well, we're not really sure. This isn't very clear what it's trying to tell us. But that's true no matter what age you are, no matter what field of science you're studying.
Right. Great answer. Right here we got Anthony Maurice. This is from earlier. He's asking SFT, when he says they can produce RNA in the lab, it's not. Oh, maybe that wasn't his original question was, what does Rob mean by in the lab? Does this mean highly controlled environments? To summarize his question. Yeah,
right. So you can, I guess we were talking about RNA world at that time, right?
Yes.
Right. So, you can manipulate RNA in the lab, and we have to do another show on origin of life and on RNA world.
I think that might have sparked. I'll I'm sorry. Go ahead, Rob. I was cutting you off there.
Well, just to go along with it, you do it in the lab. It's highly controlled. It's highly contrived, and it's overly simplified because you have very pure starting ingredients. A very clean flask with very pure ingredients, probably homochiral, that you add in. And you can get some results, but whether that has any relevance to some dirty rock, 4 billion years ago, tons of assumptions, an extraordinary amount of assumptions would have to go into believing that that's at all related.
Right. Great response. Let me see another question here that came in, if I could find it. Like I said, lots of good feedback. Actually, as I'm looking for this next question, Matt, did you have anything that came to mind there, brother?
Yeah, I like I like to imagine that if I drank some coffee and my headache went away, and I'll be like, oh my gosh, I got to sell coffee to get rid of headaches and just patent it. You know what I mean? As fast as I can, because it worked for me one time. That's kind of what they're doing, right? I just read a study recently. I think you'll find funny. They believe that human males grow beards because of being punched in the face. That's the evolution of beard. And their theory on this was based on that if you wrap a bone in sheep wool and drop weight on it, it doesn't take as much damage as if you don't have wool wrapped around it. What kind of confidence do you think that would be in that theory?
It is. It's laughable. It's interesting. You can make any story you want to make, and it's surprising how much of that ends up being published without scrutiny. It's quite shocking.
Yeah, that was published. That's peer-reviewed, published.
Well, Matt, if that's the case, then you should have a huge beard.
Man, you think.
He gets punched a lot.
All of that cage fighting. So a question here that came in, but I can't seem to find it to screen share it, has to do with the high confidence, low confidence science. Rob, is that a new term, or is this something that's always been recognized? Was the question in the chat?
So that specific term and these specific 6 criteria, they're new in a way, but it's kind of rehashed. I mean, if you Google levels of evidence,
yes,
you'll find it's all related to medicine, but you'll find in medical terminology, you'll find the kind of criteria we're talking about in these 6 criteria. It's not the same, but it's out there, and medicine most certainly appreciates these things.
Well, it's really, your book was really fascinating and helpful to me, because I've always been on the side that, it's the direct lines of evidence that we should be using to answer these really important questions on evolution, ancestry, creation, and genetics being a really, really direct line of measurement, being high confidence science. And as you pointed out, with the changes that we see in, say, dogs or the changes we see in these experiments where you've got thousands and thousands and thousands of generations of bacteria. And all you ever get is, in those experiments, more bacteria and oftentimes more degenerated bacteria. Do they have any real solid examples in that realm, though, that could be considered high confidence for that type of evolution? Maybe like a true gain in novelty? Or is that what Richard Dawkins, he kind of put forth what he thought was the best one in the Lenski's experiment, correct?
Yeah, it's kind of sad that in his book, The Greatest Show on Earth, it's on page 130 of his book, he interprets Lenski's experiment, and he made a bold conclusion about it before they actually had the DNA evidence. So there was a big change. I think you guys all know this. In Lenski's E. coli, there was a big change that occurred where the E. coli could metabolize citrate.
Yes.
Where normally they couldn't do that in the presence of oxygen, right? And before they had the DNA data, Richard Dawkins said, this is a quote from his Greatest Show on Earth book, he said, not only does this show evolution in action, not only does it show that new information entering genomes, not only does it show combinations of new genes, right? So he's sort of dreaming from his paradigm of what had happened in Lenski's bacteria. And it was about 6 or 7 years later that the actual papers came out with the DNA sequencing that showed, as you've covered on your show many times, that it was simply moving one gene to a different promoter and then changing how much another gene that existed already was amplified. So there was no new information. It was just a little gentle scrambling of some of the information that's already there, which actually made the bacteria less successful in the normal environment, but more successful in this particular environment. That's what happened. And it's very irresponsible for Dawkins to make such a strong, pound the table with your fist kind of conclusion based on his bias and his paradigm before we actually knew what actually happened. Now that we know what happened, it's pretty unexciting. So I think your question actually started with what's the most impressive, new information, new gene kind of thing that we've seen with high confidence, right? And you often hear of this nyelinase enzyme, so a bacteria that evolved to be able to eat nylon, right? I think it happened in Japan near some chemical factory. It sounds like a horror movie or something. So similar to Lenski, that was initially thought to be a whole new gene, a whole new protein with very creative, different design to it. And I think over time that's been proven to be some pretty modest, just a few changes in the DNA sequence that resulted in this ability to eat nylon.
Yeah, it's such a great answer, because when I first got into this and I started reading their literature, just even talking to people of the other side on the evolutionist side, they'd provide these examples. But then when you further look into them, what you see for the most part is largely reductive evolution. And like you said, even with that advantage seen in Lenski's experiment, the best you're seeing is an increase in fitness in a very narrow sense, because the rest of these genomes are decreasing in functional genome capacity. Or I just love that Lenski experiment, because I've read exactly what you quoted, Richard Dawkins saying the same thing, and yet all you see is a loss of genes. I find Lenski's own data is revealing clear evidence of genetic degeneration. So that was a really good answer.
And recently it's been published that the bacteria that develop the ability to eat citrate are actually getting pretty sick right now. They're kind of falling apart over time.
Yes. Yeah.
It's not good.
That's a really good point. I believe that was shown in a 2020 paper, which means that's as up to date as you can get for Lenski's experiment. So if what we're seeing, according to Richard Dawkins, that this is one of their best lines of evidence for that type of evolution that they believe in, that they want to extrapolate and yet that those adaptations are just accomplished by means of what? Loss of function, loss of regulation mutations. Now they're sick. I mean, that is a really good point.
And I think this is leading to this crisis of confidence in science. You know, if the public is losing confidence in what scientists are saying, it's because of things like this. You know, you got people slamming their fist and saying stuff that turns out to be wrong. And you can tell it's all coming from their bias. It's very disparaging.
It is. I couldn't agree more. And I could not recommend I couldn't recommend both your books. I recommend highly for everybody. Today, we just focus primarily on the scientific approach to evolution. So I can't recommend that one enough for the audience. It really helped me tremendously and put things into perspective. But the Stairway to Life, an origin of life reality check, hopefully we can have you on again in the future, because that is another one that we could easily talk about for another hour and a half. So, did you want to briefly, maybe in a few sentences, give everybody in the audience a brief summary of your book that we've talked about today, Rob?
Yeah, just a quick wrap up. If you're in the elevator and you have 30 seconds with somebody, you have to tell them something really quick, right? So I would say, simply that the evidence that's commonly used to support evolution, it actually fails to meet these simple criteria that tell us how much confidence we can have in science, right? But there is other evidence out there, these high confidence evolution studies, and those meet all of the criteria. And that evidence tells us very much that we're talking about a very constrained process that's only capable of taking a few steps in one direction, but certainly not capable of molecules to man.
Yeah, that's really good. And I want to thank the audience too. They've had a lot of good questions. And Dr. Stadler, there's one final question I have here from the audience, actually, that if you don't mind, I can ask right after this wrap up. And this would actually culminate all the questions that I've had. So this question was from Lena Powell. And she asks, has Dr. Stadler seen a computer evolution simulation? And does he think that using them as proof, quote unquote, of evolution, would that be circular reasoning?
Well, computer simulations, you really can get them to do whatever you want. They can be useful to provide some insights. I would say what they come up with can be hypothesis generating. In other words, it makes you think of an idea that, hey, we should test that in an actual experiment. I don't put a lot of trust or confidence in what they come up with. On the flip side of that question, John Sanford has produced computer simulation he calls Mendel's Accountant, and that's his simulation of it, which shows very constrained progress that evolution can make, which opposes what some other simulations have said, that they can come up with all kinds of stuff. So it's not very high confidence because it is a simulation. And of course, it also takes human intelligence to build the thing and to make it work.
Right. Great response. Yeah, no, that's Mendel's Accountant and Dr. Sanford's work on that is really amazing too. Well, I mean, we've had you for an hour and a half. This was awesome. Time has flown by. I've really gotten a lot out of this, and I really appreciate your time. Rob, the audience has loved it. Tons of good questions. We could probably have you here all day. What we'll do is we'll wrap it up here. Unless you had any final things to say, Matt, before we...
No, I just hope we can get Rob on just for a random Q&A sometime for the audience. That would be...
you can also, you could send questions to me. I have an email account that's scientificevolution.com@gmail.com.
Okay.
A little complicated, but scientific evolution, one word, scientificevolution.com@gmail.com. Send questions.
Awesome. Okay, well, I've got both Rob's books in the description box. Please check them out. Like I said, I flew through the scientific approach to evolution. I couldn't put it down. So it was a real blessing and a privilege to have you on today to discuss that topic. I think it's really important, and I'm really excited that our audience here got to hear all this incredible information. So everybody in the audience, share this video. Please get this out there to as many people as possible. It is important that they hear these things being discussed. So I want to hand it over to you, Dr. Stadler, for any final words, any final thoughts, and also thank you again so much. I really enjoyed this.
Great. Just thank you, guys. I really appreciate you having me on the show here and the opportunity to share, and just tell everyone to get out your drinking straw and remember this when you're reading the newspaper or looking online tomorrow.
I love it. That's awesome. Okay, guys, we are here on Standing for Truth. I'm really, really pumped for this one. Definitely excited. We've got Dr. Rob Stadler with us. As you can see here from the thumbnail, we are going to be addressing some arguments made by popular YouTuber Professor Dave when it comes to abiogenesis. So before we get into that, though, I'm going to give a brief introduction of our guest, Dr. Stadler. He's received a PhD in medical engineering from the Harvard MIT Division of Health Sciences and Technology. And of course, Rob, when I'm done here, correct me on anything I may have said incorrectly, but he has been a scientist in the medical device industry for over 20 years. And literally millions of his cardiac devices are implanted in millions of patients worldwide. He has also been elected Fellow of the American Institute of Medical and Biomedical Engineers, has obtained more than 140 U.S. patents, and has 20 peer-reviewed publications. Rob is also the author of 2 must-read and must-have books that I'm going to show on screen here in 1 second. The first one being The Scientific Approach to Evolution, What They Didn't Teach You in Biology, and The Stairway to Life, An Origin of Life Reality Check. Definitely 2 must-read books. I'm looking forward to having Rob on again in the future to discuss those books. So they're right here for everybody to see. The Scientific Approach to Evolution and The Stairway to Life. So enough for me. I want to introduce Rob to the show. Rob, thank you so much for giving us your time. We really appreciate it.
Hey, thanks for having me on there. I really appreciate it too. It's great to be here.
Awesome. Thank you. It's definitely an honor. We've also got Matt and John here. John has actually had an interview with Dr. Stadler on, I believe it was his book on evolution. And I've watched that interview. I definitely recommend it. I learned a lot. It was an awesome interview. So, John, any words from you there?
No, I think if you want to learn more about Dr. Stadler and his first book, which I think is absolutely fantastic and well worth your time to read, puts evolution into a very interesting perspective. I strongly recommend head over to my channel and watch our interview, and then immediately go and purchase his second book, which is Stairway to Life, which I've read, and I can tell you from firsthand experience, as we go through this video today, a lot of the things that we're going to discuss are expanded upon in great detail in Stairway to Life. So just keep in mind as we're going through this, that there's great amounts of substance behind all of the arguments and the positions that Dr. Stadler will be telling us about tonight.
Awesome. Thanks so much, John. Matt, as well, thanks for being here. Any words from you?
No, I'm just going to sit back, relax, and enjoy this one.
Yeah, I'm definitely excited for this one. This is going to be a lot of fun. So we've got a video that we are going to go through. We're going to have Rob here. Rob, just let us know when any specific spots you'd like us to pause, give your thoughts, give your arguments, and we'll make this smooth. We're going to try and keep it under an hour. And I think it's probably going to fly by. Like I said, I'm really excited for this. So why don't we, unless anybody else has anything to say, why don't we just get this going?
Awesome.
Okay.
So the purpose of making this video is actually to provide an origin of life reality check to Professor Dave. I don't have any misconceptions that he's going to change his mind at all in the results of this video, but he does have almost 1 million subscribers. And that's really where where my heart is to make sure that those million subscribers are not misled by what he says in this video. A lot of people appreciate his educational videos on chemistry and physics, and he does a great job there. But then when he strays from science in order to promote his agenda and trying to promote a natural explanation for the origin of life, I think that prompts the need for a video like this. I also wanted to mention that we're not here to defend Dr. Tor, Jim Tor, that Professor Dave kind of denigrates in his video. We're not here to defend Dr. Tor because he's quite capable of defending himself. We're just here to correct some of the erroneous statements that Professor Dave makes about origin of life. So I think what you're going to hear today is really two main concerns that we're going to focus on. To keep it simple, there's just two things. And the first topic is about bias, because bias is so important in science, and bias often clouds you from getting to the truth in science. So we have to address that. And the second topic is about sensationalism, or about embellishing the truth, stretching the truth, half truth. And we have to address that because there's a lot of that going on in Dave's video. So Dave, in his video, he makes some statements basically saying that religious people should not be contributing or interpreting data about or evidence about the origin of life. So we have some snippets from Dave's video on the screen now, and I'd like to play the first segment here.
Although the reasoning behind his stance on abiogenesis is clearly religiously motivated, despite his best efforts to convince people otherwise, he is entitled to his religious beliefs. It is clear that his religious views cloud his capacity to interpret research on this topic.
Okay, hold it there. So that went by kind of quickly, but what you see there is Professor Dave calling out Jim Tor for being biased because Jim Tor has said that he's a Christian. And to be open and disclosing our bias, those of us on this show now also come from a Christian religious background. The trick is, and the trouble is, that when Dave says it is clear that Jim Tor's, religious views cloud his capacity to interpret research on this topic, what Dave is basically saying there is that only atheists are qualified to address the science behind the origin of life. He's basically trying to say that only atheists are unbiased and are able to get to the ultimate truth because they're not clouded by their religious backgrounds. So that actually, I think, is rather a strong hypocrisy on Dave's part, because Dave is an atheist, and that means he has an absolute materialistic or a naturalistic bias. In other words, if you look at origin of life at the highest level, the big picture, there's really just two hypotheses. One is that life began by purely natural processes. And the other hypothesis, the alternative hypothesis, is that life could not have begun by purely natural processes, which means that something beyond natural processes had to help out. But because of Dave's bias, his atheistic, materialistic bias, he blocks the second hypothesis out up front immediately. So all that's left is the naturalistic explanation. And so the data has to support that. And by blocking out a possible explanation and leaving only one behind, that's an extreme kind of bias, which I think is even greater than what Jim Tor has. It's greater than what I have. I think the two possible hypotheses should be left on the table, and then you bring up the evidence and you stack it up and see which hypothesis is best supported by the data. It's certainly an extreme bias to block one of the hypotheses out right up front.
Exactly.
So, any comments from you guys on that?
Well, something I would add would be, or not really add, just expand upon, is your point about the bias. I mean, just today, I had an interaction with somebody about this topic, and their position was that the hypothesis of the intelligent agent is purely faith-based, and there is no remote evidence you could have for it. And I find it very interesting. I hear that on a very regular basis. I know we all do. And it's like they forget that on the evolutionary side and the abiogenesis side, it's also being based on deduction and forensic evidence and historic evidence versus actually observed evidence. And Dr. Stadler, you do a great job in your first book, really breaking this down from the evolutionary perspective and looking at, talk about the bias and different things. But I find it very, very interesting how applying the same standards of evidence that you would in any other aspect of science. And I know you talked about this in context of your research. That seems to fly out the window in the context of things that we're talking about tonight, and then just evolutionary, the theory of evolution in general. Is that a fair statement?
Yeah, it is important to keep in mind, if you follow the naturalistic, the story about origin of life, you're talking about a singular event that they say happened 4.5 billion years ago. And, there is no remaining evidence of that. There are little hints that they may bring up, but that evidence, is inherently very low confidence. But we do have evidence that we can find in existing life. And we'll talk about that a fair amount, hopefully in the next hour, about the evidence we have from existing life and which of those two hypotheses that the existing evidence supports. And that evidence is directly observable, and you can repeat it and you measure it in life today. And I would like to also bring up that in order to be an atheist, in order to be a rational atheist, you have to have a natural explanation for the origin of life. In other words, believing that there is a natural explanation for an origin of life is absolutely foundational and fundamental. And if that is somehow taken away, then you could still be an atheist, but I don't think you could be a rational atheist, because something beyond natural had to help out or had to be a part of the origin of life. So my point is that I think atheists have a much stronger bias coming into this, because the very foundation of what they believe is at stake.
And I have something to say to as well. It would be more of a question for anybody listening: is if our judgment was truly clouded by our belief, then how come the amount of Nobel Prizes are disproportionately in favor of Christians over atheists? If our judgment was clouded, we would not be dominating in that area. Just an idea.
Well, and I know we're going to go on to the next segment, but one other piece, and I'm not going to turn this whole stream into a promo for Dr. Stadler's Stairway to Life book, but something I was really struck by in it, as he dove into great detail, was the plethora of paradoxes that must be accounted for from the purely naturalistic worldview. I mean, it's paradox after paradox, and you and I have talked about this, Rob, of you didn't even cover all of them.
Mm-hmm.
Right. And the irony is we seem to be discovering more potential paradox that have to be overcome. The deeper we get into origin of life research, the more barriers to entry we seem to be finding rather than solving. And
right,
to me, that really exacerbates the point you're making of who has more bias. If the barrier to entry gets bigger and bigger, and you double down and double down and double down on your belief, who is taking the greater leap of faith? I guess is the point I'd be making.
And each of those paradoxes are sometimes called causal circularity, or sometimes called chicken and egg phenomenon. Those speak evidence toward hypothesis 2, right? Because those paradoxes are unnatural things, yet we know they exist. They point to hypothesis number 2.
Right. Actually, I was going to touch on that. I'll say one quick thing, and then, of course, we can move on. But the chicken and egg problems are huge, Rob. I was going to point that out. That's a great point. And the fact that the origin of life, as you point out, once again in the Stairway to Life, as John was pointing out the origin of life is extremely low confidence science versus high confidence science. And I believe it's in chapter 6, you speak, and you just spoke to this as well, but you go over a number of criteria, 6 criteria that have been applied to the study of abiogenesis. And you point out, as you just correctly pointed out here, that the origin of life is the antithesis of repeatable. It is an event that supposedly occurred on one occasion about 4 billion years ago. But I like how you also pointed out, Rob, that even if modern laboratory experiments could create life from non-life, we would have no confidence that the conditions of the laboratory experiments were consistent with those of the early Earth. So I just wanted to point that out because I really liked that in your book. So.
Yeah, if you could do it in a lab today, it certainly would be encouraging. It would be very interesting, but I wouldn't say that that's proof that that is relevant to what actually happened in the original case.
Exactly. I like that point.
So we're back on the topic of bias here, and let's play the video forward to the next segment here.
To interpret research on this topic. Those who think, I don't know, therefore God, would do themselves a service by stopping after I don't know and learning how to cope with uncertainty.
Okay, thank you. So here you have Dave basically counter arguing against what's called the God of the gaps argument. God of the gaps is basically saying, if there's something that we don't understand, we don't know how this is working, therefore God did it. And Dave saying that's not a good argument. And I totally agree with him on that. I don't think that that's an argument that makes sense. And Jim Torr doesn't use that argument. And I try not to also, of course. Again, science should be open to follow the evidence wherever it leads. And so you should stack up the evidence against the hypotheses and see which is best supported. So following the way that Dave calls out the bias of Jim Torr, he says Jim Torr is clouded and he can't, be trusted to interpret origin of life data, and how he rejects the God of the gaps argument on the slide you see before you here. It then comes as quite a surprise that in the same video, Dave makes the following statement. If you could play it forward again. Okay, right here. So this slide, you can read the text on it. He says, about the origin of life, Dave says, we may not know precisely what happened. There is no doubt that it happened totally naturally. So this is, in place of the God of the gaps argument, this is kind of the opposite. This is what I call materialism of the gaps, or naturalism of the gaps. He's basically saying on the slide, we don't know what happened, therefore naturalism did it. Right? So it's hypocrisy against what he just said in his own video. And I'm afraid his 900,000 plus viewers are going to be misled by this statement at the bottom here into thinking that it's a solved case, that we have all the evidence to prove that it's natural. And that's completely not the case. This is sensationalism. This is a statement of ideology. It's a statement of faith. It isn't science at all. Dave here is kind of showing that he's not speaking about science, but more being an evangelist, I think, for atheism. And so you see the hypocrisy here and you see the bias. And that's kind of the main point I wanted to make. Anything from you guys?
Well, I mean, the only thing I would say is, and I've heard this, I've actually experienced this personally in a debate on this topic. Life exists, therefore the probability is 1. And I think that's a very succinct way of saying that, hey, we believe that because life exists, it happened totally naturally. And here's our evidence. And I think the point you were making a minute ago of the circular evidence and circular reasoning, chicken and egg problem, I think that's embodied in that statement of like, okay, so your answer is the only one?
Well, that again comes from taking those two hypotheses and blocking one out up front. And so all that's left is natural, and therefore it's true. That's a kind of a circular argument, right? So in the second part of this video, I wanted to talk about sensationalizing evidence or embellishing the truth. And Dave makes a statement next on our slide, which I think is very interesting. Go ahead, play that.
Sensationalist reporting of science news, no matter which way it leans, is destroying public perception of science.
Okay, hold it there. So I completely agree with Dave on this point. I think sensationalist reporting of science is destroying public faith and trust and perception of science. So I agree with him. I think the problem, though, is that much of Dave's video is sensationalist reporting of science. So this too is hypocritical. So let's begin. We're going to go through a few cases. We don't have time to go through all of it, but let's get into let's play the next segment and you'll see what I mean.
Destroying public perception of science.
So here in this slide, you can see at the bottom, it says biomolecules can be self-replicating. And then play it forward a little further.
Ribozymes can even perform self-replication. That's right. Already at this simple level, we are introducing the possibility of self-replication.
Okay, hold it there. So Dave is making a claim there that molecules, especially ribozymes, which are RNA molecules, are able to self-replicate. He said it in two places. I find this very disturbing because the fact is that no one has ever seen a true self-replicating molecule. No one ever. And I know we haven't defined self-replicating yet, but I'm speaking from a sort of common sense definition. Let's go to the next slide there where we actually have, kind of a boring dictionary definition. But here it says that, according to dictionary.com, self-replicating is reproducing itself by its own power or inherent nature. And then Merriam-Webster says self-replicating is reproducing itself autonomously, which is basically saying the same thing. And Dave is not the first person to claim that there are self-replicating molecules. I pulled this book out off my shelf. It's called What is Life from Adi Pros. And basically he's talking about naturalistic origin of life, but he says in the book here, molecular self-replication reaction is a reality, a reaction that actually does take place, and most importantly is autocatalytic. So there are a lot of people that make this claim, and I'm bothered when I hear it. So I actually went on the comments page of Dave's video on YouTube, and I asked him to please produce evidence to back this claim. And what really got me kind of bothered here is that Dave responded with insults, but then I kept pressing him for evidence, and he ended up censoring me and deleted all of my comments from his page. So I'm not able to write comments anymore. And that's really a behavior that I never see from scientists. Usually when you ask a scientist for evidence, they're willing to try to produce something. So it quite surprised me that that was his approach.
Ladies and gentlemen, that was an extraordinarily nice way of letting Dave know that that was very offensive and that was not very scientific from a professional perspective. And a PhD was asking him a question. I don't mean to interrupt you there, Rob, but as you were sitting there saying that very politely, that's what was going through my mind of, wow, you actually took the time as a well-respected researcher yourself to ask him to live up to the standard of evidence that would be expected of you and all of your peers. And he not only dismissed you, he deleted the questions and challenged his position.
Well, I'd like to think that everybody listening would agree that censoring is not a good idea, a good process to get to the truth. Let's say that.
Well, I'd like to point out something really, really quick, Rob, if you don't mind, because I see it. I think of it as an admittance of defeat on Dave's side when he responds to your 100% valid question with what? With insults. And it's not really surprising from what we've seen from Professor Dave. Ad hominems seem to be his specialty. So, Rob, all you did was ask for some empirical evidence to back up what Dave was saying, and you got censored. And that's not it's obviously not an appropriate approach to scientific discussion. So.
And so because Dave didn't provide any evidence, I thought we could go through a few papers that I believe also are sensationalizing and I think have misled people into believing the claims that Dave himself made. So if you could forward just a little touch on the video, what we'll see this right here, this is the first paper that kind of got this started. And Sol Spiegelman wrote this paper. This is 1967. We're talking about 53 years ago. Incredible. But the title of the paper is quite sensational. It's a Darwinian experiment with a self-duplicating nucleic acid molecule. And that happens to be RNA. So from that title alone, it sounds like, hey, you've got the data you want. There are self-replicating molecules out there. And when you take the time to actually read these papers, it's surprising that they're able to use such terminology as self-replicating. You know, according to the dictionary definitions we showed, and I also think common sense, there's nothing here that is self-replicating whatsoever. In this particular study, what was doing the work, what actually replicated the RNA, was called a Q-beta replicase enzyme. So this is a protein molecular machine. It's actually made up of 4 protein subunits. A lot of people think enzymes are just a single molecule, but most enzymes are actually a complex of multiple protein molecules forming a molecular machine together. So this Q-beta replicase actually has 1,200 amino acids in the sequence of those 4 proteins. So it's quite a complicated protein enzyme that he dropped into the solution, and that is what is replicating the RNA. So calling it self-replication seems, you know, a bit disingenuous there. And not only so, there are a lot of other problems with what he found there, and that is that as the RNA was replicated generation after generation by this Q-beta replicase, the RNA actually shrunk over time. It was losing information. It was chopping off because shorter RNAs are naturally going to replicate faster and outcompete the longer RNA. So it's a kind of a devolution, a loss of information, certainly not what you'd expect from a process that could lead to life. So then that's 53 years ago. So what's been accomplished in 53 years since then? Let's go forward to another paper and to the next slide. So this paper, this is from Gerald Joyce, who's quite a famous origin of life researcher. And this paper and a whole series of papers like this about this particular molecule have raised a lot of eyebrows, got a lot of attention. But you see the title of the paper here. It says self-replicating RNA enzymes. Again, it sounds like done deal, right? You got your evidence. And you can see the highlighted first sentence there. An RNA enzyme has been developed that catalyzes the joining of oligonucleotide substrates to form additional copies of itself, undergoing self-replication with exponential growth. So there you have it. It's done. But when you actually read the paper, once again, it takes a very, very charitable definition of self-replication to think that that's the appropriate title for a paper like this. It takes a very charitable definition. Let's move forward 2 slides. So one more. Yeah, so stop here. So this is complicated, and I'll talk you through it a little bit. I don't mean to, there's a lot of detail here, but the yellow part, what you see in yellow there is the ribozyme that he's talking about. It's an RNA molecule. And RNA is made up of 4 different ribonucleotides that have codes like A, G, U, and you can see those there. So in the yellow, you see a string of 66 ribonucleotides and with the codes. That is a molecule there that was designed by human intellect and passed through, you know, a sort of artificial selection process, a laboratory evolution process, if you will, with human intellect pulling out the winning molecules. And so they arrived at this molecule because it has the property we're going to talk about. But that molecule in yellow is by no means something that you could find in a prebiotic world, in a place before life existed, because, it's extremely complicated. But even looking at just the chirality of this molecule, this is a homochiral molecule, and every ribonucleotide actually has 4 stereocenters, meaning 4 different locations of chirality, meaning every ribonucleotide has 16 possible chiralities, but only one is allowed in life. Only one is used in life. So to have 66 ribonucleotides in a string, all having the exact same chirality, all linked up perfectly like this, it's impossible in a prebiotic world. Basically, if you took all of the mass of the universe, if it were all made up of 66 ribonucleotide RNA strings like this, you took all the mass of the universe and randomly had it be 66 ribonucleotide strings, you still wouldn't be able to find one that was this particular molecule, because it's that rare, that hard to come by. Now, I'm getting long-winded here, but what happens now is that in the solution, they put the yellow molecule in the solution, and then in the same solution, they also put the blue molecule and the green molecule, substrate A, substrate B. You can see them there. Now, if your eyes are good, you can tell that substrate A plus B combined is actually the same as the yellow molecule. It's just flipped over like a mirror symmetry there. So they basically put 2 pieces of ribosome of the yellow molecule. They put 2 halves in the solution. And all that this yellow molecule had to do was line up the 2 halves, like you see here, and create one single bond between the blue and the green. And you could see between the blue and the green, if your screen has good resolution, you can see PPP. And what that is a triphosphate, a very highly activated part of the molecule. It's very eager to bond because it's got this high energy state, which is also rather unnatural. So when the yellow molecule brings the green and the blue in close proximity, that PPP is very eager to bond, and it creates a bond between the green and the blue. And then there it is, you have a copy of the yellow. So that is considered by Gerald Joyce to be self-replication. So if you go to the next slide, because this is so complicated, I wanted to give you a simple analogy just to explain this. A really close analogy to this would be if I took a fully functional car, and I chopped it in half. So you have a front half and a back half. And then where it's been chopped, I install strong magnets, and I install some mechanical clasp kind of mechanisms between the 2 halves, so that if you take the back half and you push it against the front half, they would be attracted to each other, and the mechanical clasps would come in, and it would actually form a car that was actually functional. Okay. And then you put behind that, you put a fully functional car, and you drive that fully functional car a little bit forward so that it hits the back bumper of the back half and starts to push it forward. And then it bumps into the front half of the car, and you hear this click as the 2 parts come together, and there's a mechanical clasp. And now you have a fully functioning car in the front. And if I did that, and then I jumped up and down and I said, look at this, I have just produced the world's first self-replicating automobile. That's basically something that I should be laughed at, right? People should laugh at me for making such a silly claim, but that's exactly what has been done with this molecule. And so when people claim it's a self-replicating molecule, you always have to read the details and appreciate what's going on there, because there's trickery to this, and I think it's there because they absolutely have to have a self-replicating molecule, or there is no basis for a naturalistic origin of life.
Well, it's interesting you say that. I know SFT and myself hear this on a very regular basis. The when and SFT, standing for truth, actually loves using cars and various other vehicles as examples for like different hierarchical structures and things of that nature. But the thing we hear on a regular basis is, yeah, well, you're just talking about human designed technologies. We're talking about self-replicating biological systems. And the irony I know for myself of, what you just explained and why it's such a joke at this point, almost, of like why in any other context you would be laughed out of the room if you made the same argument. And yet the atheist worldview, and even in academia, they're doubling down on this position that somehow the self-replication explains how in the heck the magnets and the class mechanism came to exist, let alone the other parts that are being connected. Like, hang on a second. What are we talking about here? Like, which part are you claiming can be explained through undirected process? Because to me, all of those processes required intelligent agency in order to be executed. Now, the physical bond at the PPE site, cool. Natural forces caused the bond. Congratulations. But if, to your point you made, if it hadn't been high energy state, then the bond may or may not have happened, or it might not have happened at all, right?
Yeah, it's more likely to go the other way, actually, because it's in an aqueous solution in water. That bond actually kicks out a water molecule. And so you're in an aqueous solution that wants to drive it the other way to break apart the RNA. So you have to overcome that with some kind of energy source. And in life, enzymes do that with the help of ATP. That's how it happens in life.
I know we talk about this later, but the quantum, and there's that whole aspect that you talk about in the book, which is starting to finally get focused in academia, but the whole aspect of, hey guys, we've got quantum tunneling going on to enable a lot of these reactions we take for granted to occur. That's another, we'll go into detail on that right now, but I mean, that's a whole nother aspect of, in my opinion, what's being discovered that kind of puts this whole conundrum into an entirely different, it's a whole other stratosphere when you start to think about those kind of things, in my opinion.
Mm-hmm.
Well, I'll say one thing really quick. Rob, I think you nailed it there. That was really good, really great. And I appreciate the analogy. I think the audience will as well. It made it a lot more understandable. What you said was perfect as well, that they have to have a self-replicating molecule, or else there is no scientific basis for an origin of life scenario. And when you think about all the problems, the homochirality problem, the polymerization problem, the information problem, it makes me wonder, at what point do they tap out and admit that there may not be any plausible explanation for any type of origin of life scenario? So I really liked what you said there, Rob.
And let me add just a little more icing on the cake there, that it doesn't even make sense to think that a ribozyme, so a ribozyme is an RNA molecule that performs some catalyst function. It doesn't even make sense to think a ribozyme could reproduce itself. And that's because in order to be a functional catalyst as a ribozyme, it has to have some three-dimensional shape to it. It's the shape that makes it a catalyst. But in order to reproduce itself, it needs to be a one-dimensional string. So you can add components one by one onto the string and glue them together to make a copy of itself. So that doesn't even make sense. And then you also have this problem called annealing, where the RNA and its complement RNA are held together by Watson-Crick bonds. And once you get to more than about 30 nucleotides, you can't get those bonded complementary RNAs to let go of each other. Even if you boil it at high temperature, they won't let go of each other. And nobody's found a solution for that.
And that's where things like helicase and replicase come into the equation in terms of current biological systems, right? They actually, those protein enzymes actually cause the separation. Is that for?
Yes, that Q beta replicase that Saul Spiegelman had, that also does that. It's able to cause that separation because it's a complex enzyme. So bringing it back to our point here, we have Dave claiming that self-replicating ribozymes are real, that they're a reality. And my point is that that is sensationalizing the truth. And going back to what Dave said in his video, he said sensationalist reporting of science news, no matter which way it leads, is destroying the public perception of science. So there you go. So let's go on to some more evidence that Dave presents. We need to go forward, to 2 minutes. There it is. Yeah.
Production, if life. With regards to protein production, if catalysis via ribozymes is deemed insufficient, there are plenty of other possibilities. Hydrothermal vents or volcanic hot springs could have provided the energy required for the polymerization reactions. But my personal favorite explanation involves heterogeneous catalysis over mineral-rich surfaces in tidal pools. The small volume of the pools solves the problem of molecules having to find each other in the ocean, acting essentially like a chemist's flask in a laboratory.
Okay, so here we're trying to build a protein from individual amino acids. And Dave is kind of hand-waving his way through it to kind of make it sound easy. But you have a problem in that these amino acids, when they join together into a polypeptide, there are multiple ways they can join each other. And only the alpha peptide bond is what we find in life. But when that alpha peptide bond forms, it kicks out a water molecule. So once again, if this reaction happens in water, it's going to be driven backward. It's driven the other way by so much water being around. And you need a carefully directed source of energy in order to make the reaction go forward and produce a polypeptide. I thought it'd be fun to give a little reality check on this one too. I had a debate with another origin of life person, and I was kind of arguing that in the years since Miller-Urey experiment, which is now 68 years since Miller-Urey, that not much progress has occurred in origin of life. And he fought back and said there's been great progress. And he sent me the paper that you see on the screen here. And this paper is about forming proteins or forming polypeptides. And this is kind of modern example of how far they've come. And so you can see highlighted here is actually a statement that kind of contradicts what Professor Dave said, but it says, the mechanism by which alpha amino acids, which are the ones we found in life, were condensed into polypeptides before the emergence of enzymes remains unsolved. So they're saying, they admit we don't know how this has happened. But in this paper, they go on to show what they have accomplished. And what they have accomplished is that they can now take 14 amino acids and get 14 amino acids to link to each other. But they have to be homochiral, and the links that they form are not always the correct linkage. It's not an alpha peptide bond. And they've gotten 14 of them. And that's after 62 years since Miller-Urey. This paper came out. After 62 years, Miller-Urey had single amino acids floating around. Now, after 62 years, we can now take 14 of them, if they're homochiral, and we can connect them, although not in the way that proteins are connected. And that's how much progress we have in 62 years. And you can compare that against any other technology in the last 62 years. Computing, aviation, medicine, what progress has come in the last 62 years compared to taking 14 monomers and bringing them together into a polymer, sort of. So what are the next 62 years going to bring us? 62 years more of funded origin of life research. Now we're going to have 30 amino acids that are kind of hooked inappropriately.
Now, Dr. Stadler, can you put that into context of, they've gotten 14 incorrectly paired amino acids. How many correct pairings is a ribosome? doing when it's creating a protein. Like it's doing it pretty quickly, and usually it's pretty correct and has a relatively low error rate, doesn't it?
A very low error rate, and there's error correction schemes in there. So, you can do 1,000, like the Q beta replicase is 4 proteins that are 1,200 amino acids. So you need 1,200 correct alpha peptide bonds between 1,200 amino acids to make one of those.
It's something that I've wanted to have clarified for a while now, actually, is from a protein formation perspective, if it's not the alpha bond that occurs, does the protein formation basically stop and then get restarted, or can it get past a couple of errors and keep going, or like how does that usually work in the actual formation? Is it kind of a, hey, I got to start over kind of perspective, or
there's various error correction mechanisms built into ribosomes that is probably beyond the scope of this discussion, but it's true though that if you connect them wrongly, you end up with what's called proteinoids, which is basically garbage. It's kind of useless, and some people have claimed I made proteinoids, and that's a step toward life, but it's really just a step toward garbage. I don't think they're getting anywhere with that.
So even if where my mind's going with this is, so even correct me if I'm wrong, but what you're saying is, even if they through natural undirected process you're able to get more than 14 bonds to occur, you only need one, unless you could have 13 of them that were correct, and then at number 14, if you have the wrong one, you're screwed.
Well, I can't say that straight up. I think there will be, there could be some enzymatic function, some catalytic capability of a molecule like that, but it certainly isn't what you find in life. In life, proteins often form alpha helices or beta pleated sheets are common structures that proteins form. And it's known that if you have the wrong chirality in an amino acid or the wrong bond, you won't get that kind of formation. So the three-dimensional structure will certainly be interfered with. I can't say that the enzyme would be completely useless, but it's certainly not what you find in life. So if we can, let's go forward then to the next segment, which is on chirality.
But more importantly, the mineral surfaces could have acted as naturally occurring organometallic catalysts. These surfaces may have not only performed efficient syntheses, but given the possibility of the surfaces exhibiting chirality, it could have been the case that they preferentially reacted with one particular enantiomeric version of molecules, like perhaps only L amino acids. This would readily explain their prevalence in proteins, with enzymes arising later to perpetuate this stereochemical bias after their dominance had already been set into place. Even without this stereospecificity in polymerization, there are other ways to get enantiomeric excess with the amino acids themselves. It has been demonstrated that recrystallization of a mixture of amino acid racemates with a slight excess of asparagine caused amino acids with the same configuration to preferentially co-crystallize. You get a solid containing only one enantiomer and a supernatant primarily of the other. All that's left is some natural filtration mechanism to separate the two and the emergence of autocatalysis in one.
Okay, well there. So that's a very optimistic approach to claiming that the chirality problem has been solved for proteins. I don't think many origin of life researchers would be as optimistic as Dave is there. But what I've never see talked about, actually, for me, is the most challenging homochirality problem, and that is with RNA. And I mentioned it briefly, but each ribonucleotide in RNA has 4 chiral locations, or 4 stereocenters, and that means there are 16 possible chiral forms for every ribonucleotide. And I've never seen anyone try to propose how a natural process could select out one out of the 16 possible forms and choose that. Because if you have a solution with all 16 forms in it, the chosen ones that you like will never find each other, will never bond to each other and make a perfectly homochiral molecule. The probability is just completely against you. And in fact, in life, molecules that are chiral molecules, over time, they'll just randomly switch chirality. And so life, in order to maintain life, you have to continually fix these problems. You have to have repair mechanisms. There are enzymes called epimerases and racomases in life. These are enzymes that go around and they fix broken chirality problems. So even if you could get a homochiral molecule, it's not going to last very long. It'll degrade just in water to the point where it's not homochiral.
So, quick question for you. So, if I understand what you just said correctly, not only do we have damage repair mechanisms for deletion or damage to a nucleotide itself, we also have other ones that come and fix it could be the correct one, but the chirality is getting messed up. So there's another set of repair mechanisms comes in to fix the chirality, even though it might be the correct nucleotide. Is that what you're saying?
That's right.
Wow.
And there are actually people who have proposed dating methods where if you find a protein in some very old, dinosaur bone or something, you could look to see how not chiral that protein is, and that might tell you how old it is, because there's a natural racemization over time once something is dead.
Well, for myself, I definitely have to say, just my 2 cents, looking at these explanations, say from Dave, and not just Dave, anybody else trying to defend abiogenesis. For example, with these problems like the chirality problem, for example, I personally see a lot of storyboards and just so stories, hence why specifically you'll hear Dave here say a lot of, maybe this, maybe that, possibly this, possibly that. So I think Dave's unrealistic optimization, as you pointed out, Rob, that he seems rather optimistic, is just to deceive his audience into thinking these problems can be solved. Since at the end of the day, a lot of what Dave is saying anyways in this video is probably going over most of his viewers' heads. But that's just my opinion on it.
Yes, that's quite possible. I agree. So there's just a few more here I want to go through, and we certainly aren't covering everything that's done in this video. We're hitting the highlights, but let's move on to the next sequence.
Every bacterial cell alive today is tremendously more complex than the first protocell that must have formed.
Okay, hold it there. So this term protocell is a hot topic also. Protocell is essential to help explain a naturalistic origin of life, because we know that all known life that we can see today, what we can directly observe today in known life, is far too complicated to have come together through a natural process to create that as a cell. It's far too complicated. But here you have Dave kind of stating it as a fact that protocells preceded simple life like a bacteria, simple cells that we see today. And the truth is that there just isn't any evidence for the existence of protocells. This is sort of a materialism of the gaps entity, right? It's created it's an imaginary object to fill in the gap, because it is essential, if you're going to have a natural process that creates life, that you've got to have these protocells. So protocells, when I hear the term protocell, I think of it like a promise in the year of election. It's a promissory note. It's an imaginary thing that's put forth as a promise that is really not going to be fulfilled. There's no evidence for protocells, but there's actually strong evidence against protocells. All right, and I'd like to explain that. So we know through directly observed genetic evidence we have, such as the long-term evolution experiment of Richard Lenski and many others like it, we know that living organisms will over time simplify themselves to be the simplest that they can be and still survive. So for example, in Richard Lenski's experiment, his E. coli bacteria were fed glucose every day. You know, what's for breakfast? Glucose. What's for dinner? Glucose. Every day. And so they have machinery built into them that's able to process ribose. But it turns out they never needed that because glucose was always on the menu. And so they found that in 12 separate colonies that were tested in Lenski's experiment, after only 2,000 generations, all of the 12 colonies had ejected the genome, the operon that's used to process ribose. And simplifying their genome in that way, they became about 2% more fit, 2% faster at reproducing. So what I'm saying is that these single cell organisms are trying to simplify themselves, and they've been doing that as long as life has been around. And so if you want a protocell, just look at the simple life that we have found living on our planet right now. That's as simple as it's going to get. It can't get simpler. It's tried. It's already tried. So we have Mycoplasma genitalium, which is considered the simplest single cell organism that can autonomously reproduce. Okay, and even Mycoplasma genitalium needs to have a kind of a coddled environment, but it is able to reproduce itself. So that single cell organism contains 468 genes and about 580,000 base pairs of DNA. So 580,000 letters of code in the DNA. And that is a very simple living organism. And people have tried, Craig Venter has tried to take away genes to make a simpler organism, to try to pare it down. Let's try to build a protocell, sort of reverse engineering from what we have on the planet today. And they haven't gotten very far. They haven't gotten much further below what Mycoplasma genitalium has. Because as you start to strip away things, what you do is you have to provide a more and more coddled environment. It has to be just the right temperature, just the right nutrients, everything handed to you on a silver platter in order to keep the cell alive. So these people are trying to make simpler life, but I think they needn't bother to try because nature has already done this. Nature's already come up with the simplest possible life. And you're not going to get any simpler than about 400 genes and about 500,000 base pairs of DNA. So there is no such thing as a protocell, and yet it's absolutely essential for a naturalistic origin of life scenario.
I don't know how much will we get into this tonight, and I think that what I'm about to say is worth multiple multiple streams on this one topic. But you know, in your book Stairway to Life, you get into the membrane aspect and the construction process. Is that actually self-replicating, and the how different proteins are built into? I mean, the whole aspect of how the proteins partially built, protected from the water, and the other parts in the water. I mean, in the aqueous solution and stuff. I mean, to me, folks, you probably don't understand what I'm saying. Most of you, go read the book, because when you get into this whole section on the membrane formation, which obviously is kind of a big deal for the protocells, and it's one of the things that the abiogenesis folks claim, oh, wait, we can just create this vesicle and it's no big deal. When you really get into some of the nitty gritty of what's going on in terms of how many proteins are actually making up that wall, making it work, and the processes by which it's actually formed. Number one, I think it blows your mind. And number two, it puts, in my opinion anyway, it kind of puts a lot of things that we're talking about into an entirely different context of what is plausible to reach these positions.
Well, let's carry that out a little further, John, by playing the next segment, because it's about the membrane.
The first protocell didn't have to have a single surface protein. The instructions for assembly...
Go back there. So just that simple statement, Dave made a claim that the first protocell didn't have to have a single surface protein. So this gets to what John was saying, that origin of life researchers will claim that a simple A simple bilayer lipid membrane is all you need for a membrane. And no one has ever shown that you can have a living cell that can live with such a simple membrane as that. That kind of a membrane would only serve as a tomb for the decaying components that are inside there. Cells have to have active membranes. They have to have active and semi-permeable membranes. So back to the Mycoplasma genitalium we were just talking about. That very simple form of life has 140 different proteins that it produces that are operating as part of the membrane. These are active molecular machines that are sending food into the cell, and they're sending waste out of the cell, and they're keeping homeostasis, keeping the environment inside the cell distinct from what's outside of the cell. That's an active process. It's not just a simple bilayer lipid membrane. And so once again, we have a kind of a materialism of the gaps fill in here by saying that a protocell can have a very simple membrane. There's no evidence that any life could survive with such a membrane. There's plenty of evidence that membranes need to be very complex and need to have lots of proteins as a part of them.
I think before you continue, I'll make just one observation also, just to rewind a bit to what you were saying earlier. When it comes to, I find out what you're saying is fascinating when it comes to the Mycoplasma genitalium, the simplest form of life on Earth. And I'm just going to quote you, Dr. Stadler, from your book, because you say it perfectly. This simple life requires the choreographic functioning of hundreds of genes to survive and reproduce. And what you said there, I completely agree with, that this protocell idea, it's an imaginary thing that is put forth as a promise that at the end of the day will never be fulfilled. Although those like Dave so invested, unfortunately, in naturalism, and therefore abiogenesis being plausible, hope so badly that it will be fulfilled one day. So
And correct me if I'm wrong on this, but you all do the point that Stanley was just making about all of the different genes have to be working in synchronicity. You have multiple paradoxes that are being in play in this. Unless I'm wrong on that, but I mean, we have the information problem, we've got the replication problem, we've got the where the heck did the proteins come from problem. We've got all sorts of different chemical reaction problems, and then we have a lack of accounting for the energy problem in terms of focus energy. Is that right? In terms of just a few of the things that we're running into at this point, of different paradoxes that aren't being accounted for, but just accepted to have occurred in order for this to exist, right?
There are layers and layers of paradoxes like that. You know, like a cell can produce a protein that has a job of functioning inside the cell membrane, right? They do that all the time. How can a protein get inserted into a cell membrane? The answer is it takes a protein that's already in the cell membrane and other proteins to help insert a protein into the cell membrane, right? So you can't get a protein into a cell membrane unless there is a protein in the cell membrane. This is another circular causality problem that we encounter. There are many. So let's go. We're almost done here. We have one more quote about the bacterium.
Instructions for assembly are encoded in the DNA.
Okay, hold it there. So what Dave was saying, that was very brief, but what he's saying is that a bacterium can assemble itself from the DNA instructions. And this is kind of a half truth. I mean, we all know that there's a lot of information in DNA. But the simple fact is that DNA on its own can do nothing, right? DNA on its own cannot produce a protein. DNA can't produce a membrane. DNA needs all of the other tools around it, all the other supporting molecules, and they're interdependent upon each other. DNA on its own will simply degrade. That's all it can do. If you take a cell and you take away its membrane, a cell cannot produce its own membrane de novo. Just like cells only come from cells, membranes can only come from membranes. So a cell's membrane is inherited from its parents. and the cell can add to it, it can extend it, but no cell has ever produced its own membrane from scratch because it can't. Even if the DNA instructions tell it how to make proteins and phospholipids and so forth, it inherits it.
So are you saying that all the way from sperm, egg, zygote, and beyond, the membranes that are duplicated, obviously, is during replication and such, all of those are none of those are de novo. They're
Exactly.
all an extension of the original. Is that
right? It's kind of like a template that you can grow upon and make more cells from it, but no cell is capable of creating its own membrane de novo.
Now, in context of removing abiogenesis, origin of life research from the equation, is the point you made about membranes coming from other membranes and so on, is that a established, accepted position in biology outside of origin of life? I mean,
yeah, that quote I gave, that membranes only come from membranes, it came from another book I read. At the moment, I'm blocking on the author, but yes, it's established.
So if we went and talked to anybody else, any other kind of researcher, they would be like, "Oh yeah, exactly, we agree 100%." But then you go and talk to the origin of life researcher. Oh well, it must have just occurred. There must be a process, even though all of the observed evidence says the direct opposite. Is that a fair thing to say? I'm talking about in terms of what we see. It goes along with your point.
Mm-hmm.
Origin of life would have to be the direct antithesis of that.
Yeah, and this kind of goes back to what I started with these two hypotheses. Either life began through purely natural processes or it didn't. And all of this chicken and egg, right? All these paradoxes that we've been talking about are evidence of the second hypothesis. And I really think it's only by excluding that hypothesis up front, which is what materialistic thinking or naturalistic thinking does. That's the only way you can have any support for the remaining hypothesis, because that's the only one left if you exclude the other one. So we've been going through a lot of details. There's still many more. We're not going to go through the others, but the point I'm trying to bring back, I said there were 2 points to the video. The second point was about sensationalism. It's about kind of amplifying or stretching the truth to make your point. And remember, Dave said, there was a little picture of a newspaper on the video, and he said, sensationalist reporting of science news, no matter which way it leads, is destroying the public perception of science. And I really agree with you, Dave. I agree that's true. And I'm afraid that I think your video is doing that. And it's causing people to not trust science because you're making very strong statements that are not supported. And when people hear that, they're smart enough to know better. So it kind of doesn't make sense what Dave is doing here. But at the end of the video, he has a quote that I think helps to explain this. He says that spontaneous arrival of life through purely natural processes, it runs counter to common sense. Our common sense tells us that that molecules don't just come together and produce a living, self-replicating organism. That's common sense. And Dave recognizes that your common sense says it can't happen. So to alleviate that discomfort that your common sense provide, Dave produces at the end of his video, it really is my favorite quote from 2020. So here it is.
Common sense has no place in science or in an assessment of the feasibility of biological processes.
Okay. Hold it there. So Dave said, common sense has no place in science or in an assessment of the feasibility of biological processes. So I guess we should at least give Dave credit for practicing what he preaches. He's asking you to put aside your common sense, suppress your thinking, your intuition, and just close your eyes and believe. That's what he wants.
Love it.
Yeah, he wants us to imagine. I'd like to point out too something that I guess just to branch off of something. There's been so many good points and so many good things said. I've got a ton of notes here. This has been awesome. Time has flown by, but I'd like to point out, like you guys were talking about earlier, that I'm seeing numerous interconnected and just irreducible problems that have no solutions. And these problems need some type of solution in order to make an origin of life scenario even remotely plausible. So like Rob was saying, hypothesis 2, given what we know about all of these numbers of paradoxes, chicken and egg problem after chicken and egg problem, the remaining hypothesis, excluding hypothesis 1, would be the most plausible hypothesis.
Exactly. That's what we're left with.
So Rob, I have to ask you this question directly. So you are a respected researcher. You've created many devices for healthcare. I'm going to assume that the things that you have presented for funding, for grants, for whatever the process is for pursuing one of the, whatever the concept is, I'm assuming that whatever you presented had to have common sense and had to have a practical and plausible reason of expectation to be accomplished. Is that a fair position?
Yeah, that's fair.
Would if you, I know you work in the private sector now, but if you were back in academia, for example, and went for a grant and said, no, I don't think this has any common sense and I don't really have expectation that it's plausible, do you think it's a remotely plausible scenario that you would actually get funding for that?
Well, it's plausible you'll be kicked out of the room, I suppose. To be a little charitable with what Dave said here in this quote, I think what he's saying is that sometimes science does produce a result that is counterintuitive, right? Like we all make mistakes in predicting that this will be the answer, and it turns out you were wrong, right? So humility is a very important part of science. And sometimes things run against what you expect from your common sense. But I think Dave is saying it in a stronger way here. And I certainly don't agree with the statement that he wrote.
So let me get this straight. Professor Dave says that cells can reproduce or produce membranes. They can't. There's no evidence for replication that he states as a fact and that it does happen. That's not true. And then protocells existed, he's saying, that there's no evidence for. All while he's saying that the news is sensationalizing these things while he's doing the very thing he's complaining about. Seems pretty ironic.
You nailed it, Matt. Yep.
And I think, honestly, that's why he censored me from putting comments on his video.
No doubt. And look what that got him. A whole video.
Well, I tell you, one of the things I found most interesting about the sensationalization from the media and such is, and we didn't get into all of them, obviously, in this commentary, but there were multiple components of that video, which I know beyond doubt were pulled directly. The concepts were pulled directly from sensationalized articles, not peer-reviewed papers, but I mean from articles. And part of the reason I knew that is because I've read some of them myself, and I recognize some of the points that he was making as them coming directly from different sensationalized publications from, not necessarily from a scientific journal. And one of the reasons I bring this up is something I know that SFT and myself run into many times, and I'm sure you have as well. And people come after you for your books of, when we point out the papers that, just like you did tonight, that discredit their argument or their position. It seems the next time we run into them, they didn't actually go and read it for themselves to go and see that the position, the argument that you're making or we're making is correct, or that their position is not actually supported by the evidence, other than maybe 2 sentences in the abstract. But when you actually go further into the paper, it turns out that they didn't actually establish the high level position that was taken in the title or in the first sentence of the abstract. And I found that quite a bit. Is that something you've recognized as well?
For sure, but it's very tedious to read the paper. It's very hard to get at what actually happened in the paper. It takes a lot of time and energy and effort, and it's easier to just read the title and kind of wash your hands of it, say, we got the answer here, right?
Yeah, I think it's kind of interesting. Over the last several months, I've read quite a few different articles from people from journals talking about the significant problem with the lack of repeatability. And there was even one, I forget the gentleman's name, but he's refusing to now be an editor and a reviewer of articles or of papers that don't clearly delineate the process that was used to reach the conclusion. And one of the things, from you and I's first conversation, I found very interesting about that, the editorial I'm referring to was, you were talking about in your research how if you can't make it repeatable and you can't clearly show how you reached your conclusion, then in the medical device example, research field, you're not going anywhere. And yet somehow in things like Origin of Life, rather abstract theories are put forth with no substance other than it sounds good and has a great storyboard. And
we have to recognize the publication bias, that again, back to this hypothesis 1 and hypothesis 2, where number 2 is that life could not have begun from natural processes. You just literally cannot publish a peer-reviewed paper that supports hypothesis 2. It will never make it through the peer review process. If it did, the editors would they would lose their job, and you publishing it would probably be blackballed from science. And there are many examples of that. I'm not just Mr. Conspiracy Theory here. There's plenty of examples where that's really the case.
Well, let's hear it one more time from Professor Dave here, who violates Occam's razor.
Common sense has no place in science.
Hear that, everybody? Ignore common sense.
That's a great way to wrap it up.
Yeah, let me jump in here. So I was just going to say that. So this was awesome. We are going on over an hour and 15 minutes now. Time has flown by. I thoroughly enjoyed this. Rob is an incredible blessing, and I thank him for giving us his time tonight for this important video. So please, everybody watching, make sure to check out his must-read books, The Stairway to Heaven and The Scientific Approach to Evolution.
Stairway to Life, that is.
Stairway to Life. I messed that one up. We were so close to having a perfect stream. So
I make no claims to be Led Zeppelin here.
Yeah, there you go. That's already been copyrighted. So my apologies. The Stairway to Life. Any final word? Oh, go ahead, Matt.
No, I'm just saying, just re-say what you just did and do it right. I'll edit it.
That's right. That's right. We're not even live. Okay. Well, just that one part. Okay. Okay, here. Please, everybody watching, make sure to check out his books, The Stairway to Life and The Scientific Approach to Evolution. Any final words from anybody on the panel tonight before we shut it down? Rob, John, Matt?
Oh, just to thank you again for the opportunity to be here. And I do indeed hope that the people who are subscribed to Professor Dave will take this seriously and not be misled by his hidden agenda.
Amen. Amen. Any final words, Matt, John?
Yeah, last thing I'll say is, everybody, I strongly recommend getting both of Dr. Stadler's books. I believe, SFT, you're going to put the links in the description, right?
You got it.
And I strongly recommend you go and check them out. And for any of you who are thinking, hey, we're just being full of hot air and talking and we're just, completely biased, as Professor Dave likes to say, go read the books for yourselves. Go look further research beyond the standard talking points, and whether or not you come to the same conclusions we have and the positions that we're taking tonight, you'll be very hard pressed if you go into it with an open mind to at least not begin to question the proven beyond all doubt positions that are taken by folks like Professor Dave, and at least begin to potentially question and think for yourself that maybe we don't have all of the answers, and you should be a little more open-minded rather than fall for the naturalism of the gaps answer to all of the the major conundrum of how do we exist, which I think is kind of an important one to consider.
For all those who appreciate the work that we're doing here on Standing for Truth, please hit that subscribe button because we are just getting started.
Okay, looks like we are live for another great show, guys. This is another great show this week. It's a busy one, but we've got another awesome guest with us today. I want to thank Dr. Rob Stadler for once again giving us his time for this incredibly important discussion on abiogenesis, but specifically on his newest must-read and must-have book, The Stairway to Life, An Origin of Life Reality Check. Rob, thanks so much for joining us today.
Hey, thank you for having me. It's great to be here, and I appreciate you. You haven't gotten tired of me yet, I guess. This is the third time, huh?
No, you're a fan favorite. They'll probably kick me off the channel before they kick you off the channel, Rob. I want to point out to the audience, please, if you have not yet, please check out Rob's previous videos with us. We've had him on to debunk Professor Dave. That's a must watch. And we've recently had him on at the end of 2020, actually, to discuss his book on evolution. And that was an extremely informative and helpful interview. So actually, I want to thank George. George is going to be joining us for this interview today as well. George, thank you for being here.
No problem. I was encouraged by my first joke to Rob. He laughed at it. So I'd like to maybe tell him another one, either now or later on. It's up to you.
You can start it off now, George, if you'd like.
All right, Rob. What was the biologist wearing on his first date with a hot chick? Designer jeans.
Oh, designer jeans.
All right, I'll stop there then.
I can't top that, George. So no joke.
Quit your day job, George.
Good on you, Rob.
For you, George. Anyways, I want to point out before we get this party started here to the audience. Good to see everybody. Good to see so many brothers and sisters. I'm really excited for this. Please tag me with your questions. We're going to keep it on topic and keep it relevant to abiogenesis and specifically Rob's book, The Stairway to Life. So tag me with any questions. If it's any questions that pertain to something Rob's discussing in the presentation, then I can stop him and we can kind of clarify from there. Or also I'll save them for the end of the presentation where we will have a short audience Q&A. So anyways, enough from us. I'm going to hand it over to you, Rob, for some brief words in terms of introduction, and then we can get into your presentation.
Well, today we're addressing, I think, one of the most important questions you can ask, which is, how did life get started? And if you've been told that life started from purely natural processes, just chemistry and physics doing what it does, then this show is for you. So I hope you stick with us and keep an open mind.
Amen. I agree, Rob. It's one of the most important topics. And I got to say, ever since we scheduled this, I've been extremely excited. So once again, thanks for giving us your time. And would you like to get right into the presentation?
Yeah. So I have some slides that we'll go through, and like you said, we can take questions at the end or jump in if you feel the need to. But I can start to share my screen here.
Sure, I'll get that going for you as well. Here we go.
Hopefully that's pretty clear. Is that good?
Looks good. Looks great.
So here's the book we're talking about today. And I want to point out my co-author is Change Tan, and she's a professor from the University of Missouri. And she and I got very passionate about this topic and to write this book because it just seemed like that there's an increasing barrage, a kind of a media blitz going on about natural explanations for life. it seems like every day you see something on the internet that's a new theory, new data, all this that's always very encouraging about natural causes behind life. And so we felt the need to provide a reality check, and that's what this book is all about. We strongly believe that science, not only doesn't support natural causes, but actually quite points the opposite direction. So that's what this is all about. So I'd like to start with something that we can all agree on. All of us can agree on that when a living thing dies, right? Any living thing is going to die. And when it dies, there is a one-way process, and it's decay and disorder, whereby the very complex structure, the very complicated molecules, all the information content that was in that living thing decay, begin to fall apart. They lose their organization, and it degrades down to very simple, basic molecules that are left after that process. Now, of course, this is blatantly obvious. It's a natural process. This is where physics and chemistry and thermodynamics take us. It's directly observable. We've all seen it. And not only have you seen it, but you've seen it repeatedly. And it's universal, meaning it happens every time, always, to every form of life. And it's irreversible. Once you start down the path, you can't just turn the path around and go back to life. And all this is common sense. And so it becomes quite disturbing to me, actually, that a growing number of people, in fact, some very smart people, actually believe that quite the opposite has happened at least once in history, where you have, some sort of primordial soup on the earth before there was any life, or even on some other planet before there was any life. And somehow physics and chemistry performed the exact opposite, where you have very simple molecules coming together, becoming more and more complicated, becoming organized with each other, developing great depths of information content, all contained within a membrane, and you have life. And this is called abiogenesis. And that's what this is all about today. This talk is there any reality to this possibility here? And I can say up front that this is a very unnatural process, that as we saw, physics and chemistry, thermodynamics are always pushing down. There's a gravitational pull downward here, and not something that elevates this process. It is unobservable because, frankly, no one ever has observed this process. And that's kind of a hallmark of science. If you can't observe it, then what is your argument? It's unscientific then, because no one's ever observed it, and it's unnatural. And so I think it's irrational to hold this kind of a belief. And frankly, it's a faith-based belief. So I kind of scratched my head and wondering where this belief came from in the first place. So there's a little bit of history here, and some of this history will give you a feel for why I'm passionate about this. So back in the early 19th century, this man here wrote a book, and he said in that book, "There's therefore no absurdity in believing that the most simple animals and vegetables, in other words, single microbes, single-celled organisms, may be produced by the congress of the parts of decomposing organic matter without what can properly be termed generation." So in other words, no parents involved. Somehow the simplest of single-cell organisms are just produced through decaying matter without any kind of parent. That's what he's saying. So any idea who this person is? It's quite important to know who this is. This quote came from Erasmus Darwin, who is the grandfather of Charles Darwin, and no doubt a big influence on Charles Darwin. And he strongly believed that life could spontaneously generate. And his book called The Temple of Nature, I think, is quite telling, because if nature is your God, you're going to believe this is true, and you write a book called The Temple of Nature where you worship nature. So back in the early 19th century here, single-celled organisms were kind of a novelty. Nobody really knew much about microbes. And so there was a belief when they were first discovered that they must be very, very simple organisms, because they're microscopic and they just looked like little blobs. In fact, here you have Ernst Haeckel later in the 19th century. And he gives a feel here for what was thought of single-celled organisms like this. So during late years, we've become acquainted with Monera. Monera here, you could basically substitute the word bacteria, because back then they called it phylum of Monera. These are organisms which are, in fact, not composed of any organs at all, but consist entirely of shapeless, simple, homogeneous matter. The entire body of one of these bacteria during life is nothing more than a shapeless, mobile little lump of mucus or slime, consisting of an albuminous combination of carbon. Simpler or more imperfect organisms we cannot possibly conceive. So, today, looking from 2020, with 2020 hindsight, you could say, we can see this as a statement of profound ignorance, knowing that even the simplest of bacteria is extraordinarily complicated. And to think it was just a little lump of mucus or slime is a very different viewpoint than we hold today. But if this was your belief back then, you can imagine that a simple little lump of mucus or slime could spontaneously generate from some decaying matter, right? So maybe believing that life started in that manner, wasn't so crazy back then when this was the mindset. But we know better today. We're smarter than that. We know a lot more about chemistry and physics and about biology, about living things. So we ought to know better. But unfortunately, we continue to see very naive comments like this being made by scientists and very influential people. For example, we can find George Gaylord Simpson, very prominent scientist in his time. Back in 1952, we had the Miller-Urey experiment here, where some simple gases were put into, some bubbling water and some electric sparks, and they produced some amino acids. And that was revolutionary. And about 8 years later, we have George Gaylord Simpson writing this in the journal Science. He said, the consensus is that life did arise naturally from the non-living, and that even the first living things were not specially created. The conclusion has indeed really become inescapable, for the first steps in that process have already been repeated in several laboratories. This, I believe, even though it's a good 80 years after the previous quote by Ernst Haeckel, this is also a statement of profound ignorance. To think that the production of a few amino acids makes it inescapable to believe that life arose naturally from the non-living, just because you have a few amino acids, it's incomprehensible to me that you could think a few amino acids means that you could produce life. And just because you've done it repeatedly by a few labs, I don't know, this quite shocks me. And I think that the title of his paper is also very telling, The World into Which Darwin Led Us. I think this kind of a profound stupor of this statement is in fact the world into which Darwin has led us to believe such things. Unfortunately, even though this was 60 years ago, the saga continues. We have Carl Sagan. Everybody knows Carl Sagan, a very prominent scientist. And he actually said the origin of life must be a highly probable affair. As soon as conditions permit, up it pops. Astounding, because there really is no empirical evidence to support this whatsoever. But even more astounding is that Carl Sagan is rather famous for also saying, extraordinary claims require extraordinary evidence. So this is an extraordinary claim, and I don't know of any evidence for it. But he made it because he believes that life just started naturally. Then we have a prominent scientist who used to be an MIT physics professor. Now he's at Georgia Tech. This is Jeremy England. He says, you start with a random clump of atoms, and if you shine light on it for long enough, it should not be so surprising that you get a plant. I just had to pause there. Just pause there for a moment of effect. So people are still making these statements. They're still grossly oversimplifying what is required for life in order to make it seem possible that physics and chemistry can produce life. Just a few more. Here we have Bill Nye, the science guy, he wrote a book and he said the origin of life just requires some raw material that could allow the spark of life to emerge. This is not a statement of science. This is kind of a statement of faith or fantasy. I think maybe he was a fan of Pinocchio, because in that movie, of course, there was a wooden puppet that's turned into a boy through the spark of life. Of course, he means a purely natural process here. And ironically, his book is entitled Undeniable. And then what provokes me the most, actually, is, people can believe what they want to believe, but to actually force it upon our students in public schools is what I find the most disturbing. So here's a popular high school biology textbook for public schools. And it says, life began when organic molecules assembled in a coordinated manner within a cell membrane and began reproducing. And another popular biology textbook just simply says, life arose from non-life via chemical evolution. And these are statements that don't even have a hint of uncertainty to them. There's no maybe or possibly or we think or we hypothesize. It's almost like they're slamming their fist on the desk, and saying with absolute confidence this is true. And it's deeply disturbing, because to me the science is exactly the opposite. And yet these books are able to make such bold claims as this. So I think we need to start to look at the science then. What would it take to start life by purely natural processes? What would it really take? And this is where I think, my co-author Change 10 and I had a really strong synergy here, because she's the biologist. I have some biology background, but also an engineer. And I kind of put an engineering twist on it. Like, if you're going to build life, engineers know how to set requirements to get something done, to build something. So what would it take to build life? And we came up with this metaphor of a stairway, where down here at the bottom, you have simple chemistry. You just have a few simple molecules, like in the Miller-Urey experiment, and that's prebiotic. You don't have anything yet. And you're going to have to climb step by step. Each one of these steps is a requirement in order to get up to a living thing, the first living thing where you have biology going on instead of chemistry. So between chemistry and biology is this huge gap that we have to somehow span with purely natural processes. And I can tell you with certainty, this is a strong prediction, that in the years to come, as we learn more about what is required for minimal life, there will be new steps added to this stairway over time, steps that we don't know about yet. Because over time, the gap has been growing exponentially as we learn more about life. The gap between chemistry and biology is growing exponentially. And we also know that, as we already discussed, that death and decay is a kind of a gravitational force that's pulling you down the staircase, just like gravity does. And this is a very natural process. Chemistry and physics are going to pull you down here in a purely natural way to get back to very simple basic molecules after you die. And yet somehow people are trying to convince us that physics and chemistry somehow walks their way up the staircase and made it to life. And so we're going to go through some of these steps. We're not going to cover all the steps. You're going to have to, go to the book if you want more detail or ask questions. But I'm just going to pick out some of the highlights and go through some of the requirements to get to life. So let's start with the first one down here, which is the formation and the concentration of the basic building blocks. We're talking about amino acids and carbohydrates and lipids and nucleotides. These are the basic building blocks of all life. And so somehow you have to form these and you have to concentrate them. And so you'll find experiments, many of them, many published experiments, somewhat like Miller-Urey started by that concept here, where, yeah, they can produce amino acids, and yes, they can produce nucleotides in very small quantities and just under the right conditions. And so they kind of get lost in the trees and forget about the forest and claim victory for very little steps of progress toward this. But there are many things that are kind of cheating about all of these processes. And I'm just pulling out my top three here. The first one is that when they do this kind of origin of life chemistry, they're starting with a very small number of pure concentrated reagents. They go to the lab supply shop and they buy perfectly pure chemicals. put them in their perfectly pure glass container with pure water, whatever it is. It's all pure and clean. And they end up with a small amount of something like amino acid, and they claim victory. The reality in a prebiotic world would be quite messy. We're talking about a very complex mixture of many different interfering molecules. And some evidence of that comes from the Murchison meteorite, which gets a lot of attention. A meteorite fell down to the earth, and someone, looked inside of it and found amino acids. And so there's excitement there to think that out in outer space, something produced amino acids, which is really cool. But it's almost never mentioned that the meteorite also contained tens of thousands of different molecular compositions, likely millions of diverse structures in there, which suggests that the extraterrestrial chemodiversity is high compared to terrestrial relevant biological and biochemical driven chemical space. What this is saying is, for every useful biological molecule found in this meteor, there's a whole bunch of junk, a whole bunch of organic junk that is not of interest. It interferes, and in fact may even be detrimental or poisonous to life. And that's what is to be expected, is a huge mess. So in this vial, instead of 6 pure chemicals, they should start off with tens of thousands of different organic compounds, a big mess, and then see what comes out of it. Recently, just last year, there was a publication of a computer simulation called Alchemy from a Polish group. And this is computer simulation, but it's very similar. They started with just 6 prebiotic molecules and let the computer go through 7 generations of what would come out of, chemical reactions from these 6 molecules. And what they came to was over 36,000 different molecules came out of it. And they noted that only 82 out of the 36,000 were related to or found in living organisms. So basically you have 36,600 junk molecules for every 82 that are actually related to life. And this will stifle any kind of useful progress, but you don't hear about that stuff. And finally, not only do you have to pull out the good molecules you want from all of this junk, somehow you have to concentrate it then, because concentration can indeed be seen as a chemical constraint in the origin of life, since chemistry cannot operate below a certain threshold of concentration. Some of you may have learned Le Chatelier's principle, where you have to have a concentration of the reagents in order to drive the reaction forward. So this is all just my first complaint about the Miller-Urey type apparatus. Second one is that each experiment addresses only a very small portion of the overall origin of life problem, and they address it in isolation. So the way to picture this is, let's say you have to make Thanksgiving dinner. A friend of mine gave me this analogy. I love it. You have to make Thanksgiving dinner. You got to make your mashed potatoes, your turkey, your stuffing, your green beans, the pumpkin pie, the whipped cream, the ice cream, the apple pie, the coffee. The complete Thanksgiving dinner has to be made. And so typically you do each one in isolation. But for origin of life, the reality would be you've got one pot, and you've got to make the complete Thanksgiving dinner in that one pot all at once under the same conditions of heating or cooling or whatever. This is more of a reality of how you would have to do it. You can't do everything in isolation. And then finally, chemists doing these kind of experiments, they claim a victory for one-time production of a small amount of what they're trying to get, a few drops of amino acid. The reality would be, because origin of life is supposed to have happened over hundreds of millions of years, you need some type of technique that's going to do continuous production of a large quantity of the substance over great spans of time. So, let's imagine that you build yourself a chemical factory that cranks out laundry detergent, right? Of course, it takes a lot of intelligence to build that factory, and to keep it running takes a lot of intelligence and supply chain and maintenance and all that stuff. So over 1 million years, I can guarantee you that factory is not going to be still making laundry detergent. Nothing lasts that long. Nothing has that kind of stability. And yet that would be required in an origin of life kind of scenario, is to keep cranking out the same building blocks over time. So the take-home message here, and you're going to hear this take-home message a lot, and that is that concentrated building blocks, coming up with concentrated building blocks to form life is a very unnatural kind of process. It just doesn't follow from physics and chemistry. So that is actually just the first step in the stairway. And now we'll go on to the second step, which is about chirality. And I assume most of the listeners are familiar with chirality. Basically, if you have a carbon atom and you attach it to 4 different bonds, 4 different things are bonded to it, then it'll have a chiral property. So it can either be left-handed or right-handed. And if you form molecules like this in some kind of lab, you're always going to get a 50-50 mixture of left hand and right hand because these are equal energetics here. So you get a 50-50 mixture, and it's called a racemic mixture, and that is what natural processes do. So if natural processes are responsible for life, I would expect a 50-50 mixture in life because that's natural. And in fact, the reality is dramatically different than that. So the amino acids that make up proteins, there's 20 amino acids. I think 19 of them have chirality, or 18. But each amino acid then has a chiral location. And almost without exception, in living things, those are left-handed, almost always left-handed. And in fact, if you get one amino acid with the wrong chirality, then the protein can lose its function. As said here, if only one amino acid is replaced with the wrong chirality, the protein will not fulfill its tasks because you get destabilization induced by a distorted structure of the alpha helices and the beta sheets. And if you're not familiar with that, then maybe you don't know jack shits, who's the author of this paper. Now we go to beyond amino acids, which you always hear about when people talk about chirality. Not many people talk about chirality of DNA. DNA, we're all familiar with the structure here. It's a beautiful structure, and we all kind of think, it must always have this kind of structure. It's quite complicated what's behind it because you have a bunch of nucleotides all lined up perfectly. But each of the nucleotides, every single one of them has 3 chiral locations. So you could have a left or right here, a left or right here, a left or right here. That gives you 8 possible chiralities in a nucleotide, in every one of them. And yet every one of them in life, including the 3.2 billion nucleotides in every cell in your body, has the exact same chirality, the exact same, not a mixture, but the same. And it gets worse when you go into RNA because RNA has actually 4 chiral locations. And that means that there's 16 possible chiralities in a single RNA nucleotide. And yet every RNA molecule in life has the same chirality. And there's more. When you go to the phospholipids that make up every cell membrane, or big component of your cell membranes, they too have a chiral location. And in eukaryotes like us, or in bacteria, they are always left-handed. But rather unexplainably in archaea, the 3rd domain of life, they're always right-handed. And they're produced by entirely different kind of enzymes than we have in bacteria and eukaryotes. It's also interesting to know that when you die, that process of decay that we've gone through, you also have decay of this homochirality over time. You naturally move toward a racemic mixture as a living thing degrades. So you get back to 50-50. Why? Because 50-50 racemic mixture is natural. It's unnatural to have the homochirality, and our bodies are constantly fighting this battle and maintaining homochirality against all natural processes. So back to our conclusion here for this step is that homochirality is highly unnatural. It doesn't come from simple physics and chemistry. So that's the second step, and it's a requirement to get from chemistry to biology. We're now going to skip a step for the interest of time and go to one that's often ignored, is getting individual building blocks to link up in a perfect line. So back to DNA, you have this perfect line of nucleotides all linked up beautifully. And because DNA always looks like this, it kind of gives you the perception that that's just the way it has to be. But that's not true. Each nucleotide could take on different structure. In A here, I'm showing two nucleotides that are bonded together the way they are in DNA. So this is the right way to do it. But there are many ways to do it wrong. You could have this kind of bonding arrangement, which is wrong, and this, and none of these will lead to life. And so to think that you must have hundreds of thousands of nucleotides that are perfectly lined up with this chemical structure is very unnatural. And with each nucleotide you add to a growing chain, there's a chance you're going to get, oops, one wrong one jumps on the chain, therefore the chain is ruined and you're not going to get to life. So the probability just becomes astronomical of getting it right, astronomically impossible to get it right. So an analogy for this is to build a train. Let's say you want to make a train, and the train here represents either DNA or RNA or proteins. All these biopolymers of life are perfectly linked up like this, and extremely long, like, you know, the simplest autonomously reproducing living organism has 580,000 nucleotides. So it's a perfect train with 580,000 boxcars lined up perfectly, where each boxcar is like a nucleotide, or in protein, it would be an amino acid for each boxcar. And the train tracks actually help you keep things in line. If you want to add a new car to the train tracks make it pretty easy to line up the cars and connect to each other. In life, that would be enzymes, big molecules, protein enzymes, which themselves are long trains of amino acids that are required to get this job done. And you also need energy, the energy to make the cars snap together, and that comes from ATP in life. So in life, this can be done, and it is done all the time with the help of enzymes and ATP to get the train all lined up. If you go to a prebiotic world, this would be the reality in the prebiotic world, is you have boxcars, the individual building blocks that you produce from like Miller-Urey. But they're floating around. There's no train tracks. There's nothing to organize them. They're just loosely floating around. And you have to somehow connect these into a perfect line. And to make matters worse, there's some cabooses here, right? And when you hook up a caboose, that's the end of the growing train. There's no way to connect after the caboose, so it's the end. And you can also have a train that's growing in length, but the back of the train connects up to the front of the train and it makes a circle. Once that happens, you can't grow anymore. So that whole effort is wasted. And then to make it worse, as we've discussed already, that there's a whole lot of interfering junk molecules that are going to show up, like 36,000 junk molecules for every 80 that are useful molecules. So you're going to have a whole bunch of junk in here, and it's all floating around, and you're supposed to try to get 500,000 boxcars to connect and line up perfectly. You can just see how impossible that is. And this is a reality check on trying to make a biopolymer in a prebiotic world. Instead of hooking up to make a train, every time you're going to end up with a train wreck because you just can't get these things to line up perfectly. So the take-home message, once again, everybody's going to be familiar with this, is that long and perfectly arranged biopolymers are highly unnatural, very much unnatural, not something you're going to find outside of life. So we go on to the next step. And this is a step that we've addressed before on this channel, of the reproduction of biopolymers. So in order to have life, you have to have things that can reproduce themselves, right? And we know that replication of cells is an extremely complex process, and the origin of life people know that that's too complex to start there. So they talk about starting with self-replication of individual molecules like DNA or RNA. Particularly RNA comes to mind as the first self-replicating molecule. And that leads them to a concept called chemical evolution, which is where a molecule replicates itself. Occasionally, there's some errors in making a slightly different copy of itself. And then the best replicators, those that can replicate the fastest, are going to win out, kind of like a natural selection process, an evolution process, and dominate and grow more complicated over time until eventually you have a living thing. That's the sort of fairy tale that we're taught. Unfortunately, no one has ever seen a self-replicating molecule. You will see lots of claims made about this. You know, here's a book written by a chemistry professor in Israel, Adi Pros, and in his book he says molecular self-replication reaction is a reality, a reaction that actually does take place. And I find this extremely misleading. This could only be an accurate statement if you accept the most charitable and most generous definition of what self-replicating is. Just to make it clear, no one has ever seen a self-replicating molecule. A molecule that can replicate itself has never been found. 68 years since Miller-Urey, people have been trying and nothing has been found. There is a lot of claims, like you saw with Adi Pros, but nothing actually that is truly self-replicating. Here's the dictionary definition of self-replicating: something that can reproduce itself by its own power or inherent nature, or it can reproduce itself autonomously. That to me is a common sense definition. Self-replicating, it can reproduce itself by its own power. But you'll see claims made like this. This is the first paper that came out about self-replicating nucleic acid, basically RNA, self-replicates. This is from almost 60 years ago. Saul Spiegelman, rather famous, came up with this. But when you actually read the paper, you find out that the RNA is not replicating itself. It is being replicated by an enzyme called Q-beta replicase, a very complicated enzyme that takes 1,200 amino acids. So think of a boxcar with 1,200, a train with 1,200 boxcars lined up perfectly with the exact sequence of a particular protein. It's actually 4 proteins combined together to make a Q-beta replicase, a very complicated enzyme that he had to add into the solution. And this is what's doing the replication. The RNA didn't replicate itself at all. And here's another claim, a more recent paper, where they call it a self-replicating RNA enzyme. And they say here that the RNA enzyme catalyzes the joining of oligonucleotide substrates to form additional copies of itself, undergoing self-replication with exponential growth. So this sounds like, you know, they've got it. It's a done deal. We have a self-replicating molecule. But when you actually read the paper, you find out how they're stretching the definition of self-replicating to an extreme. What they actually did was they took an RNA molecule. This is like our train here, where we have boxcars. Each of these is a nucleotide lined up like a train, lined up like boxcars in a train. And it's a long train that goes all the way over here, and there's like 66 boxcars. Now, each one of these nucleotides we mentioned is homochiral, meaning they have the exact same chirality. Even though there's 16 possible chiralities for each of these letters, they're all perfectly homochiral, perfectly lined up to make a train. So this is a very unnatural molecule. It had to be produced from living organisms with some human engineering, some human intelligence in order to get this molecule in the first place. But this molecule, it's a long RNA, you call it a ribozyme because it's capable of performing a catalytic function. In the solution, they put a lot of this molecule in the solution, and they also put 2 other molecules in the solution. The other molecules happen to be 2 chunks of the same molecule. If you take the yellow molecule and you flip it vertically and then you flip it horizontally, you'll see that it looks like exactly what we have here. The blue plus green equals the yellow. just flipped upside down. And it's cool that the blue plus green has kind of the opposite nucleotides here, so that there's a pairing. These Watson-Crick base pairs form. And so naturally, the yellow molecule attracts the green molecule here, and it attracts the blue molecule here and brings them into close proximity. And then they kind of cheated here by having a triphosphate, a nucleotide triphosphate here, which is a highly energized portion of the molecule that's really ready to undergo a reaction. When they come close together, a reaction happens and these two bond together, and then you make basically blue plus green equals yellow. You make another yellow molecule. So the yellow molecule has replicated itself, if you will, by producing one single bond in, two substrates. This is a very generous, charitable claim of what a self-replicating molecule can be. It's a lot like this. Let's say I take a car and I saw the car in half. It's a fully functional car, but I saw it in half, so now it doesn't work. But inside the car, I put some strong magnets and I put some mechanical clasping mechanisms in here. And then I take another car that's fully functional and I just push the back half of the car. I push the back half into the front half and you hear this clicking sound. It clicks together. And now you have a completely fully functional car from the two halves. And basically then if I were to jump up and down and call the newspapers and say, I have made the world's first self-replicating car, you think of what an absurd claim that would be, that we don't need car factories. We just let the cars reproduce themselves. It's an absurd claim, but that's exactly what they've claimed in that paper. So to summarize, there is no such thing as a self-replicating molecule. And self-replicating molecules would be highly unnatural. I mean, if self-replicating molecules like self-replicating RNA is actually the pathway to life, if that was the pathway to life via chemical evolution, self-replicating RNAs should be pervasive. They should be all over the place. Anyone who works with RNA should be like finding them all the time. Oh man, there's another self-replicating RNA. They're all over the place. That's the way it should be if RNA world and self-replicating RNA is actually the path to life. And that's because this process of chemical evolution would require millions and millions and billions of different self-replicating RNAs progressing forward, getting more and more complicated, leading to life through that process of chemical evolution. So self-replication should have or must have been prolific if that were in fact true. And yet no one's ever seen one. So what is the science telling us? It's pretty obvious. Now we're going to jump ahead a few steps for the interest of time. Now we're going to get to an interesting topic of repairing biopolymers. This one is hardly ever addressed. But when you have an RNA or a DNA or a protein, they fall apart pretty quickly. And the really strong example here is the COVID vaccine, right? Everybody's familiar with that right now. We're all excited about the COVID vaccine. That is an RNA molecule. And you've probably heard, how are they distributing the COVID vaccine, right? The Pfizer COVID vaccine has to be, shipped, maintained at much lower than freezing. It has to be at like minus 100 degrees. And why is that true? Because the molecule falls apart so quickly. Just spending 1 week at room temperature, it's useless. It'll fall apart. It has to be very carefully preserved because it's a weak molecule. So fortunately in life, we have repair mechanisms. Water, just water itself, UV radiation, oxidative damage, chemical attacks. We constantly have degradation of the RNA and the DNA in our bodies. DNA in every cell in your body is being broken apart thousands of times every day. Fortunately, we have a dozen or so mechanisms, basically protein machines, that go around and repair these problems. So one such repair mechanism, and this is like the simplest repair mechanism, is pervasive throughout all of life. It's called the base excision repair pathway, and it's shown here. And it it takes one damaged base in DNA. This is like a straightened out DNA double helix, and you have one bad base, and you have an enzyme that extracts it, an enzyme that kind of opens up the DNA, an enzyme that prepares it, and then you have two enzymes to put back a proper base to repair it. So together you have 5 enzymes. Each of these is a complex protein on its own, and they together work together to get the job done. It's kind of an irreducibly complicated system to repair the DNA. And this is absolutely essential in order to have life, because the DNA falls apart so quickly. Now, the real challenge we come into here is that DNA repair is essential for life. That is a scientific fact. But the mechanisms in order to have DNA repair, which are these enzymes, where do the enzymes come from? Well, they are encoded in the DNA, okay? But you can't have the DNA unless you have the repair mechanism. But the repair mechanism is in the DNA. Does that make sense? How could this have started naturally? This is what's called Eigens Paradox. Anytime an information containing molecule, like DNA, gets to a certain length of, say, 100 nucleotides, it's going to fall apart unless you have a repair mechanism. But the repair mechanisms are going to take more than 100 nucleotides of DNA to be encoded in the DNA. So you can't get this to work. And this is one of a dozen different repair mechanisms in life. Now, because we have this sort of chicken and egg phenomenon here, a fancy term for that is causal circularity, right? You can't have A before you have B, but you can't have B before you have A. That's called causal circularity, and that is highly unnatural, right? Natural processes do not create things that have causal circularity. And this is just one example of that. We're going to see more. So that's the means for repairing biopolymers. Let me take a break at the moment to say that in this staircase, we have lots of required steps. I don't want to get into an argument over which step came before which step in the formation of life. I don't care what order these are in. Now, the simple fact is that all of them are required to get from chemistry to biology. So some people might prefer a different order. That's fine. You can do it however you want. I just happen to do it this way for convenience. Next step is about the membranes. You know, every living thing, every cell has to be surrounded by a membrane to separate what's inside from what's outside, because the inside of the cell has to be a consistent environment called homeostasis. You know, no matter what happens to the environment outside, you can keep a nice, consistent environment inside, and that's required for life. So membranes in all of life are made up of phospholipids, which are kind of shown here. If you take phospholipids and you place them in water in just the right conditions, they will naturally form what's called a bilayer, like you see here. And this is, everybody knows this, I think. And this bilayer will curve up and form into a sphere. And so the origin of life people get really excited about this because they think here is one requirement, one step on the staircase that is easy. It's a slam dunk. And they get quite happy about that. But those people, and I see these claims all the time, people claiming that membranes are very easy to form and phospholipids are basically the same as cell membranes. They really don't know what they're talking about. They don't understand the complexity of any living thing's membrane, because any living thing has a membrane that's extraordinarily complex compared to this. Living things, all living things, even the simplest, require membranes that are active, meaning they're expending energy in order to move stuff across the membrane and doing it very selectively. So only things that they want to come in, only things you want to get out. That's what membranes do in life. So one big example, an essential function of all membranes in life is to maintain an electrical voltage across the membrane. And this is done by a difference in the concentration of protons. You have more protons outside than you have inside. So that means the membrane has to be impermeable to protons, right? Protons can't get through the membrane on their own. They can't just diffuse across it. But that same membrane has to allow big stuff to get through, like building blocks for life or nutrients, right? It has to come in, and we have to get the waste out, right? So the membrane needs to be really tight, but yet it can't be so tight that nothing gets in or out, or else it's just a tomb where you hold on to decaying, useless stuff. So how do you meet this conflicting set of requirements, a very tight membrane and a very open membrane? The only way to do that is with the use of proteins. So a true cell membrane is extremely complicated because it's filled with transmembrane proteins that have very specific roles of bringing stuff in that you need in, or kicking stuff out that you need out, responding to stimuli, keeping homeostasis within the cell. The simplest known organism that can reproduce itself autonomously is called Mycoplasma genitalium. It's a very simple bacteria. Even the simplest has 142 different kinds of proteins that it produces that have a job of operating within the cell membrane. So this little cartoon is still very simple compared to the reality. We should have 142 different kinds of proteins working here in the cell membrane, keeping things straight. Now, another interesting point is that no cell on its own is capable of producing its own membrane. I'm going to say that again. No cell can produce its own membrane. Each cell inherits its membrane. And the cell is capable of augmenting it. It's capable of building more membrane onto its membrane. But no cell can start from scratch and produce a membrane. They don't even know how to do it, I would say. You inherit it. It comes from your parents. Now, if the cell produces a new protein that is supposed to go out into the membrane, how does a protein get inserted into this membrane? Because the membrane is rather tight, right? Well, it turns out you have to have a protein in the membrane in order to help sort of chaperone or escort a new protein to get across and be stuck within the membrane. So this is another kind of circular causality, that you have to have protein in the membrane in order to get a protein in the membrane. So to summarize this one, you've heard this line a few times, but here we go again, that active and selectively permeable membranes are highly unnatural. It's a very unnatural thing to find. Okay, just two more steps, because we're going to skip the top one. We need a means of harnessing energy. Energy, of course, is absolutely essential. And cells have to do it in a very particular way. Life, all of life requires, this is some thermodynamics here, all of life requires very high enthalpy in the molecules of life and very low entropy. So very organized molecules that are in a high energy state. This is highly unnatural. I'll say it again, it's highly unnatural to have this. Any chemical reaction that happens spontaneously would be the opposite of this. You don't have spontaneous chemical reactions that drive high enthalpy and low entropy at the same time. It just doesn't happen unless you have a way of harnessing energy through a thermodynamic engine. In other words, energy that's applied very specifically to do a certain task. Not energy like an explosion, which is very non-specific, right? Or just radiation or heat. You need something that applies energy very specifically. So in life, we have something, and this is across all of life, we have a process called chemiosmotic coupling. And what this does, basically, it takes your food and various sources of food for various kinds of life and converts it into a voltage across the membrane, right? You have more protons outside than you have inside. That's the voltage across the membrane. And that's a very complex process. We'll show a little more about that. And then that voltage is used to convert ADP, adenosine diphosphate, into adenosine triphosphate. And this is a lot like charging up batteries. So your body and every living thing is constantly converting ADP into ATP. And then the ATP, which is the charged up batteries, are used for all kinds of purposes to run molecular machines that do very specific applications of energy to keep these thermodynamics going. To give you a little feel for the first part of it, this is how do you generate the potential across the membrane. There is the respiratory complexes. This is complex number 1, and it's made up of 16 different proteins, unimaginable complexity here. And it lives in the membrane, as you can see here. And this thing actually strips electrons from the food, whatever food you've got, strips those electrons. You have an electron transport chain, a very delicate process of handling hot potatoes, the electrons coming through here. And as the electrons are stripped off, you have protons being pumped across the membrane. So you have fewer protons inside the cell and many protons outside. This is constantly pumping protons out against the diffusion gradient here. And this is one of 3 respiratory complexes that work together to get this job done. And together they require 25 different proteins of great complexity just for the process of pumping protons across the membrane. And that's incredibly complicated, and it's in all forms of life in one form or another. So pumping protons across the membrane is awesome, but it doesn't do anything unless you can make use of this voltage gradient. And that would be what ATP synthase does. It harnesses the energy of the proton gradient. So we have few protons inside, many protons outside. This is an electrical voltage which drives this electric motor. ATP synthase is made up of 20 different proteins. And it's a rotating shaft driven by this voltage. It allows the protons to get into the cell only if they drive the motor around. And by driving the motor around, we convert ADP into ATP. We're charging up those batteries. It can spin at 7,800 RPMs. So if you combine the complexity of this with those respiratory complexes, we're looking at 45 different proteins that are required to generate ATP through that process of chemiosmotic coupling. I hope I'm not losing people with all this complexity here, but just to appreciate that this is in every living organism. And, just generating the potential across the membrane does nothing for you unless you have this machine to make use of it. It's kind of an irreducibly complex system. You got to have all of it together because just having a few pieces will get you nowhere. And that's how you take energy and apply it very specifically to build up the cell and to keep life going and to maintain life. Even those DNA repair mechanisms that I talked about, they need this ATP energy. Everything needs energy. Okay, so we have one more step I'm going to go over, which is the interdependency of DNA, RNA, and proteins. So the 3 main biomolecules of life are hugely interdependent. You can't have one without the other. So that creates a big chicken and egg problem of which one came first. There are so many of these examples, chicken and egg examples in life, which the fancy name is causal circularity. But we already talked about 2, that the DNA repair mechanisms can't exist without the assistance of DNA repair mechanisms. And we talked about how proteins getting into a membrane, proteins in a membrane are required in order to get new proteins into a membrane. I mean, there's many more examples of this. There's hundreds of them, but let's just do a few. In order to replicate DNA, you've got to replicate DNA in order to replicate a cell, right? But in order to replicate DNA, you need a minimum of 25 very specific proteins that come together to make 14 enzymes. And that's the minimal set required in order to replicate DNA. And it's believed that those proteins evolved. Well, how do you evolve if you can't replicate, right? Evolution requires replication, but you can't have replication until you have these 25 proteins. So they can't evolve because you can't replicate. That's a chicken and egg problem. It's essential if you're going to make a protein that you already have proteins. And that's because ribosomes are the things that manufacture proteins. And a lot of what constitutes a ribosome is protein, but the ribosomes make the proteins. So how do you get the first ribosome? That's a chicken and egg problem. If you're going to make a protein, you have to transcribe DNA into RNA. And what does that is a protein, which is called RNA polymerase, right? But RNA polymerase, which is a protein, had to be formed by transcription. But the transcription is done by RNA polymerase. So how do you get this started? There's another chicken and egg. Ribosomes are also have an essential component of RNA. RNA is an essential component of ribosomes, but the RNA comes from a protein. The protein comes from the ribosome, but the ribosome has to have RNA. So once again, chicken and egg problem. And there's so many of them. So back to what I said before, causal circularity is highly unnatural and it's prolific within living things. So because of these things all being interdependent, the stairway to life is actually kind of the wrong metaphor. It's really more of a cliff that you would have to have all of the things all at once in order to get it done. Not just steps, but you got to have all of it at once like a cliff. Now, I wanted to address one other point here, and that is very often you hear the term protocell. People use this term to try to fill in the gap because getting from chemistry to biology, that big staircase, the big gap there, everybody knows that's a problem. And so they start to use the term proto to sort of fill in the gap. You could have proto-RNA or proto-DNA or proto-proteins or proto-membranes. And often you hear the term protocell, which is supposed to be a really simple, basic cell that got things started. And this term really kind of gets my blood pressure up because it makes no sense. The smallest, simplest living organism today, we already talked about, Mycoplasma genitalium, that can reproduce itself autonomously. It has 516 genes and almost 580,000 base pairs of DNA, okay? This is the simplest organism around today. And people imagine that there are simpler organisms in the past, protocells, which is what got us to where we are today. So these are imagined to bridge the gaps. And this doesn't make any sense because, well, let me start off by saying people are trying to simplify life beyond Mycoplasma genitalium. A lot of people are trying to knock out genes to see what is the simplest life that can reproduce itself. And the best effort to that end, I think, was Craig Venter. He recently reported, the simplest possible cell with 473 genes and 513,000 base pairs of DNA. And what he found is that as you start to knock out genes, you may be able to still have a living thing, but it becomes very dependent upon a coddled environment. You need an environment that hands everything to you on a silver platter. It's kind of like this, can a human being live without legs? Sure, you can live without legs, but you kind of need help. You need an environment that's going to make life a little easier. Can you live without arms? Can you live without eyeballs? Yeah, you can, but you need more and more help, right? And that's what's going on as people try to knock out genes. And the whole process to me doesn't make sense. And that's because life on its own is constantly working to try to simplify itself. Okay. You may know Richard Lenski did this experiment with E. coli, with 70,000 generations of E. coli in his laboratory. And he found that after only 2,000 generations, the E. coli in all 12 flasks completely ejected the operon to metabolize ribose. Now, let me explain that. So ribose is a sugar. Ribose can be used by E. coli as a source of food. But in the environment that Lenski gave these E. coli, there was no ribose. They had plenty of glucose to live on. Or at least that was the main source of food. So the DNA, the bacteria, by random mutations, dropped an entire chunk of DNA that was there to metabolize ribose, and they could survive better. In fact, they could reproduce about 3% faster because they got rid of the baggage of this unnecessary DNA. And they did better. So they're always trying to simplify themselves. They're trying to reduce their genome to be the best surviving they can. This is a natural process. So having humans come in and try to chop out pieces of DNA isn't going to be productive because it's already happening. So unless you provide a very coddled environment, we're not going to get any simpler than what we already have. And a very coddled environment is not consistent with prebiotic situation, right? You can't have everything provided for you in a prebiotic situation. So I'd like to say very strongly that protocells are imaginary. They're an imaginary entity that are used to fill in the gaps. It's kind of a materialism of the gaps approach. And they make no sense and they don't exist. Okay, so back to the full stairway. We're stepping back to the big picture now. And we know that gravity, the natural process of physics and chemistry, drag us down the stairway as we die, as we decay. And yet some people believe that the opposite is true, that you can ascend the stairway. To me, those people believing that you can ascend this stairway are well depicted by this cartoon. It says here, for years, he had spent every free moment working on his perpetual motion machine. And so far, he was up to 18 seconds. People that are trying to convince us that life began through natural processes are essentially working on a perpetual motion machine. And they can make a little progress. Maybe they can get it up to 20 seconds. And they can pound their fist and get a lot of media attention that they're making progress. But in the end, it's a waste of effort. We know enough about science. We know enough thermodynamics that we know perpetual motion machines are not natural. It cannot be obtained by a natural process, so don't waste your time trying to build a perpetual motion machine. And the same is true about a natural origin of life. We know enough, as I've shown you in the staircase and all the different examples, we know what natural processes can do. We know thermodynamics and chemistry and physics, and we know that you can't have a natural origin to life. And yet, there will always be people who believe. And so you have all these people that we talked about at the beginning and our high school biology textbooks that still believe. So I'm going to diverge for the last 2 minutes here, just talk about some philosophy of why these people believe this. And I think this analogy helps. Let's imagine that you have a dead body appears. Somebody finds a dead body. Nobody knows where the dead body, how they died. Nobody saw it, no witnesses. So, of course, the coroner comes in. The coroner is trying to figure out why the person died. And at the highest level, the coroner is trying to answer a question. What is the cause of death? And trying to answer that as two parts. Either it was a natural cause of death, like cancer, heart attack, stroke, or it's an unnatural cause of death, like a murder or a suicide. So to answer that simple question, which one of these is it? They're going to look at the evidence. And for the evidence, in this particular made-up case, there's a single wound above the left ear of the deceased. And when they open up the skull, they find extensive disruption of the brain tissue. And they recover a single foreign object. And it was composed of lead and copper. And it looks like this. And of course, any coroner would know that this used to look like that. That's how it got started. Now, this evidence seems pretty clearly to point in the direction of an unnatural death, right? I hope everyone will agree with me. Unfortunately, this coroner has a very strongly held belief. This coroner believes that under no circumstances ever has anyone died from an unnatural death. It's just not possible. That's what they believe. And so they see the evidence here, and they're trying to get it to point to a natural death because that's what they believe has to be true. So they dig a little deeper. They're trying to explain this, and they find out actually that the body was found in the vicinity of a volcano, an active volcano. And they think, aha, now I can explain it. The volcano spit out this foreign object, which unfortunately ended up in the head of the deceased. And so the cause of death was a volcanic eruption, a natural cause of death. Now, if you think that this is a bad coroner, if you think this is bad science, I agree with you completely. But I ask you then to ponder this question of how did life first begin? Was it, and there's a simple two-part answer here. It's either a natural source, simple physics and chemistry doing what they do, or did life begin from a supernatural cause? So let's look at the evidence, right? The evidence is everything I've been presenting over the last hour. You have homochirality, which is very unnatural. You've got cell membranes of extreme complexity that are active and selectively permeable, very unnatural. You have the energy harnessing process. You have protein repair. You have, DNA repair processes. All of these chicken and egg circular causalities going along. And in fact, the entire stairway to life, all of these steps. Each of these pieces of evidence strongly points away from natural and strongly, therefore, points in this direction. And that's what the science says if you're free to follow the evidence where it leads. Unfortunately, we are in a society where science is defined as only being able to address natural causes. Science is defined in a way where it can only speak to natural causes. In other words, this cannot be considered in a part of a scientific investigation. So, rather than thinking what is actually the answer to this question, the scientific method as we've defined it is seeking the best natural explanation, and that's all that's allowed. In other words, we've constrained science so that it's not actually capable of getting to the truth. It's blocking out one of the possible explanations, and it's only possible to obtain a natural cause because that's the way it's been defined. And so the conclusion that you hear in newspapers and media, in many scientific manuscripts, is that life started naturally and the case is closed. And that's because we've started off by blocking out the other possibility. And what I think is even worse here is that in our schools, the blocking of this possibility is actually concealed. It's not like they explain to the students, hey, it is possible that there was a supernatural cause to the start of life. But for now, we're just going to think about natural causes and we're going to do science focusing on natural causes. They don't explain it that way. All they say in the schools is that science encourages a belief. They're going to say science points to a natural explanation for life. That's what they're teaching our students. And I think hiding this is actually making things much worse. So, wrapping up here, why do people believe in abiogenesis? Well, science has been defined in a way where it can only point to natural causes. Science is not really concerned with finding the truth. It's concerned with finding the best natural explanation. And I think another reason why people believe in abiogenesis is that atheism has no rational basis unless you have a natural cause for the beginning of life. You've got to have abiogenesis in order to be a rational atheist. You can certainly be an atheist just because you feel like it, but you probably won't have that comfort of knowing that there's a rational basis. So I end with this quote from Miller-Urey of the famous Miller-Urey experiment back in 1952. He said, "All of us who study the origin of life find that the more we look into it, the more we feel it is too complex to have evolved anywhere. We all believe as an article of faith that life evolved from dead matter on this planet. So he said it's an article of faith, and I completely agree with that. It's an article of faith. And thank you all for listening for a long presentation. And that's all I have to share, and I'm happy to discuss and answer any questions.
Well, I have to say, Rob, that was an absolutely amazing presentation. We've got a lot of kind words in the chat. We've had over 30 people the entire time. So you've kept everybody's attention. Looks like we got 35 people right now. Lena says, legendary presentation. Sal says, bravo, Rob Stadler. This is the best ID-friendly presentation for biology ever. So, and I want to point out those slides, Rob, I loved them. Great visuals. Kept my attention the entire time. Very impressive. So great work. And I love, I'll say, that just officially put an end to abiogenesis and naturalism. You know, I
wish it were so.
And I love your materialism of the gaps point. You know, that is a lot of what I see personally in terms of their rescue devices, especially with this imaginary protocell appeal that you covered in your presentation for a bit there. I do have a ton of questions, actually. We've had a very lively chat, but I want to hand it over to George. George, if you had any comments, brother, go ahead.
Yeah, well, Rob actually had me in stitches with not knowing jack shit. But I have to say, Rob, I don't know jack shit, but I know his brother, chicken shit. No. For those that are listening, look, I ordered the book some time ago, but they told me that it would take a few months to get to me, so I ordered the Kindle version. And for those that haven't read it, please do so. There are so many other things that Rob hasn't mentioned that are in the book that will really open up your eyes. I was surprised, actually. We all know about the DNA repair system, but reading the book, I was surprised that there's actually so many specific or a variety of specialized DNA repair mechanisms to counter so many of the daily attacks on our DNA. And I'll just mention some of those. The base in excision repair system, the nucleotide excision repair, the homologous recombination repair, the mismatch repair, the photoreactivation, the non-homologous end joining, the translation synthesis and processing by the mRN complex. I never knew about any of these. So I urge people listening, get the book, read it, and you'll be amazed at some of the information that's in there.
Amen. I couldn't recommend it more. It's a phenomenal book, Rob.
Thank you. And you know, it's what's astounding to me is you see this this conflict between belief in RNA world hypothesis, which is that over millions of years, RNA molecules became more complex through self-replication and led to life. And yet here we are with the COVID vaccine, and the COVID vaccine being an RNA molecule that has to be kept at minus 100 degrees or else it falls apart. You know, it can't survive at room temperature for like a week. And yet we're supposed to believe that RNA, you know, bumbled around on the earth for 100 million years, becoming more and more complex until we have life. Come on, people, wake up.
Yeah,
well,
Rob, I've heard numerous biologists tell me that I won't name them, but they say that biology is equal to chemistry plus energy. But what they're forgetting is the many layers of information that are embedded in there. So they have to explain the origin of that information. And if I can quote Werner Gitt, most people probably know Werner Gitt. He says the grand theory of evolution would gain some empirical support if it could be demonstrated in a real experiment that information could arise from matter left to itself without the addition of intelligence. Despite the most intensive worldwide efforts, this has never been observed. So they have to figure out where that information came from. And we know that information can only come from a sender, an intelligence.
And I think you know that Perry Marshall has this $10 million prize
Yes.
for someone who can demonstrate what you just suggested as a challenge. Yes.
Standing, I don't know if you caught most of the questions, but I caught some of them. Do you want me to ask one question that logical, plausible, probable, dear friend John Maddox asked?
Sure, yeah, go ahead.
Yeah, John asks, you mentioned almost all amino acids are homochiral. Are there exceptions in DNA/RNA, or is it exclusive?
So DNA and RNA made up of nucleotides, those are all chiral molecules, right? They all, like DNA always has 3 chiral locations, RNA always has 4 in every nucleotide, in every living thing all the time. There's no exceptions for that. But in amino acids to make proteins, I believe alanine is the most simple amino acid, and that is not a chiral molecule because it's very simple. It doesn't have 4 different attachments to the central carbon.
Yeah, he follows it up by saying, do cells separate chirality or create it?
I think both is the answer.
Okay.
There are special enzymes called epimerases or racemases that are enzymes that living things produce that specifically change the chirality and enforce homochirality. And of course, interesting to say, those enzymes themselves are homochiral, right?
Yeah. Over to you, Standing.
Yes, I've got a question here. Godzilla Freak question. How would you respond to those who say that future research will find how abiogenesis can happen, which I've personally heard over and over and over again by the critics?
Yeah, you clearly see that. That claim is made constantly. I think I have a simple response to that, and that is that, and I think you saw in the beginning of my presentation, back to the Miller-Urey experiment, that's 68 years ago. That experiment really kind of kicked off a lot of research, a lot of interest in origin of life. And in those 68 years, the goalpost, right, which is producing a living self-replicating cell, has pulled away exponentially because we've learned so much about the complexity of life in 68 years. If you look at the staircase with those 12 steps that I talked about, 68 years ago, I don't think we didn't know 11 of the 12 steps existed, right? We hardly knew what DNA was back then. And so back then there was only one step and they thought Miller-Urey pretty much took care of that step, so that George Gaylord Simpson was pretty convinced that it was a done deal. In 68 years, we've added 11 steps, and I'm sure there's actually more than that. And the progress in laboratory efforts to make life has been extremely minimal compared to this exponential growth in the goalposts, right? And I can predict very strongly that the next 100 years is going to do exactly that again and again and again. Even in the simplest form of life, and this is from Craig Venter, in that, he tried to simplify life. He found that 20% of the genes in the simplest form of life he came up with, 20% of the genes, we still have no idea what they do. We don't know what they're doing. And I can guarantee you that when we figure out what they do, it's going to add steps onto the staircase. And that is also just looking at the DNA information. And we know that life has a lot of epigenetics, a lot of information above and beyond DNA that we still haven't explored, like the sugar code and histones and all. There's so many codes in there that we haven't explored yet. So we're going to be adding a lot of stairs onto the staircase. Meanwhile, the origin of life community will still be back at step number 1, trying to find a way to make the building blocks of life and concentrate them.
Well, that's a great response. That's a really good response. Yes, the more we discover about the complexity of the genome and the DNA molecule is only going to add steps while they're still stuck on step one. And I really enjoyed, well, I enjoyed your entire presentation, but I like how you pointed out the chicken and egg problems that abound, right? Chicken and egg problem after chicken and egg problem. You touched on the DNA repair enzyme and how it's essential for life, but the mechanism for DNA repair are encoded in the DNA, right?
Yeah.
So how did this have started naturally? And one analogy that I liked earlier on in your presentation, Rob, had to do with the Thanksgiving dinner. You've got your mashed potatoes, your stuffing, your gravy, your apple pie, and you got to make everything in isolation, for example. But with the origin of life and all these experiments, in a real origin of life situation, if it happened, you have to make your entire Thanksgiving dinner in one single pot. So isolation isn't going to work, as you pointed out, which these abiogenesis experiments tend to actually do.
Yeah, they're all in isolation like that, and they claim victory for some little progress made in isolation. And they expect people to kind of glue together those little success stories into believing that the whole thing is a possibility.
Right. Well said. Well said. We've got our brother Sal here, Evidence and Reasons. Anybody who has not yet subscribed to Salvador's channel, please do. He's putting out some great content. His question for you, Rob, he's curious, any more books or videos in the works?
I hope so. I'm hoping with the group to produce a series of animated short videos that explain this kind of in lay terms. It's hard to do a presentation like this because you don't know the level of the audience that you're trying to reach. So you got to try to reach people on all different levels, but we want to make kind of animated short videos that explain it to the lay audience. And that's been a big project for now.
Rob, one of the things I find amazing with biology books, when they quote the Miller-Urey experiment, they always tend to say they were successful in producing 2% of that mixture as amino acids, but they never tell you that it's racemic, that it's a 50-50 mix, and that simulates death. It doesn't simulate life.
Right. Yeah, it's amazing. If you're out for a walk in the woods and you come upon on a rock somewhere, you come upon some amino acids, you could actually take them to the lab, and if the amino acids are homochiral, then they came from a living thing. And if they're racemic mixture, then they don't have anything to do with life. So that's how definitive homochiral is in separating living from non-living things. And in the last 20 years or so, there have been some chemical processes that can move racemic mixtures toward homochirality. I don't think they can even get there, but they can get to maybe 90% of there. The most famous, I think, is the Soai reaction from Japan. But when you actually look at the chemistry behind that, it's extremely not prebiotically plausible, right? The kind of chemistry that goes into that is some nasty, very unnatural chemistry that can, it can move you toward homochirality, but can't get there. But somehow that kind of recent science is enough to convince some people that homochirality is not a problem anymore. But again, they're just so willing to accept what they find. Like, no one, I think, has talked about the chirality problem with DNA and RNA, which, as I explained, is much more difficult than with amino acids.
I've got a question here, Rob. I can't put it on the screen, unfortunately. This was from earlier on in the in the lecture. It's from Brandon, and he asks, for the skeptics that haven't read his book or page 214, can Dr. Stadler refute the silly God of the gaps argument? Oh, it's so hard to do, therefore God must have done it.
Yeah, good question. Thanks. I get that a lot. People say, "Oh, you're just doing God of the gaps argument all the time," and so God of the gaps is basically saying I have no idea what's going on here, and therefore God did it. So you're filling in the gap of lack of understanding with God, and so people often claim that that's my approach here. We don't know what's going on, so it must have been God. And I'd like to say, really, it's the exact opposite of that. That we know enough. I've been trying to say this in the talk, is we know enough about biology. We know enough about the minimum requirements for life and what living things have in their complexity. We know enough thermodynamics. We know enough chemistry. Bringing that all together, we know enough to know that natural processes cannot have accomplished this. So there's no gaps. There's no hand waving here. It's that we now know enough to say that natural processes can't do this. And then I think the opposite for those who believe that life started naturally. I think that they're using a materialism of the gaps, basically saying, we know that natural processes tend to degrade life over time to simple molecules, but I believe that simple molecules through natural processes complexified to become life. I have no idea how that could happen, but I know it was a natural process.
Right.
That's naturalism of the gaps.
Right. Naturalism and materialism of the gaps. Great response. I apologize, George, if you had a question, maybe right after this one. This one just came in, and I can at least pop it up on the screen. Question from Praise I Am That I Am. What abiogenesis hypothesis, in your opinion, Rob, is more absurd? The primordial ooze or ocean vent hypothesis?
Well, this question takes me on a little tangent, and I think you had John Sephardi on your show at some point, and I remember he mentioned that one of my favorite papers on this too. There's a paper on abiogenesis, and I think it's titled like the RNA world hypothesis, the worst hypothesis for abiogenesis, except for all of the others.
Yeah. Oh yeah, I remember.
That was actually the title of a scientific paper. So to try to answer that question, I think they're all the worst. I can't. I mean, I don't have a favorite. When you talk to people who support abiogenesis, they all have their favorite hypothesis, and they'll rip on everybody else's hypothesis. But
right. Yeah, they're all debunking each other because they're all the worst, essentially.
Yeah, right.
One's
just worse than the other. George.
Yeah. I've got a question here from Hate Love Nothing. His question is: are there any current technologies that are capable of altering the way in which the information is handled? Would this change anything, or is the information not tangible?
Are there technologies that change the way information is handled in living things?
I'm assuming so, yeah.
Yeah, I just read yesterday about how one of the COVID vaccines works, and I found it quite fascinating, really. But so it's an RNA molecule, but somehow if you inject RNA into the body, and it's the normal kind of RNA that has uracil as one of the nucleotides, it's much more likely to be degraded, that the body will attack that and break it apart, and your vaccine will come to nothing. But the intelligent engineers that made this vaccine, they actually replaced all of the uracil in the RNA with something that's very close to uracil, but it's kind of like a masquerade, slight change to it. And somehow the body doesn't recognize that, and then it allows it to pass through and get to the ribosomes and actually produce the protein, which is the intent of the vaccine. So there, maybe that's kind of answering the question, that they're kind of tweaking the way the body is dealing with the information and fooling it in some ways in order to have the vaccine work. It's kind of fascinating.
That's a great response, Rob. I've got a question here from this is Tony. He's helped us out with some of these interviews as well in the past. SFT, can you ask Rob about the highly manipulated purification processes that chemists employ to isolate their amino acids? I know this involves pH manipulation, etc.
Yeah, if you want to get into details, I'm not the right person to get into, how you do separation and purification, but we could phone a friend maybe. But I know that it's essential for, it's essential to purify and to filter out all the junk that I mentioned and get to the one thing you have, and that's done in these manuscripts. And they don't explain how that could have happened in a prebiotic situation, but they do it in the laboratory, and that's a kind of cheating,
right?
That takes you away from reality. I've never seen anybody try to explain that in a prebiotically plausible situation.
Right. I completely agree. One thing I like about your book too, I mean, so many the things I like about it, but I like the ending where you address some of these anticipated objections, right? Because a lot of times it's the same talking points over and over again that are being employed. And you mentioned manuscripts there. And my question has to do with a question that you did answer in this section, but it's because I constantly hear it all the time in discussions, debates, or responses to interviews and lectures where the critic will point to, as a form of an argument, they'll point to the 100,000 peer-reviewed scientific manuscripts that have been published on abiogenesis as some kind of counter rebuttal, maybe insinuating that we are ignoring some of these papers that apparently have given evidence or credence to abiogenesis. What's the best way to respond to that, I guess, is my question, Rob.
So, good question. Unfortunately, we're in a situation where the literature is extremely biased. It's biased because only one answer is publishable, right? This gets back to when I put the no smoking sign on anything supernatural, right? So science has defined that the science can only address natural causes, right? And so only natural explanations are considered science. Only natural explanations can be published. And frankly, we all know the bias of scientific manuscripts, that positive results are going to get published and negative results don't tend to be published. They tend to be thrown away. So yes, you have thousands of manuscripts in the scientific literature, peer-reviewed literature about abiogenesis that are encouraging. They support natural causes because they have to, and they're positive generally because that's what gets published. So there's an incredible bias to that, and there's no balancing out there. So in writing this book, I'm attempting to provide just a little bit of balance to this, because otherwise you get a one-sided story. It's almost a propaganda, and that's all that can be published. And you also have the problem of the media. Then the media comes in and they hype it up, right? They're always kind of hyperbole applied to what they see. And they make it sound like it's a done deal. Everything's been explained. We have self-replicating molecules, on and on and on. And the media needs to take some responsibility, I guess, for falling for the clickbait kind of hyperbole that they generate a lot.
Right. A lot of it is clickbait. You're absolutely right. And, I can't thank you enough for all the amazing work you've done in not only this book, but your previous book on evolution. And a lot of the people in the audience today, they look like they have read your book. And they own your book. So, but I want to point out to anybody in the audience who may not have read it yet, I've got both of your books, Rob, in the description box of this video, as well as our other discussions on these topics. So please look into it. It's a must read. It's a must have. Personally, my last question, and then I'll hand it to George, and we'll start winding it down, Rob, because you've given us almost 2 hours of your time, and I can't thank you enough. I hear this all the time as kind of a counter rebuttal or lack of, in that, we are hindering further research and possible advancement on origin of life, on abiogenesis. They're saying, okay, maybe we don't have an answer now, but what do you want us to do? To just stop? And I believe you actually do answer this in your book as well. I'm curious as to your thoughts, because I personally have heard that a lot, that we're hindering scientific advancement.
Mm-hmm. Well, two things. First is I can think of thousands of things that science could be applied to that are more beneficial for society, right? I mean, if you have $1 million to spend on science, you got cancer, you got COVID, you got all kinds of problems that society faces that science can improve our lives and make things better. So I would hope that the vast majority of the money goes to those kind of useful things, and almost nothing goes to abiogenesis, right?
Right.
It's a waste of money. But the second thought that comes to mind goes back to what I mentioned in the talk, is that we don't, as a society, we don't invest any grant money that I'm aware of into finding a perpetual motion machine, right? Because we're smart enough now to know, we know enough science to know that that is a waste of money. Maybe there's some crazy rich person who's investing in it with his own personal funds. I don't know. But in general, hopefully our government and big agencies are smart enough to know that that is not a good use of research money. And in my strong opinion, any money dedicated toward abiogenesis research is very similar to that. It's not productive for society, and it's basically throwing your money down the hole.
Amen. Amen. Well said. I really wanted the audience to hear those words. That was a great answer. It's a waste of money, and what you said really stands out to me in the fact that the money is far better put into research programs on cancer, fighting disease, the COVID, everything to do with COVID that we're experiencing right now. There's far more important things for us to study and research than abiogenesis, which, as we've seen today, has been dismantled, has no evidence for. So great response. George, I'll hand it over to you if you have some final words or a couple final questions.
Yeah, I've got a proposition regarding consensus science. It's a 4-step process. Step 1 says every scientist accepts evolution is a fact. Step 2, one scientist comes out and says, I don't believe evolution is a fact. Step 3, the guy is censored, thrown under the bus, loses his funding, he's sacked. And step 4 now says, now every scientist accepts evolution as a fact. That's consensus science for you.
Right, that's brilliant.
But I've got one question, Rob, before we finish off. I read somewhere about a 4-stranded knot of genetic code. Apparently, it's been controversial among scientists for years. There was a journal, Nature Chemistry, provides the first direct evidence that it does exist and that it may play an important role in regulating our genes. Now, I'm always careful to accept something when it says may. Can you expand on this if you're aware of this paper?
I'm aware of it in that I've just heard about it. Maybe I read the abstract, but I can't speak to the details of it. But we certainly are finding over time, we're learning more and more about layers and layers of information in DNA that's beyond just the simple one-dimensional code that's contained in there. That the way the 3-dimensional structure of it, and the way some parts of DNA come in proximity to other parts. And as you said, at some points you can have actually instead of a double helix, you can actually have a quad. That there's so much we need to learn, and this is a good use of money, right? This kind of research to understand these things. I was just reading today about how we in prokaryotes, they don't have organelles in the single cell bacteria and so forth. There's not organelles. But now they're thinking that you have what's called condensates, which is pockets where a bunch of proteins come together, and it's kind of like a drop of oil in water kind of thing, where you have a condensate that is very important for the function of that cell. And we're just beginning to learn about that you have areas that are kind of membraneless organelles. Anyway, I'm getting off on a sidetrack, but there's so many things we still need to learn. And that's a very good investment of money, because understanding this stuff will lead to disease cure and so forth. That's very important for society. So let's put our money into that.
Yeah, I was at the grocery store the other day, Rob, and I asked the store worker there. I said, "Are these genetically modified eggplants?" And the store worker says, "Why do you ask?" And immediately after that, the eggplant says, "Yeah, why do you ask?"
I don't know where
sorry,
man. But keep it up, brother. Keep it up.
It's good to have some levity in an otherwise heavy topic. So thank you, George.
Yes.
Yeah, a bit of relief. Yeah,
lightens it
Good
up.
on you, Rob. We all love you, Rob.
Thank you.
Yeah,
Appreciate
we love
it.
you. And this was the type of presentation where I'm probably going to make a coffee after this and re-listen to it and take some more notes, because wow, it was so informative, and the visuals, the slides, everything about it was awesome. I mean, we're going on 2 hours. We still have almost 40 people in the chat, so we could probably ask you questions for 10 hours, but we're not going to do that to you, Rob. You've done an awesome job today, and we appreciate your time, and this was awesome. I want people to share it around, please. We want to get this information out to as many people as possible. And once again, thank you, Rob. I want to give you the floor for some final words, some final thoughts before we close it down.
Well, thank you. Thanks for your time, everybody. I think you saw in the last slide I presented, there's an email address for me. You can send questions that way or contact me. So I appreciate that. And thanks everybody. Remember that life is just chock full of very unnatural things that clearly separate, and this is very different than, years ago, there was what was called vitalism, which was a belief that, life fundamentally was constructed of different molecules and different processes than non-life. And that, of course, went away as we found, through reductionism, we found that the molecules of life, the atoms and so forth, are really no different than in non-life. But I think we overdid it. I think the pendulum swung too far, in that, as you saw in my presentation, there really are some very special things about life that we need to appreciate, that separate it from non-life, such as the homochirality.
And Rob, I'd like to finish off by wishing you a happy new year. Hopefully 2021 will be a better year, and God bless you.
Ladies and gentlemen, welcome back to Logical, Plausible, Probable. Today, I am pumped to be bringing you the first and hopefully an ongoing series of interviews with the one and only Dr. Robert Stadler, a man of some serious knowledge, credentials, scientific accolades, and all-around critical thinking skills. Now, for those of you who may instantly want to discredit the arguments that we will be discussing today, please keep in mind that the good doctor received a PhD in medical engineering from Harvard MIT Division of Health Sciences and Technology. He has been a scientist in the medical device industry for over 20 years, and literally, not figuratively, literally millions of his cardiac devices are implanted in millions of patients worldwide. He's also been elected fellow of the American Institute of Medical and Biomedical Engineers, and has obtained more than 140 U.S. patents and has 20 peer-reviewed publications. Doctor, thank you so much for sharing your time with us and coming on to share your knowledge.
Thank you, John. It's a pleasure to be here. I really love your passion for this, and I appreciate what you're doing with this channel.
Well, thank you so much for that. And it's it's fun. It gives me an excuse to go and make inquiries of people like yourself that normally I never would get the time of day on. It's a complete selfishness on my part to want to be able to interact with PhDs who usually wouldn't come talk to laymen. So I appreciate that.
My pleasure. Good to be here.
Now, today we're going to be discussing some of the fundamental questions raised in your first book, the scientific approach to evolution, what they didn't teach you in biology. And this is something I strongly recommend to anyone who's actually considering expanding their knowledge, but in terms of biology and intelligent design, these different facets, but also who are looking for a very analytical breakdown of the standards of evidence for scientific research and their application. And in my opinion, I think it's something I will talk about, is the suspension of these standards in relation to evolutionary theory. Now, before we get into all of that, I'd very much like to know, and I think the audience would as well, what led you to write this book and applying the very detailed level of analysis to a supposedly proven consensus on Darwinian evolution?
Yeah, as you mentioned in my background, I've been a scientist in medical research for 23 years now. And when you're trying to produce medical products, a product that will be a part of someone's medical care, the FDA is there, the Food and Drug Administration, and they require strong evidence that whatever you produce is safe and effective, right? And I think medicine is a field that has very strongly appreciated good science, and that's because it's life and death. When you're taking a pill or going through some procedure, this is your life on the line, and so science better be right. So medicine,
especially now in the modern era of lawsuits, you better be you better be golden, right?
Yeah, and of course mistakes are still made, and it's not perfect. But medicine is an area that really appreciates good science and has built up these criteria of what it takes to be confident in some result from science, right? And so with 23 years of dealing with FDA and going through this process, it's kind of become ingrained in my head as to what kind of evidence is good at providing confidence, right? So that with that backdrop, then my son became a high school student in public school, went to biology class, and I was curious about his teaching there in biology class. So I read his biology textbook, and I knew what would be in there, but it still surprised me as I read it that the evidence which is presented in favor of large-scale evolution, molecules to man kind of evolution, it actually met none of the requirements that I got very used to being held to in my field of research. And so that dichotomy kind of struck me, and I said, I got to write a book about this, because I've never heard someone describe it this way. But it just hit me like a brick in the face that there's a double standard here that needs to be exposed. And to make it clear, it's not, these criteria we're going to talk about today are not just for medicine. They're for all fields of science to determine the level of confidence that your evidence provides.
It's interesting as you're talking about that, and I think that you alluded to this directly in relation to your son and his biology textbook. I mean, the supposed proof of the consensus that is presented in textbooks and even all the way up into high levels of academia, I mean, to me, it's reached borderline dogma status in so many ways. And as you just alluded to, and something I found very interesting throughout the book was how you made it a point to apply those 6 criteria that in any other context, as you just mentioned, are required to reach high confidence in research, and how they related relate to the evidence for or against evolution. So before we dive into the more detailed aspects, I'm going to bring this up on the screen. Can you kind of give us an overview in general of how the criteria for high confidence science is usually approached in terms of research and like things that you're doing, medical device research, for example?
Yeah, thank you. Yeah, so in the field of medicine, these 6 criteria are commonly applied. They're not articulated exactly as you see here, but the theme is very clear throughout medicine in particular. And medicine, as I said, appreciates these the most, perhaps because it's life and death. But these apply to all fields of science. And so I'd like to explain these criteria maybe with sort of a black and white example of, on the one hand, asking a question of science where all these criteria are easily met, and you end up with high confidence. And for that, I'll use the example of simply dropping an object and trying to assess how fast the object is going to fall to the ground, right? That's an experiment we all do all the time. And then the contrasting example, which cannot be addressed with high confidence, would be an evolutionary question, such as, do humans share a common ancestor with something like bacteria, right? That's a belief of large-scale evolution, and we're going to ask, is that backed by high confidence evidence, right? So the first of the criteria you see there is called repeatable. And of course, if an experiment, if evidence can be generated repeatably, that creates higher confidence in the result. So the object we're going to drop, and we're going to try to determine how fast it falls. That is an extremely repeatable kind of evidence. You can do it again and again and again, and you can give it to someone else and they can repeat it. You can give it to someone in another country and they repeat it. And by getting the same result again and again, the confidence level rises. That one's quite simple, right? But contrast that now with this other question of, do humans and something like bacteria share a common ancestor? Well, is that repeatable? You can't possibly repeat the process that produced the bacteria and then supposedly much, much later produced the human. That is a process that supposedly occurred once, of course, over many, many, many millions of years, and it can't be repeated. It's not a repeatable process, so we have much lower confidence. The second one is directly observable. When I drop the object, it is, in fact, directly observable what happens. I can see it with my own eyes. I know what's going on here. It's right in front of me. And again, the opposite is true for the evolution case. The supposed evolution from a common ancestor to bacteria and then to humans is not at all directly observable. It's if at all, extremely indirect, any evidence you have to suggest that. Number three is to do things with a prospective study. And what that means is that you design a study in advance, you plan the study, and then you conduct the study according to your plan. Now, why is that important in science? It's important because by designing the study in advance, you can plan to block out anything that could confound the results. So with the example of dropping an object, I can plan the study so that I'm in a place where there's not a strong wind that would get in the way. In fact, it'd be even better if I get all the air out of the room. So I could plan to have a room with a vacuum that sucks all the air out of it. Then I drop the object and I've gotten rid of this interfering confounding factor of the air in the room. So I can do a much better job at getting to the truth. When we're talking about the supposed evolution from molecules to man, that is certainly nothing that you can study prospectively, at least on the scale of that scale of evolution. Number four is to try to avoid bias. Now, we all have bias. All of us do, all the time. And you have to actively try to block bias out of your study, out of your experiment. When it comes to something simple like dropping an object and seeing how fast it falls, I would say there's hardly any basis for bias there. It's not like I'm going to win $1 million if I get a certain answer, or I'll have a much better career, or, we're all kind of indifferent to that result. On the other side, when you look at evolution, there is a very strong bias in all of us, depending upon your worldview. If you're more of a naturalism or atheistic worldview, you're very biased to say there must be a natural reason for all of life. And the opposite is true, of course, if you have a faith, a Christian or a Muslim or whatever theistic view, you also have that as your bias. But that bias, in the case of evolution, it can sometimes be, quite strong and can kind of overwhelm the weak evidence that you can actually accumulate. Number five is to try to avoid assumptions. And assumptions are what you do in science when you try to fill in a gap that's unknown, or you're trying to take a shortcut. So in the dropping of the object, maybe maybe we want to assume that the air that's in the room is just not a very big factor. And it's so hard to take the air out of the room. Let's just leave the air in and we'll assume it doesn't matter. And you always take some assumptions when you do research, but it's important to state those assumptions and to justify them and to understand their impact on the result. With the evolution example, there are just a host of assumptions built into that. And just to give a simple example, when you look at, say, the fossil record, one assumption is that if a given kind of life form is not present in a given layer of sediment, if you can't find that kind of life form, that means that that life form wasn't around at that time, right? And that's a common assumption. But that group that makes that argument often sometimes also makes a counter assumption, which is that just because a transitional form can't be found in the fossil record, that doesn't mean that transitional form didn't exist at some point, right? So just because we can't find a fossil of it doesn't mean it never existed. So those are two assumptions that are actually contradicting and conflicting. And it just gives you a feeling for how many assumptions are built into the evolution from molecules to man. And then finally is number 6, which is what I call a sober judgment of the results. This is how you kind of package up what you've learned and what you've found. And you present it in a way that doesn't embellish the result and it doesn't state the result beyond the experimental conditions. So, for example, when you drop the object and I learn how fast it falls, it would be wrong for me then to assume, how that object will behave if I were standing on the moon or on Mars or on the top of Mount Everest. I'm going to present the results given the conditions that I studied it under, and my results apply to those conditions. That's a sober judgment of my results. Whereas on the evolution side, a sober judgment of results should put into perspective what confidence you actually have in the data you have, which is really quite weak. And you can't extrapolate, you can't embellish and build a story of it. That's not a sober judgment of results. So we see a black and white example here between those two cases.
So it's kind of like in so many of the, high level academic papers I've read on, a variety of topics, but in the discussion portion, there's a whole, I joke about this, but I say the first 9 pages, they don't mention evolution one time in relation to whatever it is they've discovered and have been analyzing. And then in the conclusion and discussion, it's, oh, well, it must have evolved over X, timeline. And it's like, I think it kind of dovetails with everything you just talked about of like, wait a minute, how did you come to that conclusion? The storyline that you were referring to suddenly become very, very creative.
Yeah, very creative. And you've probably noticed then that the news media picks up on that and they kind of amplify and embellish those results. And it just kind of grows and grows into something that's well beyond the original scope of what was studied.
One of the points you make throughout the entire book, and before I get to this very important point, for those of you who are thinking, hey, I need to get this book, the title is The Scientific Approach to Evolution, What They Didn't Teach You in Biology. And I've got a link down in the description, so go get it. It's well worth the time to read it, and will definitely give you a very new perspective. But the point I really want to hammer home to the audience as we get ready to transition into specifics is how the standards you've outlined are applied by the FDA for any of your medical research. You kind of mentioned that earlier, but is that only applied to medical research, or is this logic and these standards, are they in place for pretty much any other field of science? But as we've been discussing, are being tossed out the window in relation to evolution. Did I misinterpret that, or is that?
No, you're right on it. They apply to all fields of science all the time, and they give you a perspective of the level of confidence. Now, some fields of study just fundamentally can't reach all of those particular 6 criteria, and they're not meant to be black and white. They're kind of a spectrum of colors, right? But let's consider the example of I want to study black holes in space, right? I can't ideally, to do those 6 criteria, I would bring a black hole into the laboratory, design experiments, poke it, prod it, and learn from what is a black hole, right? That would give me great confidence. I could do it repeatably and so forth. But of course, that's not practical. It can't be done. And you can't even see a black hole. A black hole is kind of the absence of electromagnetic radiation. So we learn from black holes in a very indirect kind of way, and that gives us lower confidence, and that's the best we can do. But because that's the best we can do, that doesn't give you the right or the license to slam your fist and say, I have high confidence in this. It's not really.
Consensus says if you disagree, therefore you are not using ration, you're being blinded by faith, which is one of the, a paraphrase of some of the common retorts that I get in relation to any kind of challenge. Now, before we get into some of the really juicy aspects of your book, something you discuss with substance, and honestly, I think is taken for granted by the average person, are the fundamental lines of demarcation between the concepts of micro and macro. Could you give us a kind of quick explanation of the differences and perhaps address the common argument from evolutionary biologists that people who reject Darwinism overall just refuse to accept any possibility of modifications between generations, for example.
Right. So there are terms microevolution and macroevolution, and micro is often defined as being changes that occur within a species. I don't particularly like that definition. To draw a line at the species level, I'm not very comfortable with that. But anyway, they are smaller changes, smaller degrees of change. And then macroevolution would be the big changes between families or between even domains of life, the molecules to man part of it, right?
So we're talking about like, full-scale body plan changes and things of that nature.
Yeah, right. Bacteria to human kind of change. That's definitely macro. Now, of course, the dividing line is hard to define, and it's not so clear. If you look at Michael Behe's work over the years, if you take the taxonomic scale, or you take the taxonomic levels of species, genus, family, blah blah blah, all the way up to domain, it'd be nice if you could cut a line there somewhere and say this is where it cuts off. Well, Mike Behe's doing a great job of basically coming to a cutoff point, which is somewhere around the family. The tax of family is where evolution seems to run into a brick wall and can't go beyond that. Now, unfortunately, this micro and macro, often there's a kind of bait and switch that goes on there, because the micro changes can be shown with high confidence. You can see them repeatably. You can directly observe them. They're right in front of you, and they're very hard to refute. Who wants to refute that? Because it's so obviously true. But it's used as kind of a bait and switch, that because the micro can be seen, that must mean the macro is true. But that's a gross, it's an exaggeration, but it's an extrapolation. Extrapolation makes me very uncomfortable as a scientist. And to extrapolate from the micro to the macro it's just not something that I can swallow, but it's often handed to us as a dish in media and papers all the time.
Well, and I completely agree with what you just said. I mean, one of the things I find very interesting is the, as you said, how overblown the minor adaptations are in microevolution in relation to the requirements and the context of modern genetics and biology, known requirements that would have to take place in order for this macro change to occur. Yet that the gigantic chasm between the two is completely glossed over and ignored, or congratulations, you guys came up with an algorithm that these are all the changes that would have had to have happened, and then just assuming that they did.
And there's nowhere else in science where that kind of extrapolation is acceptable. It's just I've only find that happening in evolution, where you could take, for example, everybody talks about the species of dogs and how dogs have diverged through human intervention, but look at all these different types of dogs. And then they use that and extrapolate that to, well, that's how bacteria evolved into mankind. And it's a ridiculous extrapolation, a millionfold, that you'd never find that in science.
Well, let's talk about one of, quite honestly, one of the most touted examples of evolution that is used, which, in my opinion, with just ridiculous amounts of confidence by the atheist / evolutionary biologist crowd, and that is the Lenski E. coli experiment. Supposedly, selective pressure has shown the formation of brand new functions. Therefore, evolution is proven, and anyone who still has questions is just living in a full-scale state of denial. Now, don't give away everything you discussed in the book, but can you give the audience a kind of an overview of this experiment and what was actually quantified? Do they look more, were the end results more in relation to micro or macro evolution?
Yeah, great question. So this question addresses the field we call experimental evolution, which actually can meet all 6 of these criteria of high confidence science. And that's because it is a prospectively designed study that you plan out in your lab, and you conduct the study experimentally trying to produce evolution in the lab. And so Richard Lenski is really kind of an amazing scientist, because he spent 30 years of his life on one experiment so far, where he's taken E. coli, a bacteria. And the reason why you pick E. coli is because it's cheap, but it reproduces very rapidly. You can get 8 generations in a single day. So it's kind of a time acceleration experiment because they reproduce so quickly. And so in 30 years, he's gone through over 70,000 generations of bacteria, all in the lab. And this is all prospectively studied, and it can be done repeatably. He can freeze samples and then repeat evolution experiments and so forth. And there are many studies like that. Lenski's not the only one. But in his particular study, the most exciting finding so far, it happened after about 33,000 generations, where he found that the E. coli suddenly developed the ability to reproduce much greater, much faster. And Richard Dawkins, I'm sure all of your viewers are somewhat familiar with Richard Dawkins, but he made a big deal of that. He made a big deal of that there's a massive evolution that occurred there. And he claimed that it was a beautiful demonstration of evolution in action, with new information entering genomes and new combinations of genes that made this possible. But that was before they actually had published the papers on what exactly had happened by looking at the genome of these bacteria. So that when they dug into the genes and they dug into what happened, it turned out that the bacteria were now able to metabolize a chemical, a nutrient that before they could not metabolize. It was called citrate. The citrate was always there in their solution. They just weren't able to eat it, in the presence of oxygen anyway. So all of a sudden they developed the ability to eat the citrate. So all of a sudden there's much more food on the table, and of course now much more bacteria resulted from that. And so Richard Dawkins made a big deal of that, thinking this is a great demonstration of evolution in action. But subsequently a couple of papers came out where they looked at the genome before and after, and they compare the genome head to head. And what turned out is that one gene, which already existed in that bacteria, which could only be turned on in the absence of oxygen, allowed it to eat the citrate. But you had to have no oxygen in order to use that gene. Well, that gene got duplicated to a different location. The same gene, new location. And now it could be used in the presence of oxygen. And so now the bug, this bacteria, could now eat the citrate in the presence of oxygen. And there was one other change that happened that had amplified another gene, meaning another gene got expressed more frequently.
So if I'm understanding what you just said correctly, the capacity to metabolize citrate already existed.
Exactly.
However, the on-off switch was set to off.
Right. Because the gene is preceded by a promoter, and that's the on-off switch. Yep.
Right. So and then it's duplicated without the promoter, the on-off switch. So it's now defaulting to on. And now the limiter, which in this context was oxygen, no longer applies. Therefore, a pre-existing capacity is now being executed. Is that
right?
Okay.
Right. So no new information, no new genes. It's kind of just a rehash of what you've already had, just a little scrambled, a little changed. And that was the big evolution explosion that was seen in Lenski's experiment.
So one of the things I find very interesting about what you just said there is, in my opinion, what you just described is exactly what we're trying to accomplish with machine learning and artificial intelligence in modern dynamic systems. We're trying to say, okay, we've got an existing function. Can we make it possible for the system to modify this pre-existing function on the fly based on new variables without having to go back and rewrite the code, for example, from scratch, right? Or come up with an entirely new function to account for this.
Right.
Or we can have one in the background that can be activated if we've already accounted for this potential variable. And to me, what you just described there literally fulfills those types of trying to account for these types of variables.
It's a lot easier to reuse some code you've already generated than to come up with a whole new invention, a whole new creative code.
Right. I find that very, very intriguing. The more we learn about these sorts of things, to me anyway, the more it seems that we're just discovering some of the most ridiculously amazing artificial intelligence, if you will, that's ever existed, and vastly superior to what we're coming up with right now. And yet we're supposed to conclude that this happened through undirected process. And to me, it blows my mind sometimes. But no, as a PhD, you've challenged the status quo. So I can only imagine, the pushback you've received on this next question I have for you. But I think this is something that, the average person really, they need to become aware of. And that is that the rapidly growing evidence that our genome isn't quite as similar to chimps as we've been told. And, you make this an entire section of the book. And, to me, it's amazing, what we're told about, the chimp human comparison. It's 100% proven beyond all doubt fact. But it actually doesn't come close to meeting the criteria of high confidence. Can you kind of expound on that, maybe starting with how and what is actually being compared?
Yeah, that's a lengthy question to answer. It's funny, I think, if you talk to anyone on the street and just ask them what's the similarity between human and chimp DNA, they'll immediately spit out 99% or 98%, and it's so embedded in our minds because I guess the media has pressed that upon us time and again. And there is so many, there are a lot of errors, I think, that came into coming up with that number, and now it's embedded in society and very hard to pry us free from it. That number came from only comparing the parts of the DNA that encode for proteins, which is about 1.5% of DNA straight encodes for proteins. And then among those that encode for proteins, they only looked at genes where you could find the same gene in a human and the chimp, take those select genes that are basically the same, and then compare the sequence within those genes. And that's where they got this 98.5%. But there are a couple other errors there as well. The chimp genome, actually, when they produced the genome of the chimp, this early version anyway, they used the human genome as a scaffold. To sequence the genome, they break up the DNA into lots of tiny little pieces, and they have to glue the pieces back together. And they took the human genome as a kind of scaffold to help you put the pieces back together again. And that inherently puts a little bit of bias in there. And there's even a fair amount of evidence that there was contamination of human DNA getting into the study, getting into the chimp DNA. So there's a lot of questionable parts of that. Now they're making better chimp genomes over time. The most recent one is called Pantro6. It's the latest chimp genome. And people like Richard Buggs and Jeff Tompkins have been looking at that, and they're coming up with a number of about 85% similarity between the human and the chimp. something in the mid 80s. So unfortunately, I don't think that number is going to end up embedded in the mind of humans because we're all stuck with 98 now.
Now, are they in with Tompkins and Bugs? Are they that 85% number? Is that still in relation to just the coding regions, or is that a like comparing the genome in its entirety?
It's trying to compare the genome in its entirety, and it's very difficult to do that. It's very difficult to come up with one number, but the take-home message is that there are a lot of differences in there, and it's not what you've been told. Let's just say that.
And something I find interesting about that is even if the 98.7% or whatever the standard talking point is percentage-wise, even that I think comes out to something like 35 million base pair differences.
Right.
Which, it's
a lot of information.
When we think about it in the relation to, it's only 1.3%, like it's so similar. Yeah, folks, we're talking about 35 million base pair differences, and then in the relation to me anyway, in relation to the evolutionary argument, you're talking about things that one mutation can cause a frame shift that has complete cascade failure. I mean, so you're we're talking about 1 point mutation can cause you to die, and we have to somehow have had 35 million of them happen correctly.
And all be positive, all be good. Yes.
Right.
And you have to do that within the 6 million years, supposedly, that separates, our common ancestor with chimps and humans is about 6 million years. So you got that much time, and that's called the waiting problem. You only have that much time to get this new information accumulated.
Well, and let's talk about that, because you actually you bring this up in the book. When you actually are consider these differences, and just the ones that we know or currently aware of, even if we did have a common ancestor, there literally haven't been enough potential generations, I think is the way you placed it or framed it, in the past 6 million years to plausibly, I think that's an important word to put in there, plausibly account for them. Can you expand on what you were just mentioning, that waiting or the timing problem? Can you give us a little more detail about that?
Right. So, experiments like Richard Lenski's that we described and others like that, which do meet all these criteria for high confidence, in my mind, they help to put a bound, a constraint, a clear bound on how much evolution can accomplish. And when you look at his 70,000 generations of bacteria, if you translate that into the life cycle of a human or a chimpanzee, you're talking about a million years of time. 70,000 human generations is about 1 million years. So Lenski has shown us it gives a hint of what you could accomplish in 1 million years of human or chimp lifetimes of evolution. And as we said, his study, quite in contrast to what you hear about Lenski's study in the popular media, his study actually shows that almost nothing of substance or new information or new genes developed in those 70,000 generations. So you apply that to the human scale and to this question of, did we have a common ancestor with chimps? And creating those, as you said, 35 million base pairs of new information. And we're going to talk about orphan genes later, but there's something like 1,400 new genes that humans have that chimps do not have at all, nothing related to them. Where are those going to come from when studies like Lenski tell us that in 1 million years, none of that has developed? So how are you going to get all of that in about 6 million years? That's the waiting problem, as it's called.
So, yeah, the point you're making is something that, it really strikes me, because it seems that most scientists are just accepting that, well, the math doesn't line up, but somehow, and also the math doesn't line up from a evidentiary perspective, as you were mentioning in relation to like the Lenski experiment, but somehow this just happened because our assumption is that evolution is true. Am I misinterpreting that, or is there a gigantic portion of academics just making a massive assumption that is contrary to the observed evidence?
To me, the strongest evidence the strongest backing for evolution is the fact that it is a naturalistic explanation. It is the only naturalistic explanation. And for those who want to define science as a field that can only speak to naturalistic explanations, they've constrained science to sort of block out anything that isn't naturalistic. That leaves evolution as the only option, the only hypothesis. And that is really why it is so well supported, because it is the only alternative. It's not because the evidence is crying out, this is the right answer. It's because it really is the only naturalistic explanation.
Interesting. Okay, so that leads me to another question. I think it's going to expand on this problem in a very specific and quantifiable way, and that is that not only are there differences, but we're seeing a large percentage of the coding genes being, completely unique to individual species. I think you were mentioning that with, orphan genes. Can you explain, the top level of this problem and how? And correct me if I'm wrong on this, but I think that the more research we're doing, the bigger this problem is becoming for evolutionary theory.
Right. So, for evolution to be true, you have common ancestry. And so if you look at genes through lineages of living things, you should be able to see common ancestry in the genes, meaning the same gene appears here and here and here and here, same gene with some modifications. And so as genomes were sequenced from different life forms, it was quite a surprise to find that there are genes in the genome of every living thing. And we're talking about 10 to 15% of genes in every living thing that no one can find an analog of that gene in any other living thing. And they're called homologous genes if they're similar between one life form and another. Those are homologous. Well, in 10 to 15% of genes, they cannot find any homolog, any analogous gene in any kind of life form. And so that concerned people at first, but they figured, you know what, the more we sequence genes from different kinds of life forms, we'll find traces of where this gene came from. It's got to have a family lineage to it. And the more they do gene sequencing from all kinds of life forms, the more trouble this is growing into. So as you said, they're finding more and more orphan genes, so-called orphan genes, meaning they have no family lineage. They just pop into existence. And in fact, if you take a pool of all the genes of all of life, there are some that are conserved that are pretty similar, even from bacteria to human. They're similar looking genes. Those are a small minority of this massive pool of all the genes of life. And the orphan genes, which is 10 to 15% of all of genes in an individual life form. But when you pull them together, and it's 10 to 15% of humans, of chimps, of cats, dogs, 10 to 15%, the common genes that are shared become a very small part of it. And the orphan genes become the majority of this gene pool. And there really isn't a decent explanation of where they come from. They seem to just pop into existence. And I found it particularly interesting that Jerry Coyne, who you may know, is a very strong proponent of evolution, large-scale evolution. He wrote in his book, Why Evolution is True. It quite surprised me. He actually wrote in there his surprise to find that humans have about 1,400 orphan genes that you cannot find anywhere else in any living organism except in humans. They're unique to human at 1,400 genes. And it amazed me he said that, because it counters his own arguments in his book.
To expand on that, I'd love to ask you a couple more questions on that. So the other night, there was a debate, and there were different advocates of evolution who were strongly arguing that no. The more sequencing we're doing, we're finding more answers to the orphan gene problem. I know you've been researching this relatively recently. Is that a unaware talking point that's being spewed? Are we actually finding more explanations, more homologous gene sequences to explain orphans, or is that just a Fantasyland explanation?
So there are two possible explanations from a naturalistic standpoint of where an orphan gene could come from. One is that you take an existing gene and it gets duplicated. And then over time, that duplicate gene, because it's not essential, it can rapidly change and evolve into becoming some other purpose, to the extent that it's no longer even recognizable from its twin from a long time ago, right?
And is that co-option? Is that the term that's used for that one?
Yeah, in a sense, it's co-option, but it's duplication with modification. That's number one. And the second possibility is you have a piece of DNA that is not being used to code for a protein. So you could, maybe it's just a random segment of DNA that's just sitting there doing nothing. Suddenly it becomes able to produce a protein. So it becomes an open reading frame, and now it's coding into a protein. And so it's sort of a random shot of creating some protein with a random functionality, probably useless, probably detrimental. But eventually they're saying that could turn into a de novo gene that has some useful function.
Now, is what you're describing there, these hypothetical explanations, are they, in terms of the criteria, are they observable explanations, or are they hypotheses?
Well, it's some combination of those. You can observe new reading frames happening, and there are examples that they will show in the literature where they can see something like that happening. Whether it's as clean of a story as they make it out to be, I don't know about that. But probabilistically, you can just look at what's the chance of taking a random segment of DNA, say, oh, let's build a protein out of that. Well, it's extremely likely it'll be detrimental to the cell. Right. It could just get in the way, but it's extremely, like 1 with 70 zeros behind it, probability that, the inverse of that, a chance that it would be useful to the organism. It's most likely going to be detrimental.
Right. And okay, so part of the reason I'm asking this question, where my mind is kind of going, is it was duplicated and then modified to something that we now don't know isn't homologous to the gene it was supposedly duplicated from. That, correct me if I'm wrong on this, but that gives you literally zero observable evidence other than pure supposition of the origin of this new functional gene. Is that fair?
Yeah, it's the evidence that they try to build a story is from what's called synteny in genes. So you'll see in one organism, you'll see gene A, then gene B, then gene C, then this area, another area, and then a gene D. And then you take another animal and you see A, B, C, D, and you're saying, oh, this area now is a gene in this animal, but below it wasn't. So there's a break from A, B, C, and D. There's this new thing showed up in there. So that gives them a hint of evidence that maybe something developed. But that story that they tell has as its foundation the assumptions of large-scale evolution, right? It's a house of cards built on those assumptions that evolution is a fact already,
right? And
then they can build a story based on that.
Now, but that, and the reason I'm asking these clarifying questions is that is not a repeatable and observable conclusion. What you mentioned before in terms of, hey, we have seen new open reading frames happen, and then adaptation of something we thought was non-functional into something functional. Mm-hmm. But that is not following, correct me if I'm wrong on this, but what we're observing in that context is more of a almost real-time adaptation versus a long drawn-out process. Is that fair?
Well, what I described was something comparing existing genomes from today and kind of inferring the past because of evolutionary inference or assumption. going back millions of years, it must have developed this way to get to where we're at today. So there is a lot of historical story added to it, I would say. The other thing about orphan genes you'll find interesting is there are claims in the literature that these orphan genes are relatively simple and that they don't really have very important functions. And this is a prediction that I would make, is that the orphan genes are what makes each living thing unique. It gives each living thing its so-called superpowers, right? So in the human being, what we do that no one else does is our language, our big brain, our consciousness, our ability to think. That's kind of our superpowers. And if you're able to trace that back to genes, my prediction is it'll be these orphan genes, right, that are giving you that ability. If you look at a cat and its very great agility that cats have, that you can drop them upside down and they turn around and they're incredible how they do that. It's gonna map back to orphan genes. So they're not going to be really simple little rudimentary genes just getting started. They're going to be these unique, powerful functions that each living thing has.
And I think something a lot of times the average person don't really think about is, a lot of times these genes, these orphan gene sequences that we're talking about, it's 300, 400, 500, 1,000, 2,000, 4,000, 5,000 amino acids in length that have to be accounted for in the sequence. And I think very often, because of the sheer stunning both complexity just in general, but also the improbabilities, the average person doesn't really contemplate the lack of plausibility in what is being argued as having occurred in the storytelling that's being created to explain it. Is that a fair point to be making here of like, hey folks, this is not like, hey, we tossed dice and landed, well, we had 6 dice and we threw them, oh my goodness, it's a miracle, they all rolled 6s. Like, no, we're talking about having, you have to have rolled all of these dice correctly for literally thousands of times in a row. Like, not just one dice in a row, you have to roll all these different dice correctly in sequence every single time, and if you don't, you have abject failure.
And let me explain. Let me answer your question by explaining an experiment that I really particularly like. And this is an experiment that meets all of those 6 criteria for high confidence because it was prospectively designed, it can be repeated and so forth. This is an experiment from Ann Gauger and colleagues. But they went into bacteria and they intentionally damaged a gene that's responsible for being able to manufacture a necessary nutrient called tryptophan. And they went in there and damaged the gene to sort of test evolution, to say, hey, if I just break it in this one spot, can evolution come in and fix that break? Now, the gene they're talking about, this enzyme that can produce tryptophan, it involves about 1,000 nucleotides of DNA. So 1,000 letters of the code of DNA. So they went in and they changed just one letter out of 1,000. And then they went through, just like Richard Lenski, generation after generation after generation, and they put them in a bath in a solution that had a very limited amount of this nutrient. So they could kind of survive, but there's a lot of stress there, and they have to fix this gene in order to really survive, right? So there's a pressure. And they found if they just changed one nucleotide, that evolution worked. It could replace that one nucleotide by random luck, and then you have something that can produce tryptophan, survive, outcompete its neighbors, and win. So they said, good, okay, we know evolution can accomplish that. Then they went back and they changed 2 nucleotides. Out of the 1,000, they just changed 2, and they did the same experiment. And even with 10,000 generations of E. coli, not only did it not solve that problem, it wasn't able to overcome that kind of barrier, but they actually found that the bacteria preferred to just eject the gene entirely because it was a waste of energy, a waste of precious resources to copy this gene that's useless. So what this is saying is, if you have this gene of 1,000 nucleotides, if if it can't repair itself with only 2 missing out of 1,000, where did the 1,000 come from in the first place? How could we say that that 1,000 nucleotide gene evolved from nothing if it cannot fix itself from 99.8% finished? It can't get to 100%. So the same thing applies to these orphan genes. How could they come out of nowhere as 1,000 nucleotides of, how can that come out of nowhere if I give you 99% finished and you can't get to 100% finished?
And that would, whether it be coming from scratch or a modified duplicate, the same logic would apply in terms of why would it not just be ejected?
Right. So very small changes are possible and have been shown with high confidence they can happen. But any kind of complicated redesign seems to be beyond what evolution can accomplish.
In line with what you're just talking about there, just a quick rewind to the Lenski experiment. When you were talking about the copy with promoter, without promoter, were there any modifications to the actual sequence itself in terms of the citrate metabolism? Do they know any significant modifications to the sequence itself, or just to the regulator portion?
What mattered was the changes to the regulator portion. When you produce 1 trillion organisms through this experiment, there are going to be random hits of errors and changes popping up like popcorn. But those are often neutral, insignificant. They don't matter. But here, the case of what mattered was all in the promoter, is where the so-called improvement came from.
Yeah, it's interesting to me how so many of the fundamental requirements that we're discussing right now, they get glossed over in the arguments related to whether or not evolution is the reasonable explanation for our very existence. Which to me, it seems, if we're going to talk about how do we exist, that you might want to look at some of those things as an important variables to be accounting for. But to kind of expand on this topic, you're a medical research engineer. You're diving into how things work and figuring out ways to fix them, and new ways to create function or to adapt function. And the more I learn about adaptations in microevolution, the more it seems these supposed proofs that we've been talking about, they're either activation or modification to a pre-existing function. And as you've talked about in the orphan genes, not formation of completely new from scratch novel features. To me, that just seems to be stretching plausibility to the nth degree. As a researcher yourself, do you see that same lack of plausibility and it's all imagination, or are we actually finding things that would explain some of the stuff that you and I have just been talking about for this last 15 minutes?
I agree with everything you're saying. I think going back to what I said, that evolution is the only naturalistic explanation, that's why people hold on to it. And that's why it's what we hear so much about. We hear only positive results about it. We haven't discussed yet another aspect, which is very well.
All right, folks, we had a little technical issue there. Thank goodness we got the good doctor back. We were talking about the level of implausibility that is having to be explained, if you will, to account for all these different complexity issues. And you were talking about Michael Behe in his latest book, the degradation and devolution rather than evolution.
Darwin devolved.
Observing.
Yeah, Darwin devolved. So it's in that book, and through many of his books, he speaks to the fact that most evolutionary changes are accomplished through actually degrading function rather than adding function. I like his analogy of, let's say your town is about to be attacked by an invading army. It takes a lot of energy and creativity to come up with some new weapon, right, to defend your town. But it sure is easy to just burn the bridges. And if you burn the bridges, you protect your town. But there's some side effects that aren't so awesome about that. But it's the easy way out. And evolution will always take the easy way out. It's so easy to burn a bridge, to actually break an enzyme or something to stop an invading army. And he's got great examples of things like chloroquine to get rid of malaria. And malaria breaks an enzyme in order to survive an attack from the antibiotic chloroquine. And so many examples like that are prevalent. So that's something we hadn't talked about before, but evolution is most often degrading rather than building up.
And obviously, to degrade something, you have to be operating from a higher level starting point, right?
Right. And after that offending army is gone, you're actually at a disadvantage, and the wild type organism will end up beating you out. And that happens time and again with these single cell organisms that face antibiotics and so forth. They end up worse off than they were originally.
I'm trying to remember where I came across this, but I'm drawing a blank on the exact source of this. But I had come across something where they were showing that, bacteria, as an example, actually have the ability to raise and lower the fidelity of their replication.
Mm-hmm.
And basically in new disadvantage or non-advantageous environments where they're actively needing a solution to survive, it seems that they magically have a lower fidelity rate than when they're in a stable environment. And I found that. Have you come across anything like that? I thought that was a very fascinating concept. Like, wait a minute. So they actually have a variable that enables them to have a greater probability of finding the correct solution. That's cool.
Right. And that's another aspect of this whole thing, is that a lot of changes to DNA are actually directed in response to a stimulus. So it's not so much of a random, there's completely random processes, and then there's a spectrum of how much it was directed to be changed. So there are certainly many cases where some kind of a stimulus comes in, and the organism is designed to respond to that stimulus by changing the DNA in some way that's advantageous. It's like it's built in, it's designed or engineered into the system to be able to live or overcome that kind of stimulus.
I tell you, for those watching, if you've never looked into this, I strongly recommend on YouTube right now, when we get done with this, get done watching this interview, search for antibody formation animation. I remember the first time I actually watched a breakdown of like the actual process for antibody formations. And I'm like, wait a minute, this protein has fixed stable structure regions, and then there's a variable component that can be modified in real time, basically on the fly, until they find, the immune system finds the appropriate one shape to bind with the current antigen. Like, what?
Fulfill a very specific purpose. Yeah.
Right. I mean, when I came across that and really looked and saw visually, like the process that was being executed, I was just blown away. Like, wow, that's amazing. But then when the point you were making about the engineering of like, this is like, I don't understand how you cannot come to I don't understand why the rational conclusion would not be, wow, this is a pre-programmed antivirus system that we wish we could create to protect ourselves from in computers, for example, of hey, wait, that's exactly what we're trying to do.
With human intelligence.
Right. And I don't think there's anybody on this planet who would ever conclude that, McAfee antivirus software would ever have existed without John McAfee originally having created the original version. And then obviously from there, other highly intelligent folks have expanded it. And
great analogy.
And obviously, since you do research in finding solutions to health issues, I'm assuming you probably run into all sorts of things on a pretty regular basis that are fascinating examples. Other than what I'm talking about in the immune system, is it fair to say that you find all sorts of stuff like that over your 23 years of research?
And that's what I enjoy so much about studying physiology. Just every time you peel back the layers of complexity of life, you find some amazing engineering. It's just you take any piece of life, look at ATP synthase, just look at that thing, look up YouTube examples of videos or animations of it, and you just marvel at that thing and what it can accomplish with.
I did I had a live stream a couple weeks ago where let's just say I was rather irritated with some different opponents, shall we say, talking about how all these things were just nothing but chemical reactions. And I'm like, okay, fine, let's go observe what are nothing but chemical reactions. And I agree with you. I think it was Harvard did a very interesting in detailed animation of ATP synthase. And to me, when you really look at the different mechanical components that are required for that process to exist, to conclude that all of those interdependent parts somehow just magically pops together. I mean, to me, that's taking the 6 criteria of high confidence and basically tossing it out the window.
And almost everyone would agree that ATP synthase had to be present in the very first life.
Well, folks, we had a little bit of a technical issue. First, it was Dr. Stadler's fault. Then it was totally my fault. But now we have managed to come back. As we're wrapping up our great conversation today, we've covered a plethora of amazing, amazing topics. And, as we come to an end for the day, I'd really like it if you could, can you share with the audience, what are some of the top 2-3 things that, if people don't and go look beyond any of, just these factors, what are the things you would recommend folks go and, look beyond the surface and, it may, if they do, it potentially will shatter their confidence in evolutionary theory as an explanation for their existence.
Well, we mentioned some of them already with the orphan genes and with these studies of experimental evolution like Richard Lenski did. And when you dig into those and find the gaps that exist between organisms and then what evolution actually can accomplish when these 6 criteria are applied and you have experimental evolution, you can see what evolution is capable of accomplishing and how constrained it is versus the gaps that exist between living forms. I think that's very telling. And then a third piece of it is, it's related to that. But if you look at the gaps between eukaryotes, which is one domain of life, and prokaryotes, another domain of life, the evolution story will tell you that the prokaryotes evolved into the eukaryotes. But if you actually look into it and you look at their DNA reproduction methods and the translation of the DNA and transcription, you'll find fundamentally different machinery that's not like an improvement on the old theme, but it's a completely different process that cannot be related. These are unbridgeable gaps. And so that story of one evolving into the other one is quite a challenge. And digging into that is what changed the thinking of my co-author on my next book. Her name is Changed Tan. She's a professor of biology in Missouri. She was an atheistic evolutionist, and digging into that is what changed her mind about this, seeing those unbridgeable gaps.
It's interesting, you're talking about all, the replication process, just as an example. I find it so interesting how, in the storyline of prokaryote to eukaryote, and, something as fundamental as mitochondria supposedly are accounted for by endosymbiosis. And, to me, when you really go under the hood, as you're talking about, and you've run into these gigantic barriers to entry and the bridges that, hang on, this isn't a remotely plausible gap to be bridged here. to me, that really just challenges the entire fundamental foundation of evolutionary theory that is accepted by huge portions of academia as being proven. I mean, would you agree with that position? Like, it's people just accept that it's proven, but don't actually go and look and to see if it actually is proven. And I mean this not just in terms of laymen like myself, but I mean your peers.
Unfortunately, I think people are quick to brush off evidence that doesn't agree with what their preconceived notion is. And it's so important to emphasize that what we're talking about are unbridgeable gaps between essential functions like reproduction of DNA. You can't live if you can't reproduce your DNA. But to take a replication process here and get to this one, which is completely different than this one, you have to keep the organism alive. It's like saying I want to change my gas engine of my car into an electric engine, except you have to keep the car running the entire time. You can never stop, but you got to pull out the old engine and put in a new motor. That's what we're talking about, and it's just not possible.
That's actually I'm going to steal that one if you don't mind. That's a very good thought experiment there of like, seriously, folks, how are you managing to maintain the thrust requirement to
while making these major changes? Yep.
Yeah, like you there's no way you'd be able to have the combustion requirement. Like, you would have to have a break at some point. Right?
A really simple example in prokaryotes, there's a protein called DNA, and that is required for replication of the bacterial's DNA. And it starts the replication process. And that's a single protein in a prokaryote. In a eukaryote, it's a complex of 6 different proteins that come together to form a molecular machine. And it's there's no relationship between DNA A and this new complex of 6 proteins that gets the job done in the eukaryotes. So there's this unbridgeable gap, and you got to have this process or else you die. So how are you going to get from A to B?
So here's something I'd be very curious about is, I don't know, this has been experimented on. Has anybody tried to take a DNA and have it do transcription for eukaryote DNA and vice versa?
It can copy DNA, right? DNA is just 4 codes, and it's the same. Right. That's the same in all of life. But it's the translation of those nucleotides into a protein that's slightly different. You know, we're told that the code of DNA, translating that into proteins, is universal in all of life. But in fact, you look it up, there are 33 different codings that have been found in life. Some of them are very subtle, but they are different codings, to the point where in one organism, a triplet of nucleotides will code for tryptophan. In another organism, that same triplet will code for stopping the process. So how can you get from here to there and still have life? It's a wholly different code.
Well, and something else about that concept that I think just a significant percentage of evolutionary biologists refuse to even consider is A, the point you just made, but B, the fact that it can code for 2 different things completely eviscerates the whole, oh, it's nothing but chemical reaction, in terms of how were the values assigned to the code. And to me, that just, I can't tell you the number of arguments I've had about this exact topic. And I'm like, code on the point you're making, like AUG equals start in this organism, it means, amino acid X in this organism. How are you trying to argue that this is somehow explained purely by I'm talking about the value assignment and how it's being translated? Like, how are you explaining this to be nothing but a chemical? Oh, they must have some chemical, affinity for this. Like, well, apparently there's an affinity for 2 completely opposite outcomes. So I'm not really sure how you're explaining this outside of having to have a separate translation mechanism which can interpret the alternative meaning.
Because the code and the decoding process are both essential. And to do what you've suggested, you have to change the code and change the decoding process simultaneously in order to continue to have life. Otherwise, you'll break one of them and you die.
Right. At that point, you have complete cascade failure all the way down the line. It's that, and something else I find very interesting is humans. We've actually been able to do the reprogramming that you're talking about. Like we've managed to pull it off ourselves
through intelligence. Yes,
through intelligence. And I'm like, okay, so we now have, and I think this goes back to the points about the criteria for high confidence. Like, okay, so we know beyond doubt, yes, we can reprogram it. But we also know the requirements for the reprogramming.
Right.
And to me, since we've been able to make it a repeatable and observable outcome, like we now know there is not a, slow change over time is not a viable option through testable, repeatable observation. And therefore, to me, the fact that it's being, these 33 different codes that you're referring to somehow have come into existence through the process that we now know through our own experimentation wouldn't be viable is somehow still presented as a plausible explanation. That seems like to me like we've suspended there's been a dramatic acceleration of bias in terms of and suspension of let's pull it up real quick, the suspension of sober judgment of results. I mean that
right.
I feel like that's where we're running into, but that's just me. Now, before we come to a close, I know you and Dr. Changed your other book, Stairway to Life, came out this year and back in March, and you guys really dove into the 12 steps, the process required for abiogenesis, and just origin. I did a great and detailed review of where origin of life research is right now here in 2020. There any before we wrap up, any things that you have that you came across in that research that really stuck out to you?
Yeah, well, it's a book that's listing the requirements basically of getting from chemistry, which is simple molecules, to biology, which is a living organism. And we kind of look at it like you're trying to build something here, trying to build from chemistry to biology. What would it take? And we break it down into these 12 required steps to get there, and then look at each of those steps one by one and find out what are the chances that natural processes could have met that requirement. And
to tease the audience a little bit of why they should go and read this book, correct me if I'm wrong on this, but where things stand right now in origin of life research, they haven't really even made it past step one. Is that a fair?
Yeah, there's a piece of step one that I think they've made it through, but the rest of the steps have not been demonstrated in the lab. And what's, I think, perhaps most interesting is that there's still a lot of life that we don't yet understand. Even the simplest of life, there are layers that we haven't peeled back yet to understand. And as we understand further what it takes to make even the simplest of life, additional steps will be added. So the goal of getting from chemistry to biology is pulling away much faster than any kind of laboratory prebiotic simulation is progressing.
So in spite of the, I'll just say it, level of propaganda that's being put out of, hey, we're getting closer and closer to understanding life, while we may be accounting for a few little minor components, the result of that is realization that we actually understand less than we thought we did, and that the overall requirements are actually getting greater and greater and further away.
Exactly.
Okay.
And their approach to overcome that is to try to simplify life and to define life as a much simpler state than a cell. So redefine the goal to make it easier to get to the goal. Yeah.
Well, that must be nice to be able to change the rules. Hey, here's what we actually observe. Here's the game we're actually playing. But we're going to claim that the game that we currently are engaged in, this wasn't the original rules. That's interesting. Well, Doctor, I greatly appreciate your time. We overcame the technical issues. I'm glad we were able to finish up our conversation. I hope we can have you on in the not too distant future to discuss some of these subjects in much greater detail and continue to pick your brain on some of the different aspects of evolutionary theory, origin of life, and just how all of this applies to the ultimate questions we need to ask ourselves, which is, hey, how do I exist? And as I love to say, what is the most logical, plausible, and probable explanation for our very existence? And what are the requirements for it? Is it undirected process, intelligent agency? And if it's intelligent agency, from there, obviously you can extrapolate out, what does this mean in terms from a philosophical perspective? I think these are things that, in modern society, we get so busy with our day-to-day lives, we kind of forget, hey, maybe I should actually ask these questions for myself and not just accept the dogma that is being presented as undisputed fact. And if you dare to challenge it, somehow you are not actually a rational individual.
Just be open to follow the evidence wherever it leads, and this is where it'll take you.
Awesome. Well, folks, Dr. Rob Stadler, thank you so much for your time. Make sure you go check out the description. I got links to both of his books. The one we discussed today, The Scientific Approach to Evolution, What They Didn't Teach You in Biology. Awesome book. Strongly recommend it. Go check it out. And then once you kind of whet your appetite, I'd recommend going and getting the Stairway to Life, because you better be prepared mentally and philosophically for the challenge that will present to your very existence. Doctor, thank you so much for your time. Appreciate it so much, and hope we can have you come on again soon.
Thank you, John. My pleasure. Looking forward to it.
If you like this type of content, be sure to hit subscribe and also smash that like button. It actually does help. Thanks for watching, and I'll see you soon.